Good morning, everyone. Very pleased to welcome you to the second day of our meeting. I have some announcements to make before we get started. So first, there were 200 to 300 people online yesterday, and we expect a similar number today. I thank all the people online for attending. And I want to remind you, please use our Twitter feed and Gmail account to send us questions. And those links are on the main screen right now, and throughout the meeting they'll be on the side screens as well. Also, the NIH institutes, who fund much of the work that you do, have prepared fact sheets about their interests in microbiome research. And those are on the table by the registration desk. So please stop by and pick up copies. Okay. We are very fortunate to have Jeff Gordon here to enlighten us this morning. Jeff is truly one of the founding figures of modern human microbiome research. Those of us who are old enough have been reading his microbiome papers for about 20 years, and all the younger people are trying to catch up. We have all been influenced by Jeff in how we design our studies, and just very basically how we think about the microbiome. And I think the most important route of Jeff's influence on the field has been through the people whom he has trained. And I would just like everyone who has spent time in Jeff's lab to stand up. Okay, now keep standing for a minute. Now you'll notice a number of the speakers among the people who are standing. Is everyone here who has or is currently spending time in the labs of these individuals, could you please also stand up? You didn't bring your labs with you? Okay. Okay, well, okay, you can sit down now, thank you. You won't have to remain standing through Jeff's entire lecture. The people you see, the people who stood up are familiar to all of us and have transmitted the wisdom of the microbiome and are continuing to do so, as are the people they're training today. And so Jeff, in many ways, has shaped the entire human microbiome research field, to which we all owe him a debt, and our presence here. So without further ado, I'd like to welcome Jeff to the podium to speak to us about the gut microbiota and childhood undernutrition.
Is Steve Banyan underneath there, by the way, or not? Well, thank you very much, Bob, for your kind words. And just in the spirit of what Bob was saying, there's an African proverb that I think captures the spirit of science. It says that if you want to go fast, go alone. If you want to go far, go together. And it's been my great privilege to be inspired by a magnificent group of students, postdocs, staff, and collaborators over the years. This is a snapshot of the lab at the time present. This is their story, and I'll highlight a few of these individuals during the course of the next 2.5 hours that I'm going to be speaking to you. Is that right, Lyda? We're funded in large part for this project by the Bill and Melinda Gates Foundation, which I think has had an enormous impact on science. It has inspired students to look beyond self, beyond family, beyond community, even beyond nation, to look at the conditions that human beings live in this defining century for humanity, where we are tested, our capacity to evolve psychologically, sociologically, and even biologically to the point where we can care for one another and respect one another. We can promote human flourishing is tested. So I thank the foundation not only for their funds, but also for the hope that they bring to all of us. I'm going to make one more comment before getting directly into my presentation. This is an inherently interdisciplinary field. It brings together people with different sets of lenses, looking at a set of problems that are very vexing, very challenging. That bringing together, that coming together is a precious moment, not only for the participants, but also for the universities that are fortunate enough to attract them. And it creates an opportunity to evolve the way we do science. I really believe that the field of microbiome research creates an opportunity for experimentation, not only educationally, but also in the structure of science. And the bringing together of people into ecosystems where high-end technology is democratized, made available on the bench, so that students can pull a hypothesis out of themselves, design well-controlled experiments, go directly to the machines themselves, look underneath the hood, gather the data, the massive amounts of data, and in order to mine the data, inform themselves about what algorithms currently exist or what scripts and algorithms have to be developed to be able to go to a cluster nearby and to be able to queue up their own jobs, install their own algorithms, receive the data that they've mined, and do the next experiment. That scales the way of thinking and doing in science. It's a precious opportunity to bring together people who can co-evolve computational and experimental biology. And I think one of the major focuses of this field, as we speak with common and dissimilar voices within our institutions, is to encourage our institutions to make those investments in these special ecosystems where direct access to technology, not only sequencing technology, but mass spectrometry and other technologies can be experienced. Create Edison laboratories where people can tinker, can think beyond what's currently available, and can know their own data. I think that's the key to success in many respects for the scientific journey. So, as I said, we live in a very challenging century, and I won't make any political statements about the challenge of the moment. But one of the most vexing, tragic challenges that are faced deals with the healthy growth of children and childhood undernutrition, which is the leading cause of death worldwide, children under 5. There have been a lot of epidemiological studies that have shown that the risk for and manifestations of childhood undernutrition are the result of a number of interacting factors, not food insecurity alone. There have been remarkable improvements therapeutically in terms of treatment of severe acute malnutrition, where ready-to-use therapeutic foods are administered for short periods of time. Children gain a certain amount of weight. They are given nutrition. They and their families are given nutritional counseling, and if the infrastructure of the healthcare system is sufficient, they can be followed up. Typically, unfortunately, that is not the case, but nonetheless, these therapeutic foods have reduced mortality but have not repaired the long-term sequelae of undernutrition, which includes persistent stunting, neurodevelopmental abnormalities, immune dysfunction. So we're not treating systems in a way that restore them to full functioning. And what might those systems be? And the hypothesis that we're pursuing is described in the current slide. So it postulates that the gut microbiota has characteristic and shared features of its developmental program. That this healthy development of the microbiota is inextricably linked to healthy development of infants and children, and that there's a disruption in this developmental process that's causally related to the pathogenesis of undernutrition. And in the upper right-hand side, you can see and envision some of the consequences of disrupted maturation of the microbiota. insufficient execution or deviant execution of this program could influence the ability of enteropathogens to find niches or jobs in the microbiota, could affect the co-development of the gut mucosal immune system. That impaired development also could affect enteropathogen burden. If the developing microbiota is able to produce a variety of signaling molecules and metabolites that are instrumental in the proper development of host cell lineages, impairment in this developmental program could affect organs well beyond the gut. So that's the hypothesis. The subtext is that if we knew how to be good microbial farmers, we could have a way of not only monitoring the trajectory of development of the gut microbiota, but to install a series of prescribed steps that may ensure the healthy development of this organ. And I think all of you who are fortunate enough to have children know some of the dilemmas that are faced, including the decision after a period of exclusive milk feeding, what kind of complementary feeding practices you should apply to your child. So let's go through the journey that all of us go through, which is defining normal in one population, determining whether that definition of normal generalizes to other population, developing metrics to quantify deviations from normal, determining whether those deviations from normal are significantly associated with disease. If there is a significant association, to at least preclinically begin a journey to determine whether those deviations are simply effects or causes of pathology. And if causality is established, then to design interventions that could repair dysfunction, this dysfunction in a safe way, both in a short term and a long term, and do so perhaps in ways that generalize across different populations. If there are deviations from normal in terms of this developmental program that are causally related to childhood undernutrition, then the ability to repair these deviations from normal, this dysfunction, may have long-lived consequences to the metabolic, physiologic, and even immunologic and neurological phenotypes of the child. So we turn to Bangladesh and our wonderful collaborators in Bangladesh. This is a slide just to give you a few definitions. We'll define stunting based on anthropometry as a height for age z-score more than two standard deviations from a WHO reference cohort. Underweight, weight for age z-score, wasting WH z-score, and I'll say that SAM, moderate acute malnutrition, will be defined arbitrarily as a WHZ score of more than -3, greater than 3 standard deviations. Moderate acute malnutrition, -2 to -3. So defining normal in Bangladesh, we turn to an urban slum in Dhaka, Mirpur, about 50,000 people living in approximately 5 square miles. Our wonderful collaborators at ICDDRB, the International Center for Diarrheal Disease Research, led by Tamid Ahmed. The trusts of the mothers for the healthcare providers at ICDDRB is enormous, and that's, of course, absolutely key for one important facet of microbiome research, the ability to do time series studies and also long-term follow-up of interventions. So a birth cohort was studied, living in Mirpur. Those individuals who had consistently healthy anthropometry were selected for an analysis of their monthly fecal samples, initially by 16S sequencing, although ongoing efforts are using a whole host of other tools. But let me just focus on the question of who's there as a function of time and their relative abundance. Applying machine learning algorithms, random forest, as data set, you can identify the most age-discriminatory strains, develop a sparse model of the most age-discriminatory strains, and plot the results in the term of a heat map, or in the form of a heat map shown in the lower right of the slide, where each row represents one of these age-discriminatory bacterial strains. We're looking at the first 2 years of life. Each column is a monthly age bin, and you can see the changing patterns of representation of these 24 different taxa. If you take a vertical slice through this heat map, you could look at the relative abundance of these age-discriminatory strains and assign an age or state of maturation to the microbiota. This age of a microbiota, calculated in this fashion, is very correlated to chronological age. And I'll give you some data about that. So when we developed together with Tamid, and the person who drove that analysis was a very talented MD PhD student in the lab named Satish Subramanian, whose picture is shown in the lower left, a number of questions immediately emerge. Is this model generated from the children of Mirpur generalizable to other populations? So the children of Mirpur have this shared feature defined in this fashion in community assembly or succession. How about other birth cohorts at other sites in one of the Gates-funded network of birth cohorts distributed around the globe representing individuals who have different cultural traditions and different culinary traditions. What's the relative maturity of the gut microbiota in children with SAM compared to those who have healthy growth phenotypes? And then finally, are these age discriminatory strains simply biomarkers of the succession that occurs in this and other communities, or are some of them mediators of healthy growth? So I will tell you that these OTUs are distributed across populations, and we've created random forest-derived models of gut microbiota development from a number of sites. They're shown in the upper left of this particular slide. And there's a plot of chronological age on the x-axis in the upper left-hand corner and microbiota age in the y-axis. And this is the Bangladeshi random forest model applied to members of a cohort, birth cohort, with healthy growth phenotypes living in a community in India. And the correlations between microbiota age and chronological age are shown in that particular case, 0.75, but all these reciprocal comparisons. So you have country-specific or site-specific RF models applied to the population living there, but also to other populations. And we see in this fashion that there are shared features of this community development. So we defined normal in one population. We looked for its generalizability in other populations, in this case, children with healthy growth. And now we can develop a metric for quantifying deviations from normal, which are shown in the lower left. So this is Satish Subramanian's idea. So the children of Mirpur who had healthy growth, what he did was to use this random forest-derived model and compute their microbiota age each month for all the children, develop a median or mean value and a standard deviation. You can see the correlations. And for those of you who are not red-green colorblind, there is a red dot on the lower left-hand graph. That is a child with severe acute malnutrition who presented to the nutritional rehabilitation unit at Dhaka Hospital with a chronological age of 18 months, but a microbiota age of 8 months. So this child has significant microbiota immaturity. And this is not unique to this child, as you'll see in a moment. Children with SAM have microbiota immaturity. What's quite remarkable is the failure of current therapeutic foods to repair this. Of course, those foods were never designed with knowledge of the normal program of maturation of the gut microbiota. So this is a clinical study that was done by our colleagues at ICTDRB. Satish asked them to provide samples of children with SAM prior to any treatment. They were randomized to a peanut-based, ready-to-use therapeutic food and a locally produced therapeutic food called Kachiri Hawa, which is rice and lentil-based that has powdered milk and micronutrients. And these children were treated as is custom for a couple of weeks until they've gained a certain amount of weight, and then they were followed following discharge. So this is the calculation of the MAZ scores. This is a healthy cohort of children. The value is around 0. This is the results obtained from the Plumpy Nut. This is the name of the RUTF arm. They present with significant microbiota immaturity during treatment, no effect, maybe a slight improvement post-discharge, and in this case, somewhat of a regression. Kachiri Hawa also failed to repair this microbiota immaturity. So these children are walking around with a persistent developmental abnormality affecting one of their organs. It happens to be the gut microbiota. And they also have persistent stunting. They have persistent abnormalities affecting bone, etc. So I said something earlier on about the importance of being able to study a cohort of individuals at a site where there's enormous trust between mothers and the healthcare providers. And that's certainly the case at ICDDRB. So this is the result of a follow-on study that we found to be very informative, which was a study where children were randomized to 3 different types of RUTFs. They were followed prior to, during treatment, and then for 12 months after discharge. Monthly for 6 months, and then every other month for another 6 months. And you notice there was a blood sample obtained together with a fecal sample at the time of enrollment. There's also a blood sample at the time of discharge and a third blood sample at 6 months post-intervention. And these are WHZ scores, WAZ scores, HAZ scores, and you can see that there's no effect on stunting. These children are severely stunted. There's some improvement, but not to normal in WAZ scores and WHZ scores. These children, this is their MAZ scores. This is with SAM. This is post-SAM MAM. These are these children during this period of time. They have persistent microbiota immaturity. So you can learn a tremendous amount about the biological features of these children at these 3 points. It's a time series study, so each child represents their own control. I think one of the needs of the field is to be able to have reasonably high throughput quantitative serum proteomics. And we've been extremely captivated by SomaLogic SomaScan technology. I don't know how many people are familiar with this. It's expensive, but you can monitor the abundances, the levels of 1,305 serum proteins. The technology is going to advance. How accessible it will be is a matter of conjecture at this point in time. But I thought that I would spend, as I said, of the 2.5 hours I've been allocated, the next hour and a half going through that matrix of proteins, where we've correlated the anthropometric indices, serum metabolites, and a variety of other features with the representation of proteins to get a whole host of biomarkers. And those have been extremely informative. And I just give you a few examples. Carrie Couarden is very interested, she's a postdoc in the lab, in the effects of the gut microbiota on bone biology. You can see these children come in with a biomarker of increased osteoclastic activity. That's the CTX, or C-terminal telepeptide of type 1 collagen, shown in the lower left here. After treatment, that value is reduced on average, 6 months of follow-up. An osteoblastic biomarker, procollagen type 1, propeptide, doesn't change. IGF-1 improves. Serum leptin improves at the time of discharge, but regresses. There are a lot of alterations, but we can see that there are many facets of host biology that are not repaired by these RUTFs. So our indicted co-conspirator and John Rawls can testify to the extraordinary nature of this individual, Chris Newgard, and the importance of targeted mass spec. I know that the ability to do non-targeted mass spec is very alluring in this field and important, but of course you need the chemistry backup to be able to identify M over Zs that you don't feel very confident about. But in this particular case, in a hypothesis-directed manner, we, together with Chris, looked at the metabolic features of these children. At enrollment, there's rampant lipolysis and fatty acid oxidation. At the end of the intervention, as shown in the slide, there's a sharp lowering of serum fatty acid and long-chain acylcarnitine levels and ketones, and also pronounced increases in amino acids and their metabolites, the keto amino acids. So they're switching their substrate utilization. And then 6 months after discharge, you start losing ground, amino acid levels decline. There is sustained lowering of fatty acids and acylcarnitine, so they haven't gone back to the rampant lipolysis. That may be because their insulin levels and IGF-1 levels are maintained above enrollment levels. So, just a flavor, define normal. generalizability of normal, a metric, MAZ score, for deviations from normal, the correlations between microbiota immaturity and severity of acute malnutrition, and phenotyping of these children prior to, during, and after current intervention. Is that immaturity an effect of, or is it causally related to the pathogenesis of undernutrition and its manifestations? Microbiota transplants were performed by some very talented individuals in the laboratory. We're going to move from Bangladesh to Malawi for this particular study, where we've also conducted analogous studies to those that I described in Bangladesh. In this case, chronologically age-matched children, 6 months, 18 months, in each age bin. Healthy growers are those that are stunted and underweight and have significant microbiota immaturity. Each donor microbiota into a group of young, just weaned onto a prototypic Malawian diet, and just weaned mice, and the mice are followed over time. I'll show you the experimental design in a second. The capacity to recover fecal samples and immediately freeze them is very important in our mind, and our colleagues at the ICTDRB are unbelievable. We use charged cryogenic canisters, liquid nitrogen temperature, that can maintain that temperature for up to 3 weeks. The average time at the ICDRB sites between the generation of a fecal sample and its immersion into that canister is 7 minutes, which for us is very, very important if we want to do metabolomics, RNA-seq analysis, etc. So not all sites around the world are capable of that, needless to say, although I think the protocols we've developed are generalizable. That also allows us to create colonial array culture collections, methods that were championed and developed by Andy Goodman when we were so fortunate to have in the laboratory. So each individual, we can capture a significant amount of the bacterial diversity in multi-well plates, where each well has a clonal outgrowth from a single starting cell, and sequence the genomes. And you can ask the question, does the intact uncultured microbiota transmit phenotypes? If so, is the culture collection transmit a phenotype of interest? If that's the case, you can go into a room and, maintaining humility, still shout eureka and voila, because you're on your way to dissecting out which organisms might be critical for transmitting those phenotypes. So we've published this, so I'll just go over this quickly. This is Laura Blanton, a graduate student extraordinaire, who is now at the Whitehead in David Page's lab. This is the experimental design. Again, the key elements: just weaned mice, prototypic diet of the population of interest administered, colonization, time series studies, and then at the time of sacrifice, multiple tissue harvests for a variety of analyses, including mass spec, take the bone out for microcomputed tomography, do serial quantitative magnetic resonance to also look at body composition as a function of time, and we have ways of taking mice out of the isolator and do MR and bring them back into the isolator without exposure to environmental microbes. Bottom line, there are transmissible discordant growth phenotypes in groups of mice that are fed the same prototypic Malawian diet, consuming the same amount of diet, but the immature microbiota transmit growth faltering, either defined by reduced weight gain, reduced lean body mass gain. There are abnormalities in bone growth, and also there's impaired metabolism evident in multiple tissues that can be characterized in part by a loss of metabolic flexibility. Laura took the fecal samples from the recipient animals, applied random forests, and asked whether she could identify growth discriminatory taxa, organisms whose representation correlates with lean body mass gain or certain metabolic features or even features of bone growth. And the answer was yes, and that was a very exciting time. She was able to culture these organisms, and for a number of them, put them together with an immature microbiota from a stunted underweight individual and show that their representation ameliorated some of these phenotypes that I just described. Okay, so that's a test of causality. I won't have much time to talk about this, but I think it's an important challenge for the field, which is to develop metrics for assessing the co-development of the gut mucosal immune system and the microbiota. So Andy Cow, who I was very fortunate to know when he was a postdoc in the lab and who is in the audience, I believe. Andy, did I see you? Yes, I think so, or not. There he is. Very modest, but very courageous. He was able to apply FACS sorting to segregate out IgA-targeted microbes in the gut microbiota from those that didn't have bound IgA. He was able to sort out these organisms into an IgA plus and negative fraction in ways that preserve the viability of a number of anaerobes. So you can take the IgA plus fraction and either enumerate who's there based on 16S. You could culture them as he did. But you also can introduce them directly into recipient notobotic animals, fed diets representative of the microbiota donor, and that's exactly what Andy did. In the case of SAM, severe acute malnutrition, so he mined the microbiota through the lenses of the host IgA response, found that an IgA plus consortium, which he subsequently cultured, could transmit an enteropathy to recipient animals, which is shown in the upper right portion of the slide. KM stands for kwashiorkor, another name for severe acute malnutrition. M, Malawi, IgA plus fraction. And you can see that disruption in the small intestine, epithelial cell adhesion molecule. There are antibodies to that. That's the staining. In the colon, we see this as well. This results in rapid weight loss, cachexia phenotype, sepsis, and lethality. And Andy found that he could mine the microbiota of a healthy child using the host IgA response and identify some organisms, Clostridium scindens and Akkermansia muciniphila, that could oppose the transmission of this phenotype. That leads me to this slide, which I think is important for a number of reasons. Largely the spirit of the slide, because it's not a densely fact-driven slide, which is how do we explore the microbiota along the length of the gut? It's still inconceivable to me that we haven't been able to recruit bioengineers who've been able to create capsules that function autonomously or semi-autonomously to sample the length of the gut and to monitor a variety of biological features. I'm talking about metabolic features of the gut along its length. I know there are attempts to do so, but I think we should really encourage those. So in this particular case, I'm talking about biogeography along the length of the gut. I believe there have already been some talks related to looking at biogeography across multiple scales, mesoscales, micron scales. Gary is here. He spent a lot of his time in the last several years developing methods for looking at the degree of admixture of microbes in the gut, not only in the lumen per se, but associated with food particles and in the mucosa. And I think the results are fascinating and should be linked to a variety of hypothesis-directed studies where we look at the co-occurrence of organisms, not only in the way that's commonly done, but also the co-occurrence spatially. And use technology like Rob Britton's mini bioreactors to follow through with that. And those mini bioreactors, by the way, are terrific, I think, potential upstream tools to design and hypothesis-based way, hypothesis-based way studies in notobotic systems as well, on our way to human studies. But anyway, I digress a little bit and just want to talk to you about environmental enteric dysfunction, the loss of villus height, epithelial surface area with an associated infiltrate of immune cells in the lamina propria. EED is postulated to be one of the confounders that contributes to the persistence of stunting and other manifestations of undernutrition. There are a series of biomarkers of EED, but they have not been really explicitly validated by concurrent histopathologic analysis of the upper GI tract. In an extraordinary study that has begun that we're part of, done in Bangladesh, as well as Aga Khan University, called BEAD and SEAM. Children who are refractory to current interventions for undernutrition are being endoscoped. There was a lot of time and attention paid to getting ethical approval for this, but this endoscopy allows us to determine whether in fact they have EED and to look at the microbial consortia associated with the mucosa as well as free in the lumen and determine whether they transmit the pathology you see here, shown in human biopsies. So the correlations that are being tested are there EED discriminatory taxa in the proximal small intestine, and are those taxa seen in the fecal microbiota? Are there distinguishing features of the EED gut microbiota that are only evident in the proximal gut? And are EED-associated taxa shared across different sites? And can we do a test of causality by introducing microbial consortia that are associated with mucosa directly into notobotic animals? That's what's being done right now. And Vanderleen Kuhn, who's a MD PhD graduate who's in a program, a physician scientist training program where you fast track, and I know a number of institutions have this, individuals with MD PhD through their clinical training. She's a trained pathologist into the laboratory. So breast milk is the first foods. We're very interested in the relationship between breast milk, oligosaccharides, and the microbiota, and how the microbiota might transmit signaling molecules that might derive from these HMOs to the host. We published a paper last year together, our wonderful colleagues at UC Davis, where mass spec was applied to breast milk obtained from Malawian mothers 6 months postpartum who had healthy infants or those who were severely stunted and underweight. We found differences in the representation of a number of HMOs. We were very interested in the differences in sialylated HMOs. I know there will be talks about these things during the course of this meeting. But sialic acid is very important. The infant isn't able to make much. The key rate-limiting enzymes in sialic acid biosynthesis are expressed at low levels in the liver in the early months of postnatal life. The mother provides sialic acid in the form of sialylated HMOs. Mark Charbonneau, who's a very talented graduate student, took a culture collection that captured most of the bacterial diversity present in a stunted, underweight Malawian infant, put it into mice, and then took a prototypic Malawian diet and supplemented it with either sialylated milk oligosaccharides, but captured from the waste streams of food manufacturing. The whey waste stream contains lots of sialylated bovine milk oligosaccharides that look just like human milk oligosaccharides. And then another group of animals received a basic diet supplemented with fructooligosaccharides, which are put in a lot of infant formulas, or at least some infant formulas. And voila, he found that isocaloric diets given to these three groups of mice, consumed in similar amounts, showed markedly different effects on growth. That the SBMO enhanced the rate of lean body mass gain dramatically. That's shown here. And these are the control arms. This is microbiota dependent, not seen in germ-free animals. And the effects on host biology were dramatic. Effects on bone biology, effects on metabolic flexibility, the promotion of metabolic flexibility. So that's a whole series of studies about the mechanisms by which these SBMOs work in a microbiota-dependent way to affect so many of these features of undernutrition. But I'm going to finish up my talk talking about another therapeutic approach that illustrates a pipeline that may or may not be useful or informative to you, which is predicated on a hypothesis that traditional complementary foods are culturally acceptable, affordable, and available, and that they represent the results of many generations of empiric observations by mothers where they try to correlate foods they give their children to healthy growth. So why not try to mine the complementary foods that are commonly consumed in Mirpur and try to identify combinations of complementary foods that might promote the representation of these underrepresented age and growth discriminatory strains. So could we develop microbiota-directed complementary foods that would repair microbiota immaturity? If we could, then those complementary foods may be part of a more generally prescribed way of sponsoring good farming of a developing microbiota. So a very talented group of people in the laboratory, Sid Venkatesh, who's a postdoc, Jeanette Gehrig, Hao-Wei Chang, and Mike Barrett, who oversees many of our clinical studies with our collaborators, rolled up their sleeves, so to speak, and did the following. This is the pipeline that represents a marriage that we think is important. The marriage is between these preclinical models, food science, and human studies. You don't need a Hubble telescope. I'll just use in words what this thing shows. And that is, you take your targeted microbes, in this case, age and growth discriminatory strains. We're going to screen different combinations of complementary foods for their effects on the representation and express functions of these targeted organisms. We're also going to spike in the same animal models, organisms, bad actors that are enriched in children with severe acute malnutrition. We're going to get leads. We're going to prioritize the leads being complementary foods linked to particular targeted microbes, and pause and ask, are these complementary foods available? How do they link to agricultural initiatives? Are they affordable? And processing considerations. It's the way to incorporate them into foods that have adequate organoleptic properties going to be possible. If those things are considered, then we can develop complementary food prototypes which we can test to see if after food processing they maintain their bioactivity against the defined community. Then we go on to an intact uncultured microbiota from the target population, in this case post-SAM MAM, see if they work, and then make a pivotal decision. Are you going to go from mice to humans, or are you going to get a second species and test your leads in a second species? But then you proceed directly after that decision is made and executed to a clinical study using knowledge gained from the preclinical model that incorporated the microbes of the target population from the first go round. So I'm going to quickly go through this because I don't know, Lita, how good I am for time. How many? 7 minutes. Excellent news. Not. This is the approach we use for screening. The rules are on the right. Each group of mice is colonized with this combination of age and growth discriminatory strains plus SAM strains. Each group of animals receives a different sequence of diets. Each diet initially is composed of 6 different ingredients. The ingredients, we're talking about, are complementary foods, and they're shown in a bubble plot for each of the diets. One ingredient is dominant per diet, and of course we do collinearity analysis to make sure that ingredient levels are themselves not correlated with one another, because we're trying to link the concentration of a particular complementary food to the response of particular taxa. Take a breath. It's different orders of presentation that are very important because we want to avoid hysteresis effect. So we do this, and then a follow-up screen, and then a follow-up screen. So 80 plus diets later, we have lead microbiota-directed complementary foods that we can test. We use monotonous feeding of animals that have these SAM strains and the targeted age and growth discriminatory strains with the prototype versus existing therapy, Kachuri-Hawa in this particular case, and use germ-free animals as a way of measuring the availability of nutrients from these diets in the absence of microbes in different regions of the gut. You use mass spec, look at levels of amino acids, micronutrients like B vitamins, you look at monosaccharides, disaccharides, and you compare and contrast the levels of these nutrients in the absence of microbes and in these particular dietary contexts and in the presence of these microbes. You can take that data, by the way, and correlate the results with RNA-seq analysis to look at which genes and which metabolic pathways for nutrient utilization are being used by these different actors on stage. We can look at transporters. It's really quite informative for looking at niches. And you can also correlate it with the auxotrophies and predictive prototrophies of these organisms. Bottom right shows the superiority in terms of abundance of these targeted organisms in the presence of this MDCF versus Kachuri-Hawa. Just advance showing that there are diet and microbiota dependent effects on serum IGF levels, This is the combination of the MDCF and the presence of all 14 actors here. We compare it to germ-free, so MDCF versus Kachuri-Hawa. IGF is elevated, phospho-AKT in liver is elevated. You have effects on bone biology. This is microcomputed tomography of femurs. We go on and see similar results with intact uncultured microbiota, and then make the pivotal decision about whether to invest in the development of a second species model. So 2.5 years later, development of a robust protocol for rederivation of piglets by cesarean section as germ-free and colonization of these piglets. Why piglets? Physiologically and metabolically more similar to humans. Extraordinarily rapid growth phenotype, 250 grams a day. And then the opportunity of looking at features that are more difficult to delineate in mouse models, including features of biogeography, but also opportunities to develop new technologies, including ways of sampling communities along the length of the gut or implantable devices for monitoring metabolism. So Hao-Wei does this experiment with 2 of our MDCF lead formulations, this defined community of age and growth discriminatory strains. And you can see a formulation, MDCF, microbiota directed complementary food, V2, version 2, with 4 of these ingredients, and only 2 of these ingredients. This is the day after the days of the experiment. The animals are re-derived as germ-free, bottle-fed with a formulation that looks like sow's milk. It's irradiated. It's called Sowina. Then they're weaned onto these foods, and then they're, once weaned, followed up to 30 days. I can go through the interesting challenges of anesthesia. The 350 kilogram sow is brought in. She'll have up to 15 to 18 piglets. You have to anesthetize the sow so that the piglets, when they're removed from the uterus, don't take a single breath until they get into the notobotic isolator and put through a dip tank, because if they take a single breath, they're going to be contaminated by environmental microbes. But they can't be so deeply anesthetized you can't revitalize them. So the bottom line is 2 minutes. Thank you. Is there any debate about that? No. So I'm almost done, folks. You have improved growth, isocaloric diets. You have increased representation of the targeted microbes. You have marked effects on host metabolism, including increased hepatic and diminished muscle beta oxidation. So now we're at the stage of doing a human study that was begun in Bangladesh, and that study is summarized here. It's a bake-off between our lead MDCFs. Again, it's following a population prior to the intervention, blood and fecal samples at the beginning of treatment, and then blood and fecal samples at the end of treatment, with fecal samples during that. The endpoint biomarkers are MAZ scores, serum proteomics, enteropathogen burden, a variety of other studies of immune function, including IgA targeting and targeted mass spectrometry of serum. But we have a unique opportunity of determining whether we can repair microbiota immaturity, whether we have any control over the rate of repair and the long and short-term impact of such repair. We also have an opportunity of these sorts of early pre-POC clinical studies to not only select MDCF, but also determine is response of these parameters linked to certain configurations of the microbiota. So we're going to be able to are we going to be able to determine whether specific clinical outcomes are correlated with the representation of specific bacterial strains, and what we'll learn from non-responders, and what additional approaches might be useful, including direct administration of probiotic consortium, though the regulatory route is much more complicated, which is why we looked to microbiota-directed complementary foods. Challenges: define the underpinnings, metabolic underpinnings of microbial succession. The implications, I think, are great. The challenge is great, but there are models for being able to do this. But I think the lessons learned could be great in terms of understanding the determinants of cooperation and competition between microbes, and studies that are intended to develop next-generation pre, pro, and symbiotics. New approaches for understanding how to mitigate or modulate enteropathogen invasion, and also potentially to look at restoration of microbiota after perturbations, including antibiotics. And the last thing is this issue, and I promise to take no more than 30 seconds, but the words are important. We're at the threshold of a revolution in our understanding of the molecular components of food with mass spectrometry. That was demonstrated earlier on looking at the glycan landscape of human milk oligosaccharides, but it's going to generalize. And that is a historic moment for us as consumers of the food, but also as us as observers of microbes. So I think that this marriage that I alluded to between preclinical models, food science, and understanding how these ingredients are transformed or not by food processing, and how to link structural features of a food with the response of microbes and hosts, and view the microbial community as transforming food into a metabolic output that has profound effects on many cell lineages and many features of our biology. I thank you for your patience, and I appreciate being here.
Okay, we have just a short time for questions, so please.
Hi, Brandon Eicher from Nutrilite. Excellent talk, Dr. Gordon. I wonder, when you watch primates wean their infants, typically there's a mastication process in the foods. Do you think that that might be applicable here for recovering some of these microbiomes in these children if the mothers were to pre-chew the food and then offer it to the infants?
Interesting thought. I have no specific data one way or the other. I think your point is an important and general one, which is number one, partner with anthropologists to see how different societies apply child rearing practices. Two, consider all forms of processing of foods in terms of how they affect the molecular composition of food and how that might impact the microbiota. I'll also say, as a parenthetical comment, that the investment in science of the type that I just described in itself is incomplete if you don't invest the time to consider ethical, social, cultural, and regulatory issues. And I think that if you don't spend the time early on in that, then moving from discovery to development to deployment will be severely impeded. So a lot of the work that we're doing is on the ESC issues and regulatory issues. And the reason we decided with food-based intervention rather than probiotic interventions initially. Okay. Other questions?
Hi, Dr. Gordon, that was great. My ancestors were actually from Mirpur. My name is Julie Mirpuri from UT Southwestern. I have two questions, one technical and one broader. The first one is, when do you think is the best time for neocolonization in mice? When you want to study infants and the pediatric population. So I notice a lot of people do neocolonization experiments at 4 weeks, but there's already a lot of development. In my lab, we're doing it more 3 to 5 days of life. So that's more the technical question. And the other broader question, which is something I think you highlighted, is how can we be better at getting samples and understanding the microbiome internationally from all over the world? Because I think we're missing an opportunity, or many opportunities, to develop good hypothesis-driven research because of differences in diet, environment, and the way we wean our children.
Well, as to the latter point, I couldn't agree with you more. And I think that's one of the philosophies of the Gates Foundation, which is to take lenses of anthropology and apply it to the world and see populations that are representative of different lifestyles, make an investment where the trust that I described is great between the population and healthcare providers, and to be able to embark on the journey that I tried to illustrate. Second of all, Kerry Coarden, for instance, studies of bone biology involve, in addition to the types of experiments we described, also colonization of dams so that they can transmit their microbiota to their pups, beginning at parturition. And I think exemplary microbial communities, either intact uncultured microbiota or defined culture collections, which are more sustainable, need to be deployed to look at the effects of exposure on different facets of host biology. So I applaud and try to reinforce the statement you made about the importance of early exposures.
Okay, we have time for one last question.
Thank you, Dr. Gordon. As someone who's practiced in India and actually seen these people, these children, right front and center, my perception is that a lot of diarrheal illnesses actually precipitate all of these malnutritions in these children. I was wondering, with light of the new study from Nature, which said that the probiotics actually reduced the effect of the diarrhea, how did you adjust for the interaction for these multiple diarrheal illnesses, and whether these interventions that you had in mind, would it also have an outcome reducing the diarrheal illnesses which in these children?
So you raise a very important set of points. In our birth cohorts, we have a collaboration with Margaret Kozak, for instance, at Hopkins, a study of Peruvian birth cohort, which is included in this network. So asking questions like diarrheal days, the nature of enteropathogen burden, antibiotic exposure, complementary feeding practices, including issues of food diversity and when complementary feeding is initiated. What are those effects on overall diversity? What are the effects on MAZ scores? Again, there are many of these parameters that have to be considered. One of the things about these transplant experiments is you could do those sorts of surveys and then take, if you have well-archived fecal samples, exemplary fecal samples representing different states of the child's health, and be able to model the effects of those states on host biology and also interventions. You can enroll a child in all arms of a clinical trial, so to speak, by taking their pretreatment microbiota and transplanting into different groups of mice and reenacting the clinical study or trying new different types of therapy. That's an important part of our discovery pipeline. But I'm finished with time, and my program officer at NIDDK is rising, and I've learned to pay attention to him over the years. He's been very supportive, but very informative. So, thank you.
Good morning. Thank you for coming to this important presentation. So we are now at session 5, which is host microbiota interaction. So we have agreed by now that we want to know what's there, but what we really want to know is how our body interacts with the microbiota, and if there is a possibility for us to modulate it in a way that would be beneficial for human body. And we have 4 exceptionally good speakers today. Dr. Andrew Gewertz from Georgia State University, and he will talk about the gut microbiota, diet, chronic inflammation, and metabolic syndrome. Dr. Dana Philpott from University of Toronto, and she will present NLRP6, current challenges on studying the impact of the microbiome on host responses. Dr. Nita Salzman from Medical College of Wisconsin, and she will present antimicrobial peptides shape intestinal bacterial niche creation and competition. And Dr. Ramnik Xavier from Harvard University will talk about microbes, molecules, and mucosal immunity. So just I invite all of the speakers and give them a round of applause.
Is it?
Okay, well, thank you for the introduction and opportunity to be here today. In the next 15 minutes, I'm going to try to convey 4 take-home messages we've learned in studying microbiota, diet, and inflammation over the last few years. So this is an artist's illustration of the gut microbiota. And beautiful as it may look, it will not police itself. Rather, it requires active, innate and adaptive immune system to keep it in check. And the first thing we learned in this area is that failure to keep a microbiota in check can result in multiple flavors of gut inflammation, including colitis, perhaps undernutrition, but also metabolic syndrome. And I think this is a pretty generalizable message that failure to keep a microbiota in check can result in inflammation. But we learned it from study of mice that lacked the flagellin receptor, toll-like receptor 5. Very briefly, toll-like receptor 5 allows detection of any motile bacteria that breach the mucosa, be they pathogens or non-pathogenic organisms. And in the absence of toll-like receptor 5, these bacteria are not cleared very quickly, and this makes mice prone to developing spontaneous colitis. It is not all of the mice, and this is certainly one of these phenotypes that is environmentally dependent and that we don't see it in every facility, and it can be removed by rederivation of the mice into another microbiota. But we see it in the knockouts and not in the littermates that are in the same microbiota. The more common phenotype of these mice is that they develop metabolic syndrome. And this has been seen across multiple facilities, but not all. In any case, these mice develop a low-grade inflammation that correlates with increased weight gain, increased adiposity, dysglycemia, and other parameters of metabolic syndrome. The extent of gut inflammation these mice exhibit is proportional to their phenotype. In other words, using fecal lipocalin-2, which we've shown is a reliable, non-invasive dynamic marker of inflammation, we see a much lesser degree of inflammation in the knockouts that have metabolic syndrome, whereas on this log scale, the levels of lipocalin-2 are much higher in the mice that have colitis. And this is proportional to the extent of their microbiota alteration. In other words, the non-colitic knockouts that have metabolic syndrome exhibit a relatively moderate but nonetheless significant alteration in their microbiomes, whereas the colitic TLR5 knockouts exhibited a much more severe alteration in their microbiota composition. In germ-free conditions, these phenotypes go away entirely. In other words, the inflammatory marker becomes undetectable in both wild-type and TLR5 knockout mice under germ-free conditions, and all their phenotype disappears, any indicators of inflammation, and the metabolic phenotype as well. And the microbiota composition is sufficient to transfer the phenotype to germ-free mice. So in other words, wild-type germ-free mice, if they got the knockout microbiota, develop the metabolic syndrome. So what are the features of this microbiota that makes it pro-inflammatory and can transfer these phenotypes? We see a lot of differences in species that I won't go through too, but one general characteristic of this altered microbiota is that it seems to be more aggressive and that it can encroach upon the host. Here using non-dehydrating stains of the mucus layer and measuring how close are the closest bacteria, we see that in mice that lack toll-like receptor 5 or that receive the transplant, the mice have bacterial encroachment into the mucus. As a general phenotype, this is true in humans. So this is a slide generated from patients at the local VA in Atlanta. Has nothing to do with TLR5 per se, but is simply saying that in humans with metabolic syndrome, they're featuring microbiota encroachment. In other words, as they have increasing levels of dysglycemia and hemoglobin A1C, there's a closer distance between bacteria and the epithelium. But certainly for a disease like metabolic syndrome that has had a huge increase amidst constant host genetics, we've really started to think more about, well, what might have changed the microbiota and or how it interacts with the host to promote inflammation. And we focused mainly on dietary factors. We thought about changes in macronutrient content and micronutrient content. So the first thing we did, like many people, And that actually brings me to lesson number 2, which is a healthy gut requires proper nourishment of the microbiota. So when we began exploring diet, like many researchers, we compared mice given a quote-unquote high-fat diet to the standard thing used in most animal facilities, which is chow. So as was discussed yesterday, a high-fat diet is an open-source diet. It's assembled from very well-defined, partially purified ingredients, such as shown here, whereas chow is a conglomerate of food scraps. You can see the list. Ground corn, beet pulp, fish meal, all sorts of things. And this makes it not very amenable for well-controlled types of studies of how various food ingredients might affect the microbiota. But it does have one thing going for it, and that is this conglomerate of food scraps, undefined as it may be, keeps the mouse intestine healthy. Specifically, these are pictures of just showing what a mouse intestine looks like on a normal chow diet. Many of you would be familiar with this. On any of the open source defined diets, the intestine is quite unhealthy. It's fragile, there's a major loss of mass, and this is irrespective of fat content. This is a feature of all of the defined diets that we've looked at so far. And this correlates with, this is just showing the same thing by histology, that on any of the purified diets, you see a significant loss of not only colon weight, but the crypt villus length gets a lot shorter. And this, working with research diets, we found we can recover if we supplement the purified diet with the fermentable fiber, inulin, but not the insoluble fiber, cellulose. This correlates with differences in proliferation. Simply on the purified diets, we would call it gut atrophy. The intestine just doesn't proliferate very well, again, correctable by inulin. And this correlates with microbiota encroachment, which we see on the high-fat diet, and again, correctable by supplementation of that diet with inulin. All of this is microbiota dependent. In other words, whether one looks at colon weight or villi length, the atrophy that we see from the purified diets, we don't see this in germ-free mice, nor can we increase the villus length in germ-free mice by supplementing these diets with inulin. So what changes in the microbiota is the purified diet having, and how can inulin correct this? Well, one of the biggest differences is one of the most basic, just measuring total bacterial loads in the feces by PCR. We see that the purified diet results in an approximately 1 log drop in the total number of bacteria, and we can fully recover this by supplementing the diet with inulin. But we don't recover the same microbiota composition. Specifically, the high-fat diet or either purified diet causes this dramatic change in composition that you can see here. Not much of an effect with cellulose, but you supplement the diet with inulin, and not only do you increase the total numbers of bacteria, but you change the composition quite dramatically. So it's really not any closer to what we would see in a chow-fed diet. It's just been altered further. Well, does it matter that we've altered the composition further? Well, we seem to recover the intestinal phenotype. It looks relatively normal. And supplementing the diet in this manner protects against high-fat diet-induced metabolic syndrome. Here I'm just showing the impressive effect of inulin in reducing adiposity from the high-fat diet, but we see similar effects on other parameters of metabolic syndrome. So maybe these differences don't really matter all that much. But I remembered something that Jeff had said at a microbiome meeting last year, which was lesson number 3. And that is to be really, really careful about telling people what to eat. And these mice on these inulin supplemented diet, that their basal metabolism seems okay, but if we administer the compound DSS, we see an extreme colitis like we've never seen before, shown in these histological images. That even at low amounts of DSS, it's just a very severe colitis, and the colons effectively disintegrate within a couple days, and very severe bleeding and death. Well, and even if we don't challenge the mice with DSS, if the mice are simply maintained on these inulin supplemented diets for a longer period of time, such as 6 months, they start to develop liver cancer. And this is a combination of dysbiosis and attempting to repair it with these inulin-enriched diets. So if it's not safe to tell people what to eat, can we at least tell them what not to eat, or some things that they should avoid? And that brings me to lesson 4, which is that some common food additives can perturb the microbiota and promote inflammation. Specifically, the class that we have studied are emulsifiers. These are detergent-like molecules that stabilize mixtures of immiscible liquids. They're a very common food additive. They're incorporated into all sorts of processed foods. We focused on 2 synthetic emulsifiers, compounds that don't exist in nature, polysorbate 80 and carboxymethylcellulose, that have been increasingly used since the mid-20th century in a rough correlation with increasing incidence of some chronic inflammatory diseases such as IBD and metabolic syndrome. And we found that both of these emulsifiers have quite a strong impact on the microbiota, altering its composition and promoting microbiota encroachment. So again, this is non-dehydrating fixative to look at where are microbiota located in the colon, and hopefully you can appreciate that in mice fed either carboxymethylcellulose or polysorbate 80, bacteria are encroaching upon the host and getting much closer to the epithelium. So what is the consequence of this microbiota encroachment? Well, if mice are prone to developing colitis, such as IL-10 knockout mice, they're more likely to develop it if they've consumed emulsifiers. These are some of the biomarkers we use to assess colitis, and this is just the brief summary. In our facility, IL-10 knockout mice are prone to developing colitis, so about 40% of them would develop it anyway. If they're maintained on either emulsifier, that incidence of colitis approximately doubles. In wild-type mice, the encroachment seems to be promoting metabolic syndrome, in that mice fed either emulsifier exhibit an increase in body weight, mostly in the form of increased adipose tissue, and this correlates with increased food consumption and dysglycemia. If the emulsifiers are given in germ-free conditions, there's no effect on phenotype, no effect on metabolic phenotype or any of the other parameters that we can measure. And in subsequent experiments using ex vivo systems, our results are suggesting that the emulsifiers are acting at least in part on the microbiota directly rather than directly on the host. So summarizing the emulsifier work, which was published last year, is that this class of food additives seems to be promoting bacteria encroachment and misbehavior to a place they shouldn't be. They may like it there, but I don't think it's very good for the host. And we have in collaboration with Gary Wu, we just got a small clinical trial funded. So we'll see how applicable this is in humans, hopefully in the not too distant future. So to finish with a gap, I think the general gap I'd like to highlight is a need for better understanding of how a range of major and minor food components impact microbiota composition and gene expression. And to me, one critical specific gap that's going to make this a lot harder in mice is at present the lack of a compositionally defined, i.e. open source diet that can maintain a healthy intestine reminiscent of a chow-fed mice. I think it's nice to standardize with diet, with facilities of microbiota, but I worry that in the risk of standardizing, we get much further from physiology. So I think a very clean mouse facility is moving away from relevant physiology, and an open source diet, at least the ones currently available, are very much moving away from relevant physiology as well. So lastly, I just want to thank the people who did the work. Everything I showed today was generated by these three very talented individuals, Tom Vijay Kumar, who's now at Penn State, Benoit Chassing, who just started his own lab at GSU, and Jun Zhao in my lab. And key collaborators are listed, especially Mike Pelzon at Research Diets, and Ruth Lay, who was at Cornell and is now at the Max Planck. Thank you.
Thank you so much. So we'll have a panel discussion at the end of the session. So our next speaker is Dr. Dana Philpott from University of Toronto, and she will talk about NLRP6, current challenges on studying the impact of the microbiome on host responses.
Thank you. Thanks for the introduction, and also thanks for the invitation to be here. I think I'm the only Canadian here, which you'll probably notice when I say things like out and about, so I apologize in advance. So I'm going to talk about, as Gabrielle mentioned, NLRP6 and the impact on the microbiome and intestinal inflammation. So in my lab, we study the NOD-like receptors, and these are a set of receptors that are present within the cytosol of the cell that detect both bacteria as well as danger signals and trigger inflammatory responses. The characteristic features of these NOD-like receptors are this C-terminal series of leucine-rich repeats, which have been shown to be involved in ligand sensing in some of these molecules, especially for NOD1 and NOD2, which detect peptidoglycan. And then on the N-terminus of these proteins are different protein interaction domains that allow them to connect to different pathways to stimulate these inflammatory signals. So this also defines them into different subfamilies. So the NOD subfamily have this CAR domain, caspase activating and recruitment domain, which allows them to link up to the NF-kappa B signaling pathway. These other molecules, NLRC3, C5, and X1, which we study also in my lab, have a similar domain to a CAR domain, but defined as an X domain, since we don't really know the binding partners of these molecules. The NLRP subfamily, on the other hand, have this purine domain, and this allows them to link up and form these inflammasome complexes, which are important for the production and secretion of inflammatory cytokines IL-1 beta and IL-18. So NLRP6 is a classic, has this classic NLRP structure. Here. It has, again, this purine domain on the N-terminus and the leucine-rich repeats on the C-terminus. But I think for now, we really lack a lot of biochemical information about the NLRP6 potential involvement in the inflammasome. There has been one study to show that it interacts with ASC, but whether it really forms a canonical inflammasome has not really been formally demonstrated. And also, I guess importantly also in terms of its ligand, we don't know what triggers the activation of NLRP6. We were interested in NLRP6 because my lab studies NOD-like receptors, especially in the context of intestinal disease, inflammatory bowel disease. And NLRP6 was interesting to us because it's highly expressed in the gastrointestinal tract in humans and mice, especially in the small intestine. And this just shows an RNA-seq that we did in most intestinal organoids, so these are pure epithelial cell populations, looking at the relative expression of different NLRs within these epithelial cells. And you can note that NLRP6 has one of the highest expressions compared to the other NOD-like receptors, including the ones that we like to study a lot, which is NOD1 and NOD2. And interesting also expression that's absent or beyond the level of detection, things like NLRC5 and NLRP3 in these intestinal epithelial cells. So again, yeah, it's the most highly expressed NLR. So there's been a lot of interest in NLRP6 over the last few years. And this just summarizes its potential role in an inflammasome-dependent way and its involvement in intestinal homeostasis as well as colitis. So it's presumed that NLRP6, again, has this inflammasome-dependent pathway that leads to the production of the cytokine IL-18. And IL-18 we know is important in an intestinal homeostasis, so it helps to maintain epithelial barrier integrity. But on the flip side, if you have low production of IL-18, then you have more susceptibility to colitis as well as colorectal cancer. So there seems to be this link between NLRP6, IL-18, and its role in intestinal homeostasis. And just to summarize again some past studies looking at NLRP6 in the regulation of colitis and also of the gut microbiome, it's been shown that colitis and tumorigenesis susceptibility of NLRP6 mice, as well as the downstream potential effectors in this pathway, ASC and IL-18. First of all, it's a transmissible phenotype to wild-type mice. So they show that if you take the microbiota from these animals, put it into naive mice, then the wild-type mice now also show this increased susceptibility to colitis as well as colorectal cancer. And in line with that, then, studies also have shown that there's an altered fecal microbiota in these NLRP6 deficient mice, characterized by the expansion of these representative bacterophyla, including Bacteroidetes, in particular Prevotella, as well as TM7. But the potential problem that we've been going through, especially we've done this kind of study with NOD2 deficient mice, is that there's a problem that these studies were done with non-littermate mice. So we question whether this pro-colitogenic microbiota phenotype that's associated with the NLRP6 deficient mice, perhaps it was acquired stochastically in that certain facility and was potentially genotype independent. So then we wanted to revisit the role of NLRP6 in the regulation of the microbiota at first, and then I'll show you in a moment also in colitis phenotypes. So what we did is we took heterozygous NLRP6 mice, bred these mice to derive littermates of wild-type HETs and knockout NLRP6 deficient mice. And these mice were separated at weaning. And I'll show you the data from mice that were put into single cages, but we also saw the similar data if we put them in co-caged with, depending on sex, of course. And these mice were then, again, separated at weaning and left for 5 to 6 weeks. And then we isolated different samples from the intestines of these mice. So we looked at distal ileum mucosal scrapings, proximal colon scrapings, as well as the fecal pellet. We extracted DNA and then performed Illumina sequencing to analyze the microbiota of these different animals. So I'm certainly not an expert in microbiome analysis, so we have lots of help on this part, but we wanted to do the most robust tests to look at the different compositions of these animals. So this is just showing principal components analyses that were generated using the Bray-Curtis dissimilarity matrix, but we also showed this with other distance measures, including UniFrac, both weighted and unweighted. And I guess it's pretty clear from these studies, if we look at the feces, the colon scrapings or ileum scrapings, we saw no difference in the communities between the wild-type mice shown here in the blue circles and the NLRP6 mice shown in the green squares. So again, in the 3 different compartments. So this showed us that there is no significant effect of NLRP6 genotype on bacterial community composition, and I won't show you, but we also showed looking at different taxa within these mice, there was no difference in bacterial taxa abundance between these two groups. And I should mention also that there was a paper that just came out in Immunity that showed a similar finding to us. They used germ-free mice and re-derived the P6 mice into germ-free conditions, gave these mice similar microbiota, and also showed that there was no difference, there was no impact of NLRP6 on community structure when they looked at X germ-free mice in these conditions. So what we did find, perhaps not that surprisingly, is there was a genotype independent but sex dependent effect on the microbiota. And again, this is something that already has been shown, but just to show you in our animals. So this was shown in the 3 gut compartments that were analyzed, again looking at a Brake-Curtis distance matrix. When we studied the male mice in squares versus the female mice in circles, we saw a nice separation between the female animals versus the male animals. But again, this was completely independent of genotype. Using Lefse analyses, you can see the differential taxa that were associated with males versus females. So males had a greater abundance of these organisms, including Prevotellinaceae, and the females had an abundance of a lot of Lactobacilliaceae organisms. So again, as I said, sex also has an influence on bacterial community structure. So looking now at the role of NLRP6 on colitis severity, so the similar setup, we took heterozygous breeding pairs and derived littermates from these animals. I should have mentioned we didn't study the hets just to have less mice to study. We just looked at the wild-type mice versus the knockout mice from these littermates. These were either co-housed or we also did signal housing. It didn't make a difference. These mice were left 4 to 6 weeks and then challenged with DSS, was what Andrew already talked about. This is a typical model to study colitis in animals. So these mice were given DSS for 5 days, then water for the remainder of the study. And we monitored clinical signs daily, so this included weight loss, for example, and physical signs, diarrhea, that kind of thing. We measured the colon at 7 days, as well as collected fecal samples for lipocalin, which is a marker of inflammation, as Andrew talked about, as well as did a number of measurements of cytokines, etc., from explants of these mice. What we found, which was interesting, is that there was a genotype and sex-dependent impact on colitis. So first of all, just looking at the male mice, you can see wild-type versus NLRP6 on DSS. We didn't see any difference between genotypes, although there was definitely an effect of DSS. They were losing weight during this study. But what was interesting to us is that for the female mice, we saw a genotype-dependent impact on colitis, where the NLRP6 deficient animals had a greater severity of colitis in this model compared to the wild-type female animals. What's also notable is that the female mice have less severe colitis than male mice, and again, this is something that has been previously shown. This just shows the disease activity scores, including stool consistency, rectal bleeding, and weight loss. And again, we saw that the NLRP6, only the female mice, had increased severity compared to their wild-type counterparts. So these studies, first of all, show that female mice, as I mentioned, has already been discussed, are partially protected against chemically induced colitis. And the role of NLRP6 in acute colitis within this model is sex-dependent, only revealed in female animals. So I'm going to talk conclusions. So I think many of these statements were already brought up yesterday, especially in Thad Stependek's talk. But just to go over our particular findings, NLRP6, first of all, we showed it does not have a significant impact on intestinal microbiota at homeostasis. And we say, of course, that the use of littermate mice is really the gold standard for normalizing not only genetics, but also environmental variability in animal models. The bacterial microbiome is impacted by sex. Sex hormones are known to influence the microbiome, and this was previously published by my colleague Jane Danska at the Hospital for Sick Children in Toronto. And finally, NLRP6 deficient female mice, but not males, have this increased severity to DSS-induced colitis compared to wild-type females. So there seems to be an interplay of NLRP6 genotype and sex, but independent of the microbiome, that increases colitis severity. So getting to the critical challenges in understanding the role of the microbiome in mouse models, I think this study really demonstrates that the use of littermate mice from heterozygous breedings is imperative for well-controlled studies. I think the things that we need to think about is that the microbiome influences on mouse phenotypes are not just restricted to the gut. There's a lot of studies showing that the gut microbiome can influence other disease models in animals independent of gut. So how can we better control these studies? There's a lot of studies out there that co-house mice. Is this sufficient to normalize the microbiome? Our data says it's not. You really need to use littermate mice from mothers or related mothers to have a well-controlled study. And the other challenge we have is how to control microbiome effects in complex backgrounds. So many people, like us, for example, we have double knockout mice. Some people have triple knockout mice. How do you do littermate controls for this kind of complex genotypes in mice? Lab-to-lab irreproducibility is a huge issue. So multi-center validation, should we, contact colleagues and see if they can validate our findings in other facilities? Will we need to move to a standardization of the microbiome using defined communities? And also, as we recently shown, there's definitely other microbes that impact phenotypes. We had a paper recently showing that the parasite Trichomonas murus has a huge impact on the gut at baseline. So parasites, viruses, other organisms also should be considered. Improving this, we need to report source. So this is also taken from the recent review from Thad and Skip Virgin. We need to really report source and sex, so genetic background, control for the microbiome, discuss the breeding strategy that we use to generate littermate mice, the animal numbers, as well as we showed in our study. It's important to also include sex within this, and also the cooperation among stakeholders, funding institutions, editors, reviews need to really enforce these reporting rules. So I'll end there. Sorry, I went 1 minute over time. And thank the people in my lab that did this work, so particularly Susan Robertson, who's my microbiome expert, and Paul Lemire in my partner's lab, Stéphane Chechardin, with help from David Gutman and Heather Mohn that did all the microbiota bioinformatics analyses. So thank you very much.
So, our next speaker is Dr. Nita Salzman from Medical College of Wisconsin, and she will present antimicrobial peptides, shape, intestinal bacterial niche, and creation and competition.
Thank you for the invitation to speak to you today. I think this might be a little bit more from the side of the microbe than the host. The study of my lab has been antimicrobial peptides and factors that shape the intestinal microbial niche. We know and have been reported in the last day, and we'll continue to discuss, that there are a number of factors that shape the intestinal microbial niche, and the one I'm going to be speaking about today is antimicrobial peptides, which are immune effectors. Antimicrobial peptides are an ancient and conserved mechanism of host defense. We find them from plants to bacteria to fruit flies, moths, and humans. And we know that they have a role in fighting off pathogens, but we also are looking into the role of regulating the commensal microbiota. There are a number of sources of intestinal antimicrobials in the GI tract, which is the area of my interest. We know that panacels, and this is a study from my lab and others, produce a wide variety of antimicrobial peptides, including defensins, which are cationic antimicrobials, lysozyme, and antimicrobial lectins. We know enterocytes also produce antimicrobials. The liver produces bile acids, which are antimicrobial. And then microbes actually are a very rich source of antibiotics in the gut through the production of metabolic byproducts, bacteriocins, enterocins, and other toxins. Work from my lab a number of years ago looked at the role of enteric defensins in the regulation of the intestinal microbiota. And this is looking at defensins in panacels. This is an EM from Andy Wallet's lab of a panacel, which is found at the base of the small intestinal crypt. Panacels line mucosal surfaces in the small intestine at the base of the crypt. They produce and secrete a wide array of immune effectors and antimicrobials, including defensins. And work from our group showed that if you change the composition of antimicrobial peptides within the panacel, and this is using littermate controls with either reciprocal models of adding a human defensin or removing production of any panacel defensins, we see reciprocal shifts in the microbiome. And then we also found that if you alter the composition of panacel antimicrobial peptides, you alter the composition of the intestinal microbiota. And we and many, many other groups, of course, have shown that if you alter the microbiota, you alter mucosal immune responses, and obviously you alter a lot of the animal physiology. But then we turned to another question, which was, what is the role of antimicrobials produced by commensal bacteria in either forming or competing for an intestinal niche? And for this, we turned to use Enterococcus faecalis, which is a gram-positive coccus. It's a facultative anaerobe, and it is a commensal of both animals and the human GI tract, and it's actually quite broadly found as a commensal. It's intrinsically resistant to cephalosporin antibiotics, and it's also extremely good at picking up antibiotic resistance genes via horizontal gene transfer. This has resulted in the development of multi-drug resistant strains, which are common causes of hospital-acquired infections, both Ephesium and Eficalis, and they've been designated serious public health threats by the CDC. So our interest was to investigate how antimicrobial peptides contribute to Enterococcus faecalis niche formation and competition in the GI tract. To do this, we needed to generate an animal model that did not disrupt the GI tract with antibiotics, because if you disrupt the GI tract with antibiotics, you're no longer studying colonization, you're studying infection. We did this by feeding mice with Enterococcus in their drinking water for 2 weeks. And after withdrawing Enterococcus from the drinking water, we followed enterococcal colonization in the feces and then at harvest of GI tract portions. And we found, this is showing over 4 weeks, but we've done this as far as 11 or 12 weeks, which is as far as we've taken an experiment. And we find continuous shedding of Enterococcus from the GI tract of these mice. And when we sacrifice the mice, we find that there's consistent, stable colonization of the mouse GI tract without prior antibiotic disruption. So now we're able to get a lab strain of Enterococcus to colonize the mice, and this allows us to look at how it forms its niche and how it competes for its niche. Now it turns out that Enterococci can harbor plasmids, pheromone-responsive conjugative plasmids that produce bacteriocins. The plasmid of our interest is the one depicted here, which is PPD1, and it harbors bacteriocin 21. And what we find is that bacteriocin, which are antimicrobial peptides, they can specifically target and kill bacteria of the same genus, and they act by damaging cell membranes. And this is an example of AS48, which is essentially the same as bacteriocin 21. It disrupts cell membranes, and it is broadly anti-gram-positive in vitro. Even though these are considered to be somewhat specific, this is actually a relatively promiscuous bacteriocin. So the bacterial strain that harbors a bacteriocin plasmid also encodes an immunity peptide, and that it uses to protect itself from being killed by its own bacteriocin. enterococci produce bacteriocin to give them a competitive advantage. And alternatively, under the correct circumstances, instead of killing another organism with its bacteriocin, it actually encodes also what's called a conjugation apparatus, so that it actually directly can contact another Enterococcus and transmit that bacteriocin to an Enterococcus that lacks it. And this requires direct cell-cell contact, and we actually use that fact to our advantage to understand the niche in the GI tract. So what we found in this experiment, where we were just looking at culture of Enterococci in the GI tract from mice that did not receive Enterococcus, mice that got our Enterococcus strain that lacked the bacteriocin, and mice that harbored the bacteriocin, the mice came with a full complement of Enterococci when they arrived from the vendor. If we colonized them with lab Enterococci without bacteriocin, it colonized at a low level, but persistently. If we colonized with Enterococci that had the bacteriocin, it colonizes at a much higher level and essentially overtakes the entire niche of Enterococci in the GI tract. If we knock out the bacteriocin, part of the bacteriocin operon, here we have Enterococci colonizing, here we have the bacteriocin Enterococci colonizing, and here we have the knockout. It no longer can colonize. It turns out if we restore the bacteriocin operon on a separate plasmid, it regains the ability to colonize, but it does not regain the ability to conjugate. And we use that also later. So the other thing we noted is we were looking at what happens whether conjugation or competition is what's going on in the GI tract. So we first colonized mice with Enterococci that lacks bacteriocin, and then chased in what we would call our, our regular strain that produces bacteriocin. And what we found, this is looking at transconjugates. This is looking at the first organisms and which ones have picked up the bacteriocin plasmid. And over the period of 5 weeks, 100% of these strains that are remaining have actually picked up bacteriocin. So this suggests that when delivered sequentially, the Enterococcus harboring a bacteriocin can overtake the entire niche in the GI tract. And this also tells us something very important, which is that Enterococci localize to a specific physical niche in the GI tract and directly physically interact with other Enterococci in the GI tract. We thought we could employ this therapeutically, and so we did. We looked at VRE, and up here we colonized mice fine with multi-drug resistant Enterococci. In this case, we used an Enterococcus, our complemented strain, because we did not want to conjugate bacteriocin activity to a multi-drug resistant strain. We thought that was a bad idea to give it another mechanism to colonize. So we chased with a strain that could produce bacteriocin but couldn't conjugate it. And what we see is over several weeks after treating the mice, we can completely eliminate, almost completely eliminate colonization by VRE with the administration of this bacteriocin-producing strain. We find that because we use the complemented strain, we could not conjugate the plasmid to the VRE strain. What was also interesting is we looked at the microbiome, and we saw that when we compared the microbiota of Enterococcus colonized mice versus PPD1 colonized mice, there were some significant but subtle differences in the microbiome. They were entirely driven by changes in Mucospirillum schadleri. It turns out that Mucospirillum is not a gram-positive and is certainly not an Enterococcus. So we believe that these changes are indirect changes and not directly caused by direct killing from the bacteriocin. And we just compare this to conventional antibiotics. This is mice that are treated with streptomycin, and we see much more significant differences in colonization between mice treated with streptomycin, say, compared to mice given bacteriocin-harbored Enterococcus. So unlike conventional antibiotics, bacteriocin 21 primarily impacts enterococcal populations, and this suggests that we can actually therapeutically target a specific niche in the GI tract by engineering organisms. This takes advantage of two aspects. One is it takes advantage of the native biology of the Enterococcus to gravitate to the right place in the GI tract, and then it takes advantage of the specificity of the bacteriocin to specifically eliminate the multi-drug resistant strain. It turns out this strain works on Ephesium as well. But then we found a little hitch in what we were doing, and this leads to my discussion of the knowledge gaps that need to be addressed before we can advance these types of treatments. We looked at gene expression from these mice, from the guts of these mice that were treated either with nothing or with Enterococcus alone or with Enterococcus that harbored bacteriocin. We isolated RNA from each of these segments of the GI tract, and then we looked at the microarray. And we did a principal component analysis just to look at the microarray. And I'm just going to take you through the small intestine. We see that the control mice are clustered very closely together. The ones that have just been colonized by our lab strain of Enterococcus actually cluster quite separately from our controls. And those that received the Enterococcus harboring PPD1 cluster separately as well. So this suggests that we have differential gene expression driven by the colonization and also by the PPD1, the effect of PPD1. And this is actually a major knowledge gap if we want to therapeutically intervene in changing the microbiome. This is an important translational goal, intervention, but it may lead to unintended consequences. And I apologize for the stick drawing and all the arrows everywhere, but essentially we don't have the slightest idea at this point what is affecting what in the GI tract. Either these microbes we know are producing this toxin, it's killing other microbes. Are we signaling the host through the production of antimicrobial peptides? Is this organism directly signaling the host? Are other metabolites signaling the host? Are we directly changing other members of the microbiota, or is the host response changing other members of the microbiota? So we know that we have unintended consequences here, and we need the knowledge for precise intervention. So our knowledge gaps, what I consider a major knowledge gap is a better understanding of microbe-microbe and microbe-host interaction in the GI tract, which will allow more precise manipulation of populations. This will entail the development of good systems, genetic systems for genetic manipulation of commensal species, a greater understanding of the bacterial physiology of commensal species, notobiotic systems with graduated complexity to take apart these interactions, and also the identification and characterization of physical and metabolic sub-niches within the gut. And many of these things have been raised over time through many of the speakers over the last day. The collateral benefits to this, other than precise intervention, is just having a better understanding will enhance our ability to make sense of our large data sets in our current microbiome studies. And also, it should allow more appropriate sequence annotation and improve our database accuracy, which is clearly a concern for understanding our current studies. And finally, the people who did this work are here. Most of the work I talked about today was done by Sushma Komaneni, who was a very gifted postdoc in the laboratory. A large group of collaborators. Chris Kristich is my collaborator for all of our enterococcal genetics, and our funding agencies, of course. And see, somebody bought 36 seconds off of my talk.
Thank you. Great presentation, and 33 seconds less than the 15 minutes. So we have.
There you go, Ramnik. You get an extra 36 seconds.
So we have our last speaker for this session, Dr. Ramnik Xavier from Harvard University, and he will present microbes, molecules, and mucosal immunity.
So I want to thank Lita and the organizing committee for this invitation. And what I'll try to do in the next 15 minutes is to give you some highlights of how we are approaching the gut microbiome in health and disease. So broadly, the gut is primarily evolved to absorb nutrients, and despite sort of years of understanding of this process. I think there's still a deficiency in trying to understand what are the receptors that sense these nutrients. At the same time, the gut has evolved a sort of a complex array of various receptor systems that sense pathogens. And to date, we've sort of still to understand how these commensals are sensed and responded. But the first vignette that I'll talk to you about is this increasing recognition of how dietary factors and commensals contribute to a metabolic signaling pool that then educates gut immunity through a large panel of receptors that result in innate and adaptive immunity. So broadly, if one were to look at the gut, there are many firewalls that restrict access of these commensal microbes to encounter and signal through the epithelium. There are a number of innate and adaptive systems that in fact control these responses. And Marta and Shengwei, two postdoctoral fellows in the lab, were interested in how do these microbes or commensals interact with the mucin layer and then contribute to signaling and homeostasis. So they took a very simple approach. They asked the question, are there mucin-utilizing genes? So are there enzymes and transporters that we can manually curate and then try to identify using computational approaches which are the bugs that express these enzymes and transporters. So to illustrate the principle here, if you look at the MAC2 peptide, it's decorated by a carbohydrate chain that has terminal sialic acid or fucose that undergoes cleavage at these sites and then recognized by these transporter systems. So the initial analysis that Shengwei did with help from Curtis Huttenhauer to identify these enzymes and transporters, computationally search the Human Microbiome 1 database to identify microbes that selectively express these transporter genes, whether they be enzymes or transporters. And what they observed was a clear enrichment in these class of microbes. And as you can show here, 4 of these Clostridiales species robustly utilize mucin as an energy source. So with this background, what Marta then asked was using a simple epithelial injury model, so the DSS model that both Andrew and Dana just alluded to ask the question, if these mice were then gavaged with these individual bugs, could we then alter the susceptibility to epithelial injury with the idea that if you don't have a mucin barrier, then you're predisposed to additional injury. So what she identified was, as expected, Akkermansia muciniphila treatment attenuates the epithelial injury with DSS, and this has been shown by many labs, but also observed that another Clostridiales member called Peptostreptococcus rosellii also increased weight when gavaged in this setting of this model. Examining sort of the strains that were identified in this setting, we identified there were 5 strains of Peptostreptococcus rosellii, and by PacBio sequencing, Marta and Shengwei observed that there were 2 strains that expressed a phenylacetate dehydratase gene cluster, whereas the Peptostreptococcus tomatis lacked that initiator sequence. So using these 3 microbes, we then did QTOF-MS to try to identify, are there metabolites that could be made by these bugs that could potentially signal in this mucin-dependent program. So what they identified were 2 indole-containing molecules that were differentially produced by the bacteria that contain the phenylacetate dehydratase cluster. So they produce, for example, indole propionic acid and indole acrylic acid, whereas there was a non-differential production of another propionic acid metabolite. What we also observed is that when comparing the sequences of IPA to IA was this presence of this double bond, which I'm going to get to in some of the signaling experiments. So with the availability of these 2 molecules, then Marta did 2 simple experiments. One is to ask the question, how do organoids respond to these molecules? And then how do organoids and underlying macrophages respond to these micromolecules? And notice that both IPA and IA induced the production of mucin 2, so suggesting that they signal to goblet cells, and also increase the production of IL-10, a cytokine that then attenuates signaling. So using a reductionist system of both organoids with and without macrophages, they could show that they both have the properties of increasing the mucin 2 peptide and the increase in IL-10. The phenotype of IA as well as IPA was also seen in peripheral blood cells, showing that they both attenuated the production of IL-1 beta and IL-6. If you then took these molecules and exposed them to peripheral blood cells, you could see that IA, in fact, and IPA to a lesser amount sort of increased the expression of NRF2 genes that are listed here. And the motivation for this type of experiment was the presence of a double bond in IA compared to IPA, suggesting that this might be a better electrophilic compound. So with this data, we asked what is the functional or the human disease relevance for this observation. And so we went back to the metagenomic data sets that have been generated in the lab with both HMP2 support and other data, funding agencies to ask what happens to these two clusters in patients who have inflammatory bowel disease compared to those who did not have. And as you can see here, both patients with Crohn's disease and ulcerative colitis have a reduced incidence of this phenylacetate dehydratase cluster compared to the controls. So in vignette 1, what I've tried to show is that we've used a simple computational approach to identify transporters and enzymes that are important in utilizing or metabolizing mucins. We focused in on Peptostreptococcus as an example and showed that this gene cluster here produces a tryptophan metabolite, indole acrylic acid, and gone on to show that IA in fact has an influence both on the epithelial barrier as well as immune cells, and then gone on to show sort of the functional implications of this observation that this cluster and these microbes are in fact depleted in patients with inflammatory bowel disease. The second approach, and so what do we need going forward, and I think Eric Arm will talk about this, is that we need a better collection of microbes that come from humans with and without disease. We need to try to identify what do these microbes make and then sort of embark on some of the signaling experiments that I've outlined. The second approach that we've broadly taken is to work with well-defined cohorts, and most of these experiments have been done using a cohort of about 1,000 children who were born in Finland, Russia, and Estonia, who basically carry defined HLA alleles. These children were followed for about 3 years, and the reason for focusing on this cohort is that children born in Finland have a very high incidence of type 1 diabetes, as well as various allergies to foods, compared to children born in Russia, who had an incidence that was significantly lower, similar to what's seen in the developing world, and asked the question by sampling these children over 3 years with monthly microbiome samples, can we try to begin to understand, are there microbial factors that might tell us something about this end phenotype? So Tommy Vatanen, graduate student in the lab, set about studying this, and what I'm showing you here are these stream plots that you can clearly see that if you were born in Finland, you had a higher relative abundance of Bacteroidetes, whereas the kids born in Russia had a high incidence of actinobacteria. So with this observation, both using monthly 16S samples and monthly metagenomic samples, we're able to sort of begin to ask what are the pathways that are abundant or depleted in these two communities or cohorts of children. And one of the pathways that emerged from this analysis was there was a differential abundance of lipid biosynthesis and lipopolysaccharide biosynthesis, suggesting that this could be one of the pathways that were differential because of the underlying microbes that are seen in the Finnish kids compared to the kids born in Russia. We then next asked, with this LPS hypothesis, what are the bacteria that contribute to the different types of LPS? So if you were born in Russia, most of your LPS came from E. coli, whereas if you were born in Finland, your LPS came from E. coli, as well as a large group of Bacteroides. So with this observation, we went ahead and purified the LPS that were made by each one of these microbes, and found that if you made LPS that primarily came from E. coli, you activated pro-inflammatory signaling pathways, whether you read out with NF-kappa B or various cytokine readouts. whereas the Bacteroides dorii, which was an abundant bug found in the Finnish kids, clearly had a silent response to these pro-inflammatory signaling pathways. Again, highlighting the value of cohort studies, focusing on humans with serial sample collection. So we then asked, what about the types of LPS that are made by E. coli versus the Bacteroides dorii? As expected, the E. coli makes a 6-acyl chain LPS, whereas the LPS made by Bacteroides both had a penta and a tetraacylated LPS, suggesting that there might be certain structural features for this lack of signaling in the context of pro-inflammatory cytokine production. Tommy and Eva, another postdoctoral fellow, then went on to do a series of functional experiments, and what I'm showing you here are cells that are stimulated with the E. coli LPS that then are treated with increasing doses of the B. dorii LPS. So the 6-acyl chain LPS compared to the 4 or 5-acyl chain LPS. And as you can see here, the B. dorii LPS counteracts the immune stimulation that's mediated by the LPS that's recognized by the TLR4 receptor, both in peripheral blood mononuclear cells as well as monocytes isolated from healthy individuals. And this phenotype is seen both in children as well as in adults. To get to what might be sort of the immunological consequences, I'm going to show you two pieces of data. So as shown here is this experiment where we've looked at the ability to induce endotoxin tolerance, and by both experimental systems, the B. dorii LPS fails to induce endotoxin tolerance as well as prevent the establishment of E. coli mediated endotoxin tolerance. And finally, while the lab is primarily interested in understanding the human microbiome, the reviewers forced us to do one mouse experiment, and in this we used the NOD mouse and clearly showed that the E. coli LPS was a potent educator of the immune system, whereas the B. dorii LPS, which is comparatively inert, fails to signal, in fact increase the incidence of type 1 diabetes or a hypoglycemic state in these NOD mice. So in the second vignette, I've told you that clearly by looking at well-phenotyped cohorts, focusing here on Finland and Russia, where the incidence of type 1 diabetes is very different, we've been able to go on to show that there is an important component of immune education that's required in the first year of life for gut immune development and maturity. And if these children lack these immune education signals, one is at increased risk for autoimmune disease, in this context using type 1 diabetes as an example. The third type of experiment that we've been interested in the lab is to sort of start to do perturbation experiments, again in humans, to try to understand how perturbations might tell us about disease onset and progression. And a gastroenterologist in the department was broadly interested in is how could we think about the microbiome in responses to the current approved drugs to treat inflammatory bowel disease. So despite huge progress, if you take any of the anti-TNF or the integrin agents, basically about 40% of individuals respond to these drugs. And what Ashwin Anandakrishnan in the department was interested was to take individuals who were exposed to a biological agent, so in this case, vedolizumab, which is the alpha-4 beta-7 integrin blocker, and ask the question, what are microbial features in individuals who respond to the drug and stay responsive for 1 year, compared to individuals who respond for a short period of time, 2 to 3 months, and then lose response? And are there features, clinical and microbial, that might tell us something about sustained response in these individuals? I'm going to summarize all the data in 3 slides. So we collected multiple time points, and what Ashwin and Chengwei again were able to observe, but there were clearly microbes, in this case Rosibria, which is a robust producer of short-chain fatty acids. The presence of this was associated with a better response or a more sustained response to the integrin agents, and then went on to identify a series of microbial factors when combined with clinical factors could give us at least a preliminary insight into those individuals who would respond to the drug and stay responsive compared to those who respond and then lose response. hopefully by the end of this year, we'll be able to report on something like 450 children with newly diagnosed ulcerative colitis. So these were kids who were enrolled into this study before they got treatment, and then were binned into the standard treatment regimens that children with ulcerative colitis would be treated with, and to sort of begin to ask the question, how many of those children had severe disease, mild disease, and intermediate disease, and how one might be able to correlate this with both host factors and microbial factors. Basically, I've summarized the work of two postdocs and a graduate student. Most of the microbiome work is led by Hera, who's in the lab, and then key collaborators, Curtis, Eric, and others who've sort of enabled these studies. Thank you.
Thank you so much. We had great presentations, and just to remind you, the purpose of this session was to highlight the mechanistic and specific functional examples of host microbiome interaction and what we still don't understand about those interactions. And before the questions, I just want to give few highlights that I noted. So Andrew just mentioned that we should not tell people what to eat, but he said what should not eat, and he mentioned the emulsifiers. I imagine there are some other examples that he may give with what not to eat. And we also understand from Dana the importance of sex differences, and probably we should look in post-menopausal and this kind of sex hormones. Nita, we understand a lot about the necessity of microbe interaction, and probably microbe fungi, microviromes, and this kind of interaction. And Ramnik explained to us the importance of commensal bacteria, the importance of host genetic variants as would predispose toward certain microbiome dysbiosis, and also geographical differences into the microbiome, which could also predispose towards certain conditions. So now we are open for questions.
Hi, Phil Dashner, NCI. My question is directed mainly at Andrew. So I'm interested in your observation of HCC in those inulin-supplemented knockout mice. Do you have any idea what's driving those hepatic tumors? Is it interactions with the short-chain fatty acids or something else going on?
Yeah, sorry. I do want to say that work is being done in Vijay Kumar's lab in Penn State. I'm a collaborator on it. Essentially, it is mediated by short-chain fatty acids, and that it can be prevented if the mice are administered a yeast product, hops product, beta acids. So indicating it is mediated by short-chain fatty acids, and it's something about the context of the purified diet, dysbiosis, and then short-chain fatty acids that are driving it. Beyond that, we don't have a great sense.
Hi, Matthew Stoll, UAB. This is for Dr. Xavier. Very interesting talk. You showed that the abundance of Roseburia predicted a good response to vedolizumab. Did you also look to see what bacteria predict response to TNF inhibitors, and what are the prospects that we can use the baseline microbiota to guide which therapy to use?
Yeah, so, in the study that's published, we've done a small cohort of anti-TNF effects response with the microbiome, but the hope is that in this large pediatric cohort is going to tell us how does the sulfur agents affect the microbiome, what's the treatment response, steroids, so you do a step-up therapy to try to understand what the effects might be. So I think the larger cohort will hopefully identify additional factors that determine sort of drug response.
Thank you.
Jayajit Das from the National World Children's Hospital. The question for Dr. Salzman. So it appeared to me that spatial aggregation of bacterial species is playing a role in defining the niche here. So do you think that biofilm formation occurs here, and if the mucus layer plays a role in that?
I'm sorry, can you repeat the last part of your question?
So if the mucus layer helps in biofilm formation for this particular species you looked at, or if the biofilm formation occurs at all?
It's an interesting question and a good question. When we look actually by fluorescence in situ hybridization to try and identify an actual physical niche, we're not really very successful at finding specifically where the organisms are. And they don't seem to be associated with sort of large groups of organisms in biofilms, although they are associated with the mucus. And I think the problem, actually, to some extent, we don't have much architectural, associations that we can make. It's not, tips of villi or bases of crypts or anything like that. And the other problem is the numbers are relatively low, even though they seem like they're measurable and culturable. Compared to the populations, they're actually so low that we're having difficulty actually identifying a specific place. So it is a little bit hard to say at this point the role that biofilm would have.
Thank you.
Li Ping Zhao. I have a question regarding the use of DSS induced colitis model. So two talks may be related with this. It's actually a very tricky model, and we found that the load, the opportunistic pathogen load in the facility, in the mice, and from batch to batch, from facility to facility, actually varies a lot. That affects the severity of the disease phenotype. And so some of the results, like Dana, you see no contribution of genetic mutation on the, so just gender and contributed, not the microbiome. Would that be, this problem of not enough opportunistic pathogen load in your mice? You think, is that a possibility for the reason?
I mean, I completely agree with you. The problem I think that we all have with this DSS model is the lab-to-lab variability depending on the source of DSS, the concentration of DSS, the length of time. So all those things, I think, again, need to be reported in papers so everyone knows, everyone's on the same page in terms of the type of DSS they're using. And I guess the other thing to consider too is that something I didn't bring up as well is in terms of animal husbandry, like if they're on acidified water versus non-acidified water, that has also, we've seen, a huge impact on their susceptibility to DSS. So I guess for me it's just, making sure we report all these things in our papers so that other labs can try to reproduce them. How long they're on DSS, in terms of microbiota transplant experiments, how long that microbiota has been in those animals. I think we should have some standardized time where those microbes are in germ-free mice, for example, how long they should be there before you can say that they're actually stably colonized. I think all those things we still need to define in this field. And yes, certainly.
Yeah, just to kind of add to that, it would be nice to have reproducibility across facilities, but it would also be nice if we were working with mice that, as much as possible, were a good model for humans. And I think a paper last year from David Massapust, and I think it was in Nature, showed if you want mice with an immune system that resembles humans, you should not acquire them from Jackson or Taconic or any other commercial vendor, but from your local pet store.
Okay. Yeah, thank you.
I'm Gabriel Nunez from University of Michigan. I have a question for Nita. So very nice presentation about Pterococcus faecalis and bacteriocin. I was wondering, as far as the VRE, you try to use these to kill related VRE, but what about the opposite, that the VRE will express the immunity gene against that, and also could be this used as a way of VRE to emerge after antibiotic therapy, and how much diversity is within these bacteria, how many genes do they have, how much diversity of these molecules, porphyrin molecules exist within the genomes of Enterococcus.
Well, there are a number of different bacteriocins and bacteriocin plasmids that are harbored by Enterococcus and that produce a variety of bacteriocins. The risk, if you don't use a crippled complemented strain to transmit the immunity gene is encoded on the plasmid. So if you can't conjugate the plasmid, you shouldn't be able to conjugate the immunity gene. And so that would reduce the risk. Clearly, there is always a risk with a single approach therapeutic that a certain population of VRE will already harbor that plasmid and that immunity gene, and therefore your therapeutic, like with bacteriocin 21, will not be effective. So there's probably a point, if you want to make a therapeutic, to actually combine a variety of enterocins and bacteriocins together to limit that risk. In the native population, obviously these strains were pulled from the native population. In the mouse gut, which is where we've looked, we've never found the presence of plasmid, and we haven't found it in human isolated strains of VRE, either Fecalis or Faecium. Doesn't mean it doesn't exist, though.
You mean the immunity gene?
Well, the immunity gene or the bacteriocin plasmid. Usually they are encoded on the same plasmid.
Thank you.
Hi, Alexandria Schubert from the FDA. And my question was also for Dr. Salzman. I wanted to know, or just get clarification really, on your VRE mouse model. You had said that you didn't want to use antibiotics because then you're looking not at colonization but infection, or did I get that reversed? And I was wondering if you could clarify, because I thought that you could pre-treat with antibiotics and that wouldn't necessarily lead to an infection by VRE. It's not really, I thought, a gut pathogen per se. It's more of a problem when it gets out of the gut. And
what we find, and we've limited to cephalosporin antibiotics to try and mimic sort of a human system, that you get translocation. You don't get infection per se. In other words, the gut does not show inflammation when you get sort of expansion and translocation of enterococci in response to antibiotic treatment. You get systemic spread of enterococci. But if you use antibiotics to establish VRE in the gut, you've actually disrupted the standard interactions of the microbiota. So you will have disrupted any aspects of colonization resistance, bacterial host interactions, and that's what we wanted to avoid.
Okay.
We wanted to understand, at least to the extent that we could, the native interactions of enterococci in its environment without having massively disrupted the other colonizers. When we colonize with enterococci that lack the bacteriocin, we don't find any changes, actually, significant changes in the microbiome at all. Only when we use the PPD1. So that's probably even a better system to understand true mechanisms of colonization.
Okay, thank you.
Hey, I'm Peter Turnbull, UCSF. I'm wondering whether or not you guys have thought about ways of dealing with these. There's many examples now, like the inflammasome studies that don't replicate in one lab, whereas there was originally this really interesting finding. Like, how do we, or how are you guys thinking about tackling that? Is there a way to reconstitute the microbiome from the original studies and try to, rebuild it in Toronto or Belgium or wherever else these studies are taking place? Can you actually get the microbes from the original investigators and not only reconstitute the transgenic model, but also the microbiome it started with? Or, what are you thinking about doing?
Yeah, that's a great point, because I think, there are studies out there with NLRP6 knockout mice, NOD2 knockout mice, that show that they do have an increased severity in colitis. We've never seen it in our mouse colony, but people have seen it. And it, likely because within their facility there's a pathobiont that seems to trigger the inflammation in that particular genotype compared to the wild-type mouse. So one way to get around this, it would be pretty difficult, would be to try to use defined communities of microbes that are, defined as a healthy microbiota versus one that is procolitogenic, and try to tease out the factors from those organisms that lead to the triggering of disease in that particular genotype. That's one thing we've been thinking of, but I think we're far from that at the moment. And then again, to try to see if other labs can verify your results. That means more collaboration and discussion with other groups, but can other people replicate what you've seen in their particular facility with their microbiota? Are we going to have to report, the whole microbiota of all our mice? That might be part of what we have to do in the future. But again, I need to reiterate that you need to also think of other microbes. As I told you, we had this parasitic infection that completely flipped the intestinal phenotype in our animals. So it's not only the bacterial microbiome, but other microbiomes we have to think about as well. Did you want to add to that?
Well, I thought Thad really outlined a lot of the things that would be very helpful, but these things won't happen unless the incentives are changed. If someone is going to primarily publish papers that says, I examined this in my facility and I found the same thing, that person will quickly be unfunded and probably unemployed. So with the current incentives in place, where there's really a focus on novelty over rigor, at both top journals and funding agencies that really rely on those journals, we can say it'd be nice for these things to happen, but they won't happen.
Now, last question.
Yeah, I want to direct the question to Dr. Salzman and Dr. Xavier. Do you think it's possible, when you talk about are we losing old friends, that in the virulence and antibiotic resistance, the virulent organisms in their genes that are producing the antimicrobials, the arsenal of antimicrobials, is just this kind of war going on between the more virulent and the more commensal. And those commensal, especially in the Bifidobacterium lactobacilli that we use in probiotic, are actually going into dormant states that are sort of endospore, going into a sporulation state in these horizontal transfer of losing gene, adding gene, losing gene, adding gene, as this interaction goes on in evolutionary biology, and that when we really get to addressing the antibiotic resistance problem, it will give us clues as to how to restore better the commensal that you think we are possibly losing. And it's not just an issue of diet, but it may be an issue more of what we are beginning to call probiogenomic.
Thanks. The first thing I wanted to say is I wouldn't ascribe antibiotic production by bacteria to be equivalent to virulence, because the production of these bacteriocins by commensals, or actually other antibiotics by commensals, because bacteriocins are clearly not the only antibiotics produced by bacteria, are for colonization more than virulence. So it's actually more of a benefit for colonization as opposed to invasion. In terms of restoration, I don't know if Ramnik would actually speak to that a little bit more directly in terms of restoring commensals.
Restoring the commensal bacteria.
the model systems, so we've relied entirely on initially working with human samples, so tracking back is a hard experiment. And so I think some of the concepts you raised are very relevant. I think we need to find systems to sort of look at bacterial states and host states to sort of see how one might sort of push in both directions.
Yeah, thank you.
Yeah.
Thank you so much. One round of applause for our speakers, and we'll be back 10:55. We'll be back at 10:55 for the next session. Thank you.
Well, welcome to the second part of this morning session. And my name is Roberto Flores. Thank you. My name is Roberto Flores, and I'm a program director and the Nutritional Science Research Group in the Division of Cancer Prevention and the National Cancer Institute. Thank you, Lita. So please sit. So again, my name is Roberto Flores. I'm a program director at the Nutritional Science Research Group, Division of Cancer Prevention in the National Cancer Institute. So the session for this topic is a continuation of a continuum of this host microbe, now is microbe interactions within the human microbiome. And the session purpose is to highlight concrete examples of how microbe, and interkingdom interactions either affect or regulate human and health disease, and then identify the gaps of knowledge in our understanding about the role of microbe interactions in these conditions. So we have an excellent panel of speakers, and our first speaker is Gautam Dantas from Washington University in St. Louis, and his talk title is Understanding and Predicting the Impact of Antibiotic Therapy on the Developing Pediatric Microbiota and Resistome. Thank you.
Well, thanks, Roberto, for that introduction. As you can see, I've modified my slide title to be a little bit more zoomed out. Thanks also to Alita and the rest of the organizers for putting this on and for the invitation. And as a personal thank you to all of the NIH for funding all of us with this good work. I will also follow in Jeff's lead to start out first by acknowledging the incredible group of folks that I have the privilege of working with. I'll highlight those who led the particular vignettes that I'll mention today, but just to start out by saying that anything of importance I say today was done by these folks. I took the prompt from the end of the day yesterday to switch out the order of the challenge question to seed the context of my talk. And I thought that an interesting challenge to consider, which is a theme that we've heard before and we'll continue here, is how, at least in the ideal world of a hypothesis-based microbiome study, how do we accurately define the resolution of various omics studies that are required to answer that specific biological question? This might seem like a really simple thing, but in this day and age where we really are in a systems biology field where we're tempted to collect just as much data as we possibly can and then post hoc analyze. And so say we did not want to do that, what standards, what we need? Is it even appropriate to think of that in terms of what is the appropriate resolution to answer very specific biological questions? And again, we've heard this at least specifically with DNA and RNA omics, the type of method you use. Do you use shotgun sequencing, amplicon sequencing? Do you go into the single cell of the population? Do you do longitudinal studies? And then, of course, where do you call particular molecular entities? And is that really important for the question? Now, the flip side of this, as I alluded to, is one aspect of doing systems biology, and microbiome really is a systems biology field, is you do have this opportunity for yourself as well as the rest of the field to do post hoc analyses. And so what might you want to consider in terms of maybe diving a little bit deeper than the question you want needs to enable those studies? And how does that work into the way you'd write your grant application? And then finally, to go even one step further, not only do we want to understand the systems we're studying, we'd like to be able to predict. right? The real power of doing systems biology is this hope that you can pattern recognize so that the next study really builds off the study you have, and maybe you can do things in higher throughput. And so this, again, really comes down to a definition of resolution of what you're studying, such that hopefully in the future you and others might be able to predict. So that's my challenge slide. We and others, I think this is a theme that goes through almost all of the talks here, are very globally interested in being able to predict how microbial communities respond to perturbation, right? We've heard about this in virtually every talk. And we're very, very fortunate. I think it's important to note that many of the tools we use come from quantitative models that borrow from macroecology. And so you'll see through my talk this notion of being able to understand how either short perturbation or long perturbations result in changes in states, and what is the acute nature of that or the persistent nature of that. And so that's the sort of global framing. And the particular perturbation that my lab has focused on for the last many years to different microbial communities, including the human microbiome, are antibiotics. Now, it should be obvious why. It's because antibiotics are perhaps the greatest perturbation you can think of in terms of microbial life, because they're designed to kill microbes. And what's really important to note, and this audience certainly understands it, is that even though we think of these magic bullets as targeting specific parts of life in pathogens, all of those particular targets are shared across the entire domain of at least the bacterial kingdom. And so for that reason, there have been lots of correlative studies that have looked at, over the large span of human life, from early infancy, after birth, all the way up to old age, how various types of antibiotic perturbations can lead to changes in host phenotypes. And what we're really interested in understanding is what are the specific natures of specific antibiotics that can be used to understand the responses of microbial communities, and then again, hopefully in the future, understand what collateral damage that various antibiotics might engender, so that you have a choice that can be made from a personalized medicine perspective. Now, one of the major consequences, obviously, of antibiotic use is antibiotic resistance. These are stats that hopefully you've seen before. I'd really like to emphasize them, though, because antibiotic resistance in pathogens is perhaps our greatest infectious disease challenge right now. Right? Resistant infections claim about 700,000 lives around the planet. It's been estimated if we don't change the game in terms of resistance understanding and new antibiotics, that number will bloom to 10 million deaths by 2050 per year around the planet. To put that in perspective, imagine this meeting being held in 2050. Over the course of the meeting, about 86,000 people would die from drug-resistant infections. In addition to the huge human burden, there's also the cost. The estimate of this problem to the US economy is on the order of about $55 billion. And we scale this 10 million deaths to cost to the global economy cumulatively, it's about $100 trillion. $100 trillion is the last 6 years of the entire US GDP wiped out. And unfortunately, to make this even worse, at exactly the time that we've got these rising infections, we've got fewer and fewer drugs coming to market. So this is sort of the motivation for why we want to study this problem, both from the pathogen perspective, but also in all of the other communities of microbes, like the microbiome, that they interact with. Now, we all realize that any scientist worth their salt now associates themselves with their own ohm, so we have our resist ohm. I'm using that phrase here to describe any collection of resistance genes in a particular community or sample. And we recognize that, of course, we can turn to culture to look at the resistance genes in that culturable population. And we can complement the cultured work with, say, shotgun sequencing to look at all of the microbes in that community. And these methods can iterate between each other to ask questions about, for instance, antibiotic resistance. The problem, though, is that this is a self-fulfilling prophecy, right? You can only iterate on the known with the shotgun sequencing. And so what we've done, standing on the shoulders of giants like Joe Handelsman and John Clardy, is we've turned to this method called functional metagenomics, which allows us to expand this unknown part of the resistome pie. And what that really means, this method is actually a fairly simple method. You start out, and rather than sequencing your metagenome, you first clone it into an expression system, pop it into something like E. coli, and then screen those recombinants on antibiotics using a culture-based system, and now those recombinants have functional antibiotic resistance genes. All we've done as a lab to increase the throughput of this by about 100-fold is marry it to standard techniques in next-gen sequencing and the development of some computational methods. But really, the output of the method is that now you have a catalog of resistance genes that are compatible with horizontal gene transfer into a model pathogen. And then you can use that to infer, again, now how the microbial communities are responding specifically from the perspective of resistance genes. And we take an ecological view of this problem, not just in terms of the ecology of the gut microbiome of humans, but the ecology of all of the microbes and all of the various habitats on the planet to try to understand these transmission dynamic networks of the microbes under antibiotic perturbations, and then how they might transmit through the human microbiome into pathogens. So I'll share with you first just a couple vignettes of things we've learned from a very ecological basic science perspective, and then end on a longitudinal analysis of the pediatric microbiota under antibiotic selection. So we've used these type of methods along with the other various omics methods available to us to establish such things like, the soil resistome, so the resistome of benign bugs in the soil is exceptionally extensive, but fortunately most of it appears to be not as easily set up for horizontal gene transfers in pathogens. So that's a good part of the equation. However, we found that there are particular microbes that defy those trends, that appear to be particularly facile at crossing habitat barriers and also at horizontal gene transfer, in this case, the multi-drug resistant proteobacteria. So this allows us to start zooming in maybe the vehicles of these particular transmission events. We've also then looked at co-localized systems of both environments as well as human microbiota. And so in this study led by Erica Persson and Pablo Tsukuyama in our lab, we looked at a village in El Salvador and a slum outside Lima, Peru, which in terms of their lifestyles, these people represent about 2/3 of the human population, and asked questions across both, again, their fecal samples as well as environmental samples around their houses and as well as the wastewater treatment plants. For instance, how these people's gut microbiota, how are they structured compared to other microbiomes that have been looked at? And so we find, and confirming hypotheses that others have put out before, that the human microbiota really does seem to be structured by lifestyle as opposed to simply by geography, right? That these communities, when compared to either Western communities or hunter-gatherers, they fall in between along this gradient. And then when we zoom into both the taxonomy as well as the resistome of just the samples that Erica and Pablo looked at, again, these seem to be largely structured by habitat, right? That there is some kind of signature of the type of bugs that are in the gut of the human that are different from the type of bugs that are in the gut of a chicken, and the resistance genes generally follow that trend too. So one inference from that is perhaps there's not a ton of horizontal gene transfer going on across habitats, or at least it's not overwhelming the system in terms of sequence signatures. And so what we do then is go into this and ask for which are the genes that break those trends, because those are the ones that are going to be hotspots for transmission. And we've done that. Here are just two examples of things that Erica and Pablo found. Chicken coops in El Salvador and wastewater treatment systems in Peru seem to be really enriched for these horizontal gene transfer events that make the bugs that are compatible with the environment also compatible with the human gut. And so this has obvious public health benefits in terms of knowing that this exists, because, for instance, in Lima is a desert, so all of this apparently cleaned water, which is still enriched for resistance genes, is then used to irrigate basically every park in Lima. Okay. All right, so that's the end of the sort of survey part of the resistome. What we're trying to now do to complement these sort of basic science ecological findings is really turn to various cohorts of the human microbiota where we can do longitudinal sampling, where we have exquisite information about specific antibiotic treatments before, during, and after, and trying to get to predictive models of how the microbiome responds. And in one particular suite of studies, we focused on the developing pediatric microbiota. And you've heard many, many stories already as to why that's particularly critical. We know from Jeff and others' work that the first 3 years of life is really this critical time window when the ecosystem is established. So you could argue if you care about perturbations, that's really where we want to look, because then they might get fixed later. We've gone even one step further back, and that is looking at when the system is even more vulnerable, and that is in the state of prematurity, right? Arguing that your gut and your immune system is immature in prematurity, and then so is your microbiota. But moreover, the reason we care in this case about preterm infants is because of their high risk of infections, they just get an insane assault of antibiotics. And so in our cohort of kids who are about born 10 weeks too early, almost every single one of them receive antibiotics for the first couple of days of life. The top 2 drugs that these kids get in the NICU are antibiotics. The top 4 drugs that they get out of the top 8 are also antibiotics. So they're really, if you think about it, here's a developing ecosystem that is really carpet bombing with things that are going to disrupt it. Okay? And so Molly Gibson and my lab published on the first phase of our investigation of what happens to these kids' guts in terms of their composition, who's there, how many, as well as specifically the antibiotic resistance genes across a set of 84 preterm infants. We had samples in collaboration with Phil Torr and Barb Warner during the roughly 2.5 months of stay in the neonatal ICU. We had stratified this cohort in terms of kids who only had antibiotics for the first few days of life versus kids who had up to 20 to 30 additional days of antibiotic treatment. And then using, again, a suite of various omics methods and some computation analysis, here are some of the general trends that we found. So Molly saw that, indeed, she could confirm that the gut microbiota of these particular preterm infants is substantially reduced in terms of its diversity compared to age-matched term infants. But more importantly, the type of bugs that you see in the guts dominating these preterm infants are not what you might associate with health, right? These are the type of bugs that you'd associate with being on the CDC's list of stuff that should scare you, right? And obviously you would argue that that's not a good system to have in a preterm kid's guts. But then we start diving into this real tantalizing possibility of finding antibiotic-specific effects. So one of the things that Molly sees is that for very specific antibiotics, she can see specific taxa that are dominant go up or down. We can also extend this out into the collateral damage in terms of antibiotic resistance gene enrichment. And so here she can see that, again, in terms of specific antibiotics, you can see specific types of antibiotic resistance gene enriched in these specific states. And then you can begin to model, right? You can statistical associations, not just with the microbiome data, but all of the rich clinical metadata that you have with these kids. Molly applies a generalized linear mix model to understand how species richness, a kind of measure of microbiome health, changes, and was able to identify, in fact, 3 antibiotics that clearly depress the diversity in these kids' guts, and then actually also identify these other correlates which increase diversity, age as well as maternal breast milk, again, consistent with things you've heard about before today. And then finally, in the cases where we've got these really nice match samples before and after an antibiotic treatment, she was able to apply a machine learning approach where she was able to identify for the 2 major antibiotics used in this population, just 4 variables, 2 antibiotic resistance genes and 2 bacteria that allow you with 85% accuracy to really know what direction this is going on. So this is obviously early days, but this has, again, the hope of a personalized medicine approach to measure the impact of antibiotics. And so I have one more slide, and I'm going to steal just a few seconds of time to tell you what we're doing right now, thanks to both NIH as well as CDC support, is to say we've got a nice picture of the acute perturbations, but what happens later in life with these early life antibiotic treatments? And so with an expanded cohort, now Drew Gasparini is finished analyzing samples from 60 individuals where we have samples out to 2 years of life, added on a healthy cohort that has no antibiotics to set up sort of the matched healthy control. We've now screened every one of these samples with some improvements with our functional metagenomics for resistance genes. We've also added on a culturing component to see whether enriched drug-resistant organisms persist out to a couple years of life. And then finally, using models you've heard about before, we've established a transgenerational humanized notobiotic system where we can take these acute perturbations and really test whether our computational models hold up to repeated scrutiny using mice. And so I'll end again there. Thank you. Thank the folks who've funded this, and leave you with this before we transition on.
Thank you, Dr. Dantes. Our next speaker is Mahmoud Ghannoum from Case Western Reserve University. His talk title is Bacteriome and Microbiome, Polymicrobial Interactions Define Health and Disease, a New Paradigm.
First, thank you very much, the organizer. I really appreciate Roberto and everybody who asked me to come here, so it's really great opportunity. What's really important, I think, I'm gonna convince you that it's not only bacteria, it's not only fungi, but both of them, they work together to cause problems. And that's how we are gonna start talking about. So to start with, how do I know that? I want to tell you a personal story. When in 1974, I started to do my research, and my supervisor gave me a paper where rabbits treated with antibiotics, guess what happened to them? They developed fungal infections. So it became very clear to me that, fungi, not only they negatively impact us, but also they live in our body as commensals, and they are sometimes very beneficial. And also it became clear to me that if you get rid of one community, what's going to happen is the other will take over. And guess what? It was time runs really and flies, as you say. 43 years ago, I figured that out. Okay, so I tried to, when all the hype and everything going about the microbiome and everybody talking about the bacteriome, in 2010, I wrote an article to ASM Microbes saying, look, we need to look at the fungi as well. Since I am a fungi, I need to start working on this. So what happens, nobody, of course, listened to me. So in 2016, I went again and wrote an article in The Scientist, and there I called it the microbiome, which is really the ignored kingdom. And in that article, we showed that if you look at 2015, that there are nearly 800 articles, 737 of them were on bacteria, on viruses, and I'm not biased against it also. Just remember, we need to think of virus as well. But only 12 articles on the fungi. So to me, I opine that it's really very important that we should look at both in the same sample, because this is going to help us how to really treat with disease. The good news, people are listening, and we are starting to see a number of papers where they characterize the microbiomes at different body sites, and I want to look at particularly about the gastrointestinal tract. So I'm going to tell you the story of the interaction between the bacteriome and the microbiome as it's told through the Crohn's disease. Okay? Now, as you know, Crohn's disease is a multifactorial. So you have genetic factor, you have the intestinal microbiota, as well as the dysregulation. So the three of them work together, and it is well established. We heard in the last 2 days about the role of bacteria a lot of the time in Crohn's and ulcerative colitis. The first paper that started looking at the fungi as an important player in Crohn's disease came from Alif, David Underhill group, and they used the SS, the favorite model for everybody for the colitis. And what they found that in the gut is colonized by 10 fungal species, and really by far the most predominant one was Candida tropicalis. And their conclusion was that fungi and not bacteria is responsible for aggravating the severity and inflammation in CD patients. At least in mouse models, that we started to see that fungi could play a role. That's where we came. I started collaborating with groups of my friends in France, and we looked at Crohn's disease patients. Instead of having Crohn's disease and unrelated controls, we thought it's better to look within the family. So we looked at the people with CD, their relative who live with them, so that to try to reduce at least to some extent the genetic as well as the diet factors. But also we looked at another 4 families where they also are healthy but live in the same region. So this is starting to look at the PCA. We not only looked at the fungi, even though I told you I love fungus, but we looked at bacteriome, and the microbiome as well. And you can see when you look at the PCA, really there is no particular distribution here or clustering. CD in red, NCR the relative in blue, and green not relative but healthy. Whereas when you look at the microbiome, look at the red and blue, the CD and their relatives, they are very close together. So in a way, if you want to try to compare the microbiota in CD and people who are not related to them, you are not really getting a lot of the picture. It will be better to look at within the family. And here is the heat map, and you see the bacteriome, microbiome, similar really, the usual suspect as you expect. In the fungi, you see a lot of the Ascomycota, you have the green also. That's one point which is really lacking a lot in the fungal field, is that we don't have enough databases. This is one thing which we really need to work on, but you can see in general about 4 different phyla there. If you look more specifically at different taxa with bacteriome, this is CD, their relatives, and the other control group. And look how close they are, the CD and their members who are non-CD. Again, across all different taxa level, the same applies for the microbiome. Again, they are very close. So it's a good idea to look at within the family, as I mentioned. And we did this here. And this is for the bacteria. These are the abundance of bacterial taxa. You can see between the CD and their relatives, there is a more Oscillospira, Provitella more in the, again, the healthy relatives. And this is no surprise to anybody. More Faecalibacterium protozii is really in healthy people, which have been shown but by others. When we looked at the fungi, you can see Saccharomyces species. There is a trend in favor of where you have higher levels of Saccharomyces in healthy. Also, Saccharomyces cerevisiae. When you take all the Candida together, there is no all species there. You don't see any difference. However, we found that, like they showed in their animal study, that the increase was statistically higher Candida tropicalis in these patients. This is all and the only statistically significant was, as you can see, with Candida tropicalis, that it is higher in CD patients. Another important observation is there is a test called ASCA, or anti-Saccharomyces cerevisiae antibody. This is usually used to look at CD patients as a diagnostic markers, and it is elevated in them. And that's exactly what we saw in our study, elevated CD, not in the relative and the control. But really what we saw interesting is that ASCA was correlated with the Candida tropicalis. We then went into bacterial, and bacterial fungal interactions. Now, I don't expect you to read this, only to look at the colors. Blue, this means positively correlated. Red, they are antagonistic or negatively correlated. When you look at the fungi, you can see, for example, this is in CD patients, Candida and Saccharomyces, they seem to be antagonistic. When you look at their relative, there is no interaction inside between them. So we then looked at, is there a correlation between Candida tropicalis and other bacteria, and we found that there is 3 different organisms, Serratia, E. coli, and Erwinia, were statistically significantly associated in a positive way, and also, especially E. coli and Serratia were increased. Because these organisms in the gut can form biofilm, as we started looking at the 3 organisms, Tropicalis, E. coli, and Serratia alone, and then we put them together. So you can see here, Candida tropicalis alone usually is yeast form. You start putting it with E. coli, Serratia, or you put the 3 together, and look what happened to it. You have a lot of hyphae. And as you know, filament formation or ability to form hyphae is a virulence factor for this organism. So it's there, they form the biofilm, and guess what? They start to have hyphaeation where it allowed it to invade the epithelium, the gut epithelium. So we measured the thickness of this biofilm, and you can see here it's much easier quantitatively, CT alone, with EC, with SM, and you put the 3 together, it's really very, very thick biofilm using confocal microscopy. So it forms a robust biofilm. And this is some pictures about scanning electron microscopy, which is going to show you some interactions. This is again CT alone, really beautiful yeast. Look, this is with E. coli. And To our surprise, it was fused with Candida tropicalis, whereas Serratia had different interaction, which I will show you. This is the three together, and in general, Serratia it just sat on the on top as when you look scanning electron microscopy. We looked at transmission electron microscopy, and look what happened. This is the Candida, and this is all the different organisms. You find that with the E. coli is very nearly fused, whereas Serratia, it produced these filaments which bind not only to E. coli but also to Candida, so it's forming a bridge to stabilize the biofilm there and make it stick much better. So this was all in vitro. People saying, yeah, but you don't have the environment of the gut anaerobic organisms, so we developed the model, and this is just recent data where we had DSS model, no organisms really. You see some mucus, you see intestinal epithelium, and then look at this large biofilm forming in the colon of DSS mouse plus the 3 organisms when you have it. So we showed it both in vitro and in vivo. Now, another thing, when these organisms come together, do they do anything with respect to the metabolites? So we grew, the question was, does this interaction alters the metabolites secreted? So we did all these like alone, medium alone, and this sort of thing, and then different combination 2 and 3 together, and look what we found. It's really very, very interesting. So this is medium alone, this is Serratia, this is where is one of them, E. coli? Oh, here, E. coli. I'm getting blind now. And then when you put them, the 3 together, you can see definitely different metabolites are produced. So not only they come together, but also they alter the metabolites secreted. And when we look at that, in total, we found 299 metabolites detected, and 8 of them were statistically significant. One of them here, oxoproline, was 130-fold higher than when you have them alone, like Candida tropicalis and Serratia. So definitely, this is a way which we know we are looking at, not only with respect to how they interact, help the interaction between microbe, but also how this interaction is affected by the host as well. So this is a summary of the model, which is, this is in healthy gut. You have nice homeostasis, very nice mucin, no cell damage. You make a biofilm, and then they start secreting their aspartic proteinases and mucins, for example, by Rhamnococcus, and you break the mucin, and then fungi form these hyphae and start to invade and breaking the tissue. Okay, so this is our working model. We published this paper in MBio, and we didn't publish yet the metabolites, but this is with respect to the interaction between the bacteriome and microbiome. And what's nice about it is really was received very, very good attention and encouraged us a lot. The last thing I want to tell you about this cooperation, I don't think cooperation only occurs on Crohn's disease patients. This is another paper from University of Pittsburgh where they asked me to write a commentary in MBio, and guess what? They saw in wounds exactly what we are seeing in Crohn's disease, where bacteria and fungi cooperate together. So we need to start to think about how can we manage this, how can we break this biofilm, whether we use antifungal, probiotics, because there is no way you can get rid of these organisms. And now we just did a study which is we just submitted in cancer patients with cancer to the tongue, where we are seeing similar sort of interaction. So bacterial interaction and with fungi really spans different diseases. So to me, this is a what I believe is happening is that the organisms have evolved to form a cooperative strategy to try to attack, really benefit themselves as well as harm the host. Fungi will gain virulence factors, and I showed you the filamentation. Bacteria living inside the biofilm become tolerant to antibacterial, and also they start secreting all these enzymes, which, guess what, they break the dysfunction of the gut barrier and really cause damage. The host is impacted negatively, where you have, under the influence of these organisms, you have immunomodulatory components of the fungal biofilms, increase in the level pro-inflammatory cytokines, and causing oxidative damage and cell death. So this is the gaps. I think we need to start looking at the polymicrobial organisms. And also, we need to, you heard so much information we have. How can we harness this information through the Human Microbiome Project, all of this? How can we now look at it and start to focus a little bit? To me, studying monospecies biofilm is not the way. Neither investigating the entire microbiota at once. For example, I put a grant and they say, oh, you know, it's nice to have these 3 organisms, but you should study all the microbiota. I don't know what they were smoking, but this is not gonna happen, because when you put 3 organisms, they tell you this is very complex. I say, okay, that I agree, but now put all microbiota. Well, let's not go there. Now, there is a need to develop multiple resources, imaging and in vivo models, to address these polymicrobial organisms, define the mechanism underlying the interaction, not only between the organisms, but also between the organisms and the host, and also define how the metabolites interact with these. And this is, I'm gonna finish with this last slide. You know, this is the way of the future. We always look at the mother, which is very important, and to me, the mother is the fungus, and of course, we need to look at the babies as well. Thank you very much.
Thank you. Our next speaker is Michiko Taga from the University of California in Berkeley. Her talk title is Decoding Nutritional Interactions in Microbial Communities. Dr. Taga.
Okay. Well, I'm a molecular microbiologist, so what I'm most interested in is the molecular interactions that occur between microbes within their communities. And so we're doing studies mostly of single bacteria, and one of the challenges here is to make generalizations about what's happening in the community based on what we're doing experimentally with single bacteria. And related to that, probably within the next year, we're going to have 100,000 bacterial genomes and also many, many metagenomes. So with all this abundant sequence data, we're still limited by our ability to decode these genome sequences to make the most of the sequence data that we have to make generalizations of our experimental data and understand the bigger picture of what's going on in microbial communities. And so one of the keys that I think will aid in this transition is to incorporate bioinformatics using the sequence data that we have when we're doing these experimental studies using microbiology, genetics, biochemistry, et cetera. So my lab largely focuses on this molecule here, vitamin B12. And so I'll quickly tell you about the structure. B12, or cobalamin, is a tetraperol like heme. It has a cobalt metal at its center, which coordinates an upper ligand that can do radical chemistry or methyl transfer reactions. But we're most interested in the lower ligand, which, for reasons that will become obvious soon. So coronoids are the vitamin B12 family of molecules. And these are critical metabolites in bacteria. So genome analysis tells us that 86% of bacteria use coronoids for some aspect of their metabolism. Only 14% do not use coronoids. So in, for example, the human gut, coronoids are important cofactors for a variety of different metabolisms. For example, two-thirds of bacteria in the human gut have a coronoid-dependent methionine synthase. Coronoids are also used for nucleotide metabolism and various carbon metabolisms. So what makes us think we can learn something about microbial interactions based on studying coronoids is that the majority of coronoid-dependent bacteria cannot make their own coronoids. So almost half of bacteria are coronoid auxotrophs, meaning that they have at least one coronoid-dependent enzyme encoded in their genome, but they are unable to make coronoids. They don't have the genes for complete coronoid biosynthesis. Instead, these bacteria rely on coronoids produced by the bacteria that do produce coronoids. So to make matters even more complicated, coronoids are structurally variable. And specifically, there's variation in the structure of the lower ligand of coronoids. And here I'm showing you some examples of different lower ligands found in coronoids produced by different bacteria and archaea. And then the important thing about this is that these coronoids with different lower ligand structures are not functionally interchangeable. So this means that bacteria that are in a community with coronoids have to somehow obtain the specific coronoids that function in their metabolism. So we see that multiple coronoids are present in various microbial community samples. And so these are coronoid profiles measured by LC-MS. So in these plots, each color represents a different coronoid structure, and the height of each bar represents the relative abundance of those coronoids. And you can see in these samples were done by Bob Allen, and these were done by us. You can see multiple coronoids are present in different amounts. And so we know that there are multiple coronoids in these microbial communities, and what we're interested in is understanding coronoid sharing and coronoid specificity. And so breaking it down, we're interested in specifically, we've done a lot of work on specificity in coronoid biosynthesis. So what is it about the genome of these bacteria that cause it to produce specific coronoid molecules? We've also looked at coronoid transport in collaboration with Andy Goodman. There's also specificity in the modification enzyme for the upper ligand, which I won't talk about, as well as modification of the lower ligand and coronoid-dependent enzymes and regulation. And I'll talk about these three briefly today. So first, the lower ligand modification. So this is one example of a strategy that a coronoid-dependent bacterium uses to get the coronoids that function in its metabolism from the environment where there are multiple coronoids. And so Dihalococoides McCartyi is a bioremediation bacterium. We found that it can only use 3 of the 9 coronoids that we tested. And these are just the lower ligands of the coronoids that we looked at. So it's very particular for the coronoids that it needs. But it can take up any coronoid as long as it has also a lower ligand of one of these coronoids available. And it's able to remove the lower ligand of the coronoid it cannot use and attach the lower ligand of a coronoid it can use to produce a coronoid that functions in its metabolism. So again, this is one strategy of one bacterium showing how it can get the coronoids that it needs. So now I'll talk about some unpublished work on what we're doing studying specificity in coronoid-dependent enzymes. So we're using the enzyme methylmalonyl coenzyme A mutase, MCM, as a model enzyme because it's very widespread. It's in all 3 domains of life. It's actually one of the 2 coronoid-dependent or B12-dependent enzymes that humans use. And this is actually the structure of the human MCM. So we know that coronoid-dependent enzymes from organism to organism have different specificities for different coronoids, and we want to know the molecular basis of that specificity. So we're interested in figuring out specifically in this lower ligand binding pocket, what is it about those protein residues that make the coronoid-dependent enzyme bind and use specific sets of coronoids? And so Olga Sokolovskaya, who's a graduate student in my lab, is looking at MCM orthologs in vitro. And one of the assays she's doing is a binding assay that's dependent, that's based on the quenching of intrinsic fluorescence of the protein. So she looked first at the MCM enzyme of Cynorhizobium mellilodi. This organism normally produces cobalamin, which is shown in this cartoon diagram here. And so you can see from these fluorescence quenching binding assays that the Cynorhizobium MCM has highest affinity for its native coronoid, and this curve is shown here. And we can use these curves to calculate the binding constants. A structurally very similar coronoid binds with slightly lower affinity, and coronoids with purine lower ligands bind with much lower affinity. So that's one MCM showing that the binding affinity of the protein matches the coronoid that the organism produces. And Olga also looked at 2 other orthologs of MCM and found the same pattern where the coronoid that's produced by the organism is the same one that has highest binding affinity for the MCM. So using those kinds of examples, we are trying to do, we're trying to use bioinformatics to identify the specific sequence motifs in this binding pocket that confer specificity for different coronoids. And so in this alignment, we separated the sequences from bacteria that produce or are thought to use coronoids with a benzimidazole lower ligand, like B12, versus coronoids with purine lower ligands. And you can see some covariation in some of these residues, which are highlighted in the structure here. And so this is very new, so I don't have data yet, but we're doing site-directed mutagenesis and looking at the affinities of mutant proteins for coronoids based on this kind of analysis. So finally, I'll talk about regulation by coronoids. So many coronoid biosynthesis genes and also coronoid transporter genes are regulated by an RNA element called a riboswitch. And so these are, so in general, riboswitches are regulatory elements that when the ligand is not present or the ligand, like for example, a coronoid, is in low concentration, the RNA is in a form where the downstream gene is able to be expressed. When the ligand is at higher concentration, it binds the RNA, and in this conformation, a terminator hairpin is allowed to form, or a hairpin that sequesters the ribosome binding site. And so in this conformation, the downstream gene is not expressed. And this is the way that bacteria regulate the expression of genes, for example, like I said, for biosynthesis or transport of the ligand. And so we think that riboswitches should have specificity for the coronoids that function for the organisms so that they shut off gene expression only when the functional coronoids are present in the cell. And so another graduate student, Chris Kennedy, is at sort of the beginning of this work where first he made riboswitch reporters with M. cherry and expressed them in E. coli. And we want to know, first of all, just is there specificity in riboswitches for different coronoids? And so I'll show you data from 2 riboswitches from Desulfa bulbus, which shows you that different riboswitches have different coronoid affinities. And so as we increase the concentration of coronoid, you can see that this coronoid and these coronoids are able to robustly shut off gene expression of the reporter. And that these 2 riboswitches have different coronoid specificities. This is kind of curious to us because these 2 riboswitches exist in the same cell, and they actually co-regulate the same genes, which I don't have time to talk about, and actually we don't know the reason anyway. So, so those are some of the examples that we are using experimental microbiology molecular biology and biochemistry to study specificity in coronoid biology. And so getting back to these critical challenges, what we hope in the long term is that we will learn enough about coronoid specificity from these experimental studies that we'll be able to use bioinformatics in conjunction with these experimental studies to be able to predict coronoid metabolism. And so the sort of long-term dream is to be able to look at any bacterial genome and be able to know which coronoids are needed by that organism for which kind of metabolism. And if we are able to do that, we don't have to do experimental studies on every single organism. And instead, we can use bioinformatics. And even longer term, we hope that we can use that knowledge to someday perhaps create mechanisms to modulate a microbiome based on some kind of coronoid-based manipulation. So with that, I'd like to thank the members of my lab who contributed to this work, as well as the collaborators that I mentioned here. And I also want to thank the NIH for supporting the work that I talked about and the work in my lab in general. So thank you.
I want to thank Dr. Teaga because we gained 1 minute for QA session too. So our last speaker in this session is Forrest Rohwer from San Diego State University. His talk title is How Phages Create an Immune System, BAM Immunity, and Transcytosis.
Now? Okay, sorry about that. I have a horrible voice. Okay, so what I'm going to do, this is my favorite system that I actually study, which are coral reefs. And this is actually Millennium Island. And Millennium is about 7,000 feet of calcium carbonate sitting on the top of a mountain underneath the ocean that was built by the corals. And corals are pretty amazing because they're actually not only do they build the biggest structures, the biggest biological structures on the planet, but they're also effectively immortal, and they're some of the very first animals that ever evolved. So how do they do this? And in particular, how do they interact with their organisms? So this is where I normally work, but then I got dragged into the world of feces by this fecal matter fanatic here, who we all know. And as you know, he can sell you anything. So somehow he convinced me to look at fecal matter. And this was an older study where basically we're building off the twin study that you guys know about, where Jeff and that group showed that people that are related, in particular in families, have very similar bacteria sometimes. And what we did is we just did our typical prep where we can get the viruses away from everything else, and then we sequence them, and we just ask, one, do you have the same viruses as you travel through time if you're a person? And these are almost all phages. And two, how different are they per person? And what was really interesting about it is, one, we know that for the most part, the viruses you have today, you'll have later. So you're going to carry them through time. And the other thing that is really cool is that every individual actually has very different viral communities, and in particular phage. And that's true whether you're a person or if you're a coral. All right, now the other part of this story is the mucus. And what's interesting about mucus is that it creates these exchange surfaces that have to be renewed so that you can get rid of nutrients, or you can exchange nutrients or waste products, etc. And it's actually a glycoprotein that really is different between everyone. And that's because there's kind of a combinatorial chemistry thing that goes on to add the to add the sugar groups to the protein itself. There's all these great things about mucus, but just remember that it's essentially individualized. All right, and the starting point here for this study is that we've been looking at viruses or viral and microbial communities living on corals, and we'd go around and we'd suck off mucus, and then we'd just count the number of bacteria and the number of viruses, and we would compare that number against what we found in the water column. And then we started doing it on other surfaces because we got these weird bacteria to, or phage to bacteria ratios. And by weird, I mean that they were elevated. So we have about 40 phage for every microbial cell when we're looking at a mucosal surface. Okay? And that's very different than what we see in the water or the lumen, where you'll only have about 10 phage for every microbial cell. And you can imagine that since phage, for the most part, they're not actively hunting, so they're not able to use flagella or anything to move. Their interactions with the bacterial host is driven by mass action. So if you push up this number, you're more likely to run into a host. So this is interesting. This was our starting point. And so we started doing things like this, where you would take tissue culture cells with and without mucus. We'd add T4 phage. This becomes important later. And then we would wash it across these surfaces with and without mucus, and then we would put E. coli over the top of it, and then just count the number of plaques. And what you find is that wherever you have a mucus, here's just mucin itself actually, you would get the phage to hold on to the mucin or the mucus layer, and it actually protects underlying epithelial cells against the bacteria. So this gives you an antibacterial response by things being stuck in the mucus. Okay. And so this is what we started calling bacteriophage adherence to mucus, or BAM. And it's basically an acquired immune system, because what's going on is anytime that you pick up, anytime a microbial community comes in, it's going to bring its phage. The phage get stuck in the mucus, and they start killing the bacteria because they're at higher concentrations. The question is it specific or non-specific? Okay. And this is when we got really lucky. We got a great paper by Rick Bushman's lab, came out in 2012. And what Rick's group did is they took a whole bunch of fecal phage communities from humans, and they sequenced very deeply, and they found that there were these regions that were hypervariable in the viriome. And these hypervariable things we know a lot about, because the way they're made is that they actually use a reverse transcription mechanism to induce basically an alanine scan. And you guys have heard of different versions of them, because things like this are used in phage display systems and so forth. What's really cool about them is that they're one, hypervariable, and they maintain the original code, why they become hypervariable. They have all this massive diversity of about 10 to the 13th possibilities, which is close to what antibodies and TCRs do. And what was exciting about Rick's study is that these guys were actually Ig-like. Okay? So they look like an immunoglobulin domain that's hypervariable. Which is, of course, if you are an immunologist, gets you all excited. So here's the capsid of the T4 phage, and this is one of these things that's related to these, which is called a Hock domain, and they just stick out on the head. And so they have high avidity because there's a lot of them sticking out to this. So our working hypothesis was that phage are going to be adhering to the mucin through these hypervariable things, because the mucus is changing, so the phage needs some way to stick to it. And that's effectively what we found. So this is the same sort of study. You just have the phage plus or minus that Hock domain. And if you knock out the Hock domain, it doesn't stick to the mucus or the mucins. If you leave it there, it does stick. So what we get left with through this and a whole bunch of other stuff is that basically all these mucosal surfaces have these phage living in a particular place in them. Okay? And they're sticking to them and you're acquiring them with your microbiome. And they're specific because they're adapting to stick to your mucus, and they're also specific to the bacteria that they're killing. All right, I just put this in here to show you, like, as you guys, we wanted gaps. So one of the things that's really hard in the microbiome so far is that you guys don't really have very good dynamical models of the system. This gives you a feel of one approach that we use where we make these microfluidic systems, and then we have epithelial cells producing mucus. And then what we're going to do is we're going to run different concentrations of bacteria and phage across this. And then we just measure how many phage and how many bacteria we get, as well as cell survival. And then we put it into these models. And these models become really important because if you want to start controlling these systems, you have to have a model like this to work against, even if the model isn't right. And in this case, we're driving everything by this diffusion constant. It turns out that this is a very exciting parameter, but I'm not going to go through it. But if you take this, you actually get why this system works. And that's because it's an emergent property of just the dynamics itself. If you're a phage living in that mucus and you're holding on and you have bacteria coming in like this, you're actually much more likely to find a host, about 15 times more. So that's a very nice selection pressure. And it's bad for the microbes. So if you're trying to get in and you're running into basically this reef of phage, you actually get killed off at about 14 times more likely to die. So you can see that these guys are actually sitting there in the mucus, and they're effectively serving as a barrier to protect the epithelial layer underneath. All right, so what about inside the body? So we've seen all this stuff with the mucus, but there's lots of old observations that there's tons of phage in your blood, right? So we find them in blood whenever we do a variome or a blood variome. So that's been one of the newer questions we've been looking at, and this is how we've been doing it. So this is a transcytosis assay. And what you do is you put the cells, these are these kidney cells that will polarize when you put them in these chambers. And so you get an apical and basal side. And you put phage on one side and you see, did the phage come through? And in other cases, you put them on the basal side and you see if they go this direction. And what we always find is that the phage move from the apical to the basal side. Right? They don't go the other direction. And about 0.1% of the phage are functionally transcytosed. And by that, I mean that, so 1 out of 1,000 make it all the way through, and they're killing bacteria on the other side. And we know that about 1% of them are actually endocytosed, but of those, most of them die inside the cells. Right? It's actually fast, so we see close to saturation in about 30 minutes. And we know that it's relatively concentration independent. So it doesn't look like there's a specific receptor for it. It's something the cells are doing. And finally, it works with all phages, or all phage types that we've looked at. So these are just all the different phage, major phage groups, and we've just put them on the same sort of system, and we've just asked, are they getting through? And they do. And it's always in this apical to basal direction. So what does that mean? Well, if you just make a rough calculation, there's going to be about 30 billion phage transcytosis events per day in the average human. Okay? And that's roughly equivalent to how many white blood cells you have running around in your blood. So you've got as many phage running around there that can potentially kill bacteria. And our working hypothesis right now is that you're constantly sampling this, and you're getting them into your lymphatic tissue initially, and that this may serve as an acquired systemic immune system too. Remember, something like a coral that doesn't have anything that we would call an acquired system, this may be how they're doing it. The other thing that I think is important for as we move forward is that actually this is probably a tolerization thing. So there's this unusual thing about phage. So if I inject them into you, you'll develop a really massive immune response to them. But if you are getting them through your gut, you usually don't get an immune response. And it's probably because this transcytosis is taking them directly into the lymphatic system without a danger signal. So we are tolerized to our phage. All right, so here are the conclusions that we have these two different versions of this BAM model, which would be innate and specific. We think it's important for individualizing the microbiomes. We know that the tuning of these proteins that stick out on the phage can be used to put in particular places, which should be important for drug delivery and phage therapy. And we know that phage are also common throughout the body and may be serving as a systemic immune system that nobody's ever seen before. There's tons of people. I highlighted in yellow the people in my lab that have been working on this project. And of course, we have the math group and all of these people, including Gordon Lab and Bloomberg Lab, who have been great and very supportive of us doing some human stuff. These are the people that paid for various parts of it. And then I'm supposed to give you guys something, right? So you guys need to study phage, okay? You guys won't listen to me. I give these talks. There's always a token phage person invited to every microbiome meeting, and then everybody goes, those are so cool, and then you leave and you forget. You should actually really study them because, especially if you're young people, there are more of them than anything else out there. Most of the genetic diversity is actually encoded in the phage. We know that most of the unknowns, so like when you look at a viriome, it'll still be like, we get viriomes out of lungs, it'll be like 99% unknown genes, right? So these are just gigantic things that we know nothing about. When we do know the genes, it's usually something really cool like antibiotic resistance or a pathogenicity island or something else that's being moved around. In the human holobiont, as far as we can tell, every cell, your cells as well as your microbial cells, are lysogens. So they're all carrying temperate phages or viruses. And there's probably this new immune system. It isn't often you get a new immune system to study. And finally, this is a field that we're really suffering. So we had lots of phage biologists with lots of knowledge, and then they got pulled into other fields. Really, under Nixon was really the war on cancer. Brought all these guys into cell biology and stuff. And so we were really losing the large knowledge base right at this moment. Okay? So it's a good place to be if you're a young person. And I should mention Roger died, I guess, Monday. Okay.
Well, thank you. That was a fascinating topic. I think now we can start the Q&A session.
Hi, Rick Bushman, University of Pennsylvania. Thanks to all the speakers. I thought that was a really, really interesting session. I have a couple of technical questions. So Gautam, you're probably very well aware of the controversy about antibiotic resistance genes and their transportation around on phage. Traditional view has been there's not very much. Then there was a very prominent paper saying maybe antibiotic treatment causes resistance genes to appear on phage, and then they move around like crazy. And then there was a much less prominent paper published pretty recently saying the annotations were probably much too permissive in those studies, and probably lots of other studies, and that if you look really carefully and do it well, then you see way fewer antibiotic resistance genes. And so what advice would you have for us in this area? What's best practices in your view?
Yeah, and that's a great question. And I think it's not just antibiotic resistance, that just happens to be a selectable feature, but in the microbiome field, annotation creep is one of the things that really hampers, I think, our ability to make inference. And so for antibiotic resistance, we and others have tried to advocate for sort of a slightly better way of doing it in terms of taking protein evolution into account to build, for instance, profile HMM-based models. as opposed to your standard, most people annotate by blasting to a database, and honestly, the gene that you hit is probably 60% sequence identical. That was 60% identical to something else that someone did one biochemical assay in the 1970s, right? So I think there's an opportunity to use more sophisticated annotation. And then I think, again, we happen to be lucky with resistance that's highly selectable. I think we just need to do more biochemistry. We need additional assays on genes to prove they do what they do. So I completely agree with you. I think that's a very legit concern that many of these studies are fraught with over-annotation of function.
Okay, and then in a different field, Dr. Ghanem, at Penn we've done various profiles of microbiome from IBD subjects, and we've seen fungi that were sort of notable in a couple of studies, but they were different from yours. You were seeing Candida, one of the Candida species. We saw some examples of Candida, but different species, and then also some other species as well. And looking over the literature, there's starting to be a few of these papers now, and you're hard-pressed to find any two that are calling the same fungi in inflammatory bowel disease. Furthermore, there's the complication of the fungi may be much more commonly transients in food than we usually see in bacteria. So there's like the masking effect or danger of getting fooled. So what's your take on all of this? As far as I can tell, it's sort of up in the air as to which are the most important fungi, though a lot of people seem to be calling them.
Clearly, this is a very important observation. I mean, there is a couple of groups who are working on this area. David Underhill, which I mentioned, and then Harry Sokol in France. And usually, at least we agree, all of us, that Saccharomyces is there in the healthy people. They see Candida, Harry Sokol see Candida albicans. We saw tropicalis, as I mentioned. I think it does not mean that Candida albicans is not also present in these, because as you saw in my one of the slides, you can present, but it did not come out to the same level of significance as Candida tropicalis. The other thing is, I tried to point out that it's different control group. When you have the people who are not related and compared to those who are within the same family, having the same, live together, and their genetics are more closer, that's where we found the difference compared to the others. So the differences could be due to the different control groups that are being used. Now, the other thing is with respect to the other fungi, I agree with you that it could, we eat a lot of mushrooms and other cheese with, penicillium and this sort of thing. So some of it could be transient. Usually we try to look at core microbiome to try to eliminate these, and I showed this type of thing when we published on HIV-infected patients, where we looked at a fungi that should be present in at least 20% of at least the sample size, which controls it a little bit. Now, really the proof of the pudding is in the eating, as you say. We took the next step now, and we are starting to see that this is what happens, at least in animal models. So hopefully, if we have enough understand the mechanisms and this sort of thing, we can go back like from host to the lab, back to the host. But I think exactly, there's still a lot to be done with respect to mechanisms.
Marty Blaser. I want to ask Forrest, your talk was fascinating. And as you know, in the oceans, phage are involved in the kill the winner biology, where there's an advantage. It selects for a diverse population. That's an open ecosystem. How about in the coral or in the human, in those what we think are more closed ecosystems? Is with your diffusion constant, is that suggesting that we've got kill the winner behavior there too?
Yeah, no. I think it's what's more important in these systems are actually what we call piggyback the winner, which is the temperate phage, basically forming a mutualistic symbiosis with the bacteria, with the microbiome bacteria, both on the corals and in the gut. And they're bringing in functions of course, to enhance their hosts, right? Because they're so crammed together under those conditions. When you get lytic behavior, they can sweep through the whole system. So really the viruses and the phage are bringing in anti-other phage mechanisms, as well as things that protect them against things like protists in the coral system, which is probably where we get virulence factors from. That's my guess.
So in the coral, these are mostly temperate phage?
So on the coral surface, it's more temperate phage, and in the gut, it's essentially, it's also more temperate phage, and all of the cells are carrying prophage. It doesn't mean you definitely have lytic phage running through the system, but not as much as you get in the marine environment.
Brandon Eicher. This question is for Dr. Fowler again. I have a hygiene hypothesis question for you. So, there's phage everywhere, obviously. A lot of these are in source waters and things. We have a pretty interesting habit of sterilizing or at least treating much of the water that we consume in most Western cultures. Do you think that there is some contribution to our vanishing microbes or our dysregulation between host and microbe due to the fact of consuming this rather purified water and not getting a more interesting diversity of phage in our diet? Thank you.
No. I think we get quite, I mean, they're everywhere. I think we get a lot of them. Well, we know that they're in the breast milk, so we know that we're getting them at the beginning there. You get them, of course, off the skin of your mother and all of that. Yeah, we get a lot of phage. The interesting event is, I think it's just driven by what you guys already know, is that when the stomach actually really becomes acidified and closes off during the weaning time, those phage seem to get locked in to your gut at that point, and that may be why we have such individualized variomes, is because you're mostly stuck with those viruses.
So you believe that there's no immigration happening once the gut's closed off?
Yeah.
Hi, this is Lakshmi Goyal from Cell Host and Microbe. My question is for Forrest. So Forrest, if I'm understanding correctly, I think you're implying a little bit that the host mucosa actually recruits phages in a way. Have you looked at a MUC2 mutant mouse or any condition where the mucus is degraded and seen whether there's change in homeostasis of the phage? That's one question. Second is there an IgA response from the host to phages? So no. We have not done those experiments. That's all Jeff's fault. He's supposed to do everything with the mice. So we've done nothing along those lines, and I don't know of any experiments where people have looked. As far, what was the? I missed it. IgA. Oh, so no. For the most part, you don't get. So you can get a very strong immune response if you inject them, but when we are acquiring them from our blood, we actually did this study a long time ago because we had all these phage that we kept finding in blood, and we looked for antibodies to them in the blood, and we never found any, even though we had tons of phage in the blood samples.
I do have a question for the panel. Given the problem of antibiotic resistance, what is the viability of phage therapy in conjunction or instead of antibiotic therapy, and where we are in the field?
So phage therapy, in theory, could work. In practice, it needs a lot of science. Anybody that's actually ever tried to raise a phage in the lab knows that they're really finicky, and you have to get the right conditions when the bacteria are growing. There are many examples of where We have phage that we cannot grow in the lab, but we can grow in the mouse, or vice versa. We can grow them in the mouse, but not in in the lab, but not in the mouse. So you have lots of this going on, and that's been really, to make phage a really viable option for antibiotic-resistant treatment, we're going to have to invest in the same way that we have in drugs, to tell you the truth. And nobody is willing to spend that money so far.
I would also mention that there are a number of other alternative biologics that people are considering outside of phage in terms of being adjuvants to antibiotics. And so, we've heard of probiotics a little bit, and I think that's a big deliverable from the microbiome field, where if we recognize that there are niches that infectious agents want to occupy, you could potentially knock them out by either probiotics or symbiotics, including other prebiotic combinations. And then there's also just reversing the entire paradigm of how we go after infectious agents. We've used the warfare analogies this entire time, right? This idea of carpet bombing. Whereas the thing that we've learned from the microbiome is that it's probably a little bit more of crosstalk and dialogue between microbes. And if we adopt that, then perhaps we can use not things just like symbiotics, but perhaps more targeted therapies to, for instance, go after virulence factors, which will still eventually have resistance, but just won't have the same burden as antibiotics and so on.
I must agree that there's an alternative. For example, as you said, the probiotics. I think if we can think of ways to select the strains that are able to antagonize the bad bugs or the resistant organisms, that could be a good way to do it. And I think there is some progress in that direction.
Well, I think it's time to feed our gut microbiota. So I thank you for your presence, and thanks for the speaker.
Hi, my name is Cindy Davis, and I'm a program director in the Office of Dietary Supplements at NIH. And it's my pleasure to welcome you to this session focused on the intimate relationship between diet and the microbiome. The purpose of this session is to highlight current knowledge on the bidirectional influence of diet and dietary components on microbial and host metabolism, and how the metabolites influence host physiology, what is still not understood about diet-microbe interactions in human health and disease, and what is needed to make progress in this area. We will have 5 speakers. The first will be Justin Sonnenberg from Stanford University, who will talk about leaving limbo, stepping back to build a strong foundation and mechanism, and stepping forward to dietary interventions in humans. Andrew Patterson from Penn State University, who will talk about intestinal epithelial cell receptors as modulators of host microbiota communication. Gary Wu from the University of Pennsylvania, that'll talk about diet, the gut microbiome, and its metabolome, as a therapeutic probe in IBD. Nicole Koraptikin from the University of Michigan, who will talk about how gut bacteria eat your veggies, molecular details of glycan scavenging at the cell surface, and Johanna Lampe from the Fred Hutch Cancer Research Center, who will talk about testing diet-gut microbiome interactions, use of controlled feeding studies in humans. So it's my pleasure to introduce our first speaker, Justin Sonnenberg.
Okay, great. Thanks very much, Cindy. I want to start off just by thanking Lita and Cindy and all the other people associated with NIH and beyond that have put together this great workshop. I think it's been spectacular. There has been one thing that's kind of perplexed me about this, and that relates to the conversation yesterday about wearables. Mike Snyder, my colleague, mentioned that at the time of his talk, he was actually wearing 8 wearables. He went on to show all kinds of microbiome data, and nobody asked him where these wearables were. And we heard about the toilet of the future that can monitor the microbiome. I think we can do better than that. I think it may have to do with repurposing. Many of you may remember Google Glass. It was sidelined by Google. I think there's still a lot of potential here. Excuse me, with a slight tweak to both the design and the name. This is a real-time microbiota sampling device. this may not be the exact design, but it's coming, so enjoy your freedom and comfort now. Okay, on to the real talk. Critical gaps in the field. The title of my talk is Leaving Limbo. I think we all understand the potential of the microbiome, and I feel like the field right now is really grappling with how to translate this. And I worry that we've left the basic science a little too rapidly and a little bit too far behind. And so I think one step of limbo needs to be back towards basic mechanism. We need to do things like imaging, more genetic tools, use of other model systems like we've heard about in this meeting, and then funding specifically for non-disease related interactions in the gut. I think the step forward is to study humans more, particularly with dietary interventions. We know that manipulations of diet are safe by and large for humans, and so we can start doing experiments right away. We've seen this to great success in the field, both in published studies and some of the preliminary work that we saw presented yesterday. And so I think that we need to think about really embracing this approach and going wholeheartedly into developing humans as a model system for studying the microbiome and its importance in human biology. So my lab studies the dynamics of the gut microbiome in response to diet, and we particularly are enamored with the microbiota accessible carbohydrates, or MACs, the major fuel within dietary fiber that feed the microbiota. And the reason that we use this term is to distinguish the carbohydrates they utilize versus those that they don't utilize as they pass through the distal gut. Most of us eat a Western diet high in sugar, fat, starch, protein that gets absorbed in the small intestine, leaving our microbes starving. We've heard about microbes turning to eat us in the absence of sustenance from our diet. We know that they eat the mucus lining, also made of carbohydrates, if we're not feeding them, inciting inflammation, doing bad things. If we eat complex carbohydrates, that feeds our microbiota because we can't degrade these carbohydrates in our upper GI tract. In our microbiome, it's clear that it's one of its primary professions is degrading complex carbohydrates. And so one of the gaps that we've been trying to fill is one in imaging. And I think it's important to recognize how important spatial organization is in anything. And this is an image taken from an artist and comedian, Ursus Verley, who likes to organize things. In this case, he's organized the components of a fir branch. You can see the little branches and needles here. This is similar to performing a 16S analysis. And you can see how much more information is gained when you have the spatial structure. And so we've been working very hard to develop tools that will enable the field to move more rapidly towards studying the spatial organization of the microbiota. We've recently reported a software in cell-hosted microbe that will enable people to perform quantitative studies in imaging. We've also developed fluorescent Bacteroides species. This is an image of 6 different species of Bacteroides, each glowing with a different fluorescent signature, in this case embedded in a piece of plant cell wall material that's within the gut. And these tags allow us to differentiate these species and will hopefully help to propel understanding spatial organization. We're very interested in this idea that there could be an incompatibility between our human genome and our microbiome. The fact that we have these 2 components of our biology, our human genome and our microbiome, with asymmetric plasticity over evolutionary time. And it leads to the possibility that when pressed by selective forces, things like extreme dietary change, as we've moved to a very low fiber diet in the industrialized world, or introduction of antibiotics, that we may end up with a microbiome that actually is very different than the one that our human genome has come to expect, and the one that perhaps shaped our human genome during our evolution. And so it would be wonderful to have a time machine to go back and sample the microbiome in different humans in different parts of the planet. We can't get that application funded, and so what we're doing instead is studying traditional populations of humans. These are the Hadza of Tanzania. They're the last full-time hunter-gatherers in Africa. They spend pretty much all of their time hunting and gathering food that they eat each day. And they're a continuation of humanity's ancient past, a modern people that perhaps serve as the best modern approximation of what our microbiome looked like during 190,000 years of our species evolution, and perhaps longer. This is them sitting around a fire cooking a tuber. They have incredibly fibrous tubers, very different than the ones that we eat. They've been very gracious to participate in our research study, and we've been sampling them over time. And what we see is incredible patterns of seasonality, coincident with seasonality of their foods that they eat, great diversity in their gut microbiome, and then new taxa that we just don't see in the industrialized world. And so the wonderful former student who recently left, Sam Smits, who not only generated this data and analyzed it all, brought together as much data as he could from different traditional industrialized populations, microbiomes, and put all of that onto one PICO-A plot, and that's shown here. So each dot is an individual microbiome, and then he's compressed all of this data down onto the first principal coordinate and provided a bar plot of these different populations. And what you can see immediately is just this really stark pattern. All of the traditional populations are over here. All of the industrialized populations are over here. And so it doesn't matter whether you're in Papua New Guinea, South America, or Africa. If you live a traditional lifestyle, you have a microbiome that looks very similar to other traditional populations and very different from all of us. So it's not just a diversity thing. If we actually map abundance of taxa onto this PICO1 and look at the fractional representation across it, we see groups like spirochetes, Succinivibrio, Prevotella greatly enriched with the traditional populations. Alternatively, in the industrialized populations, what we see is a bloom of Verruca microbia, Akkermansia mucinifera, a mucin-loving bug. We can get the same bacteria to bloom in mouse experiments if we deprive those mice of MAX, or dietary fiber. So we're reconstituting a finding here on a global microbiome scale that we can see in mice just by depriving them of fiber. So what does this mean for us? The fact that we've lost species and gained some others, we likely shifted our microbiome very recently in our evolution. What does this mean for our biology? And how did we lose these microbes? So we think that diet probably played a big role. We know we've undergone a great loss of dietary fiber in the Western diet, and we wanted to test this in mice. And so we gave mice a human microbiota and gave them a high fiber or high MAC diet, and then split them into two groups. The low MAC group, we expected that the diversity would crash over the course of this 2-month period, and then asked the question, how many of the lost species could come back if we reintroduce dietary fiber? Is it too late for all of us, or if we started eating better, could we recover our microbiota? And so actually we did see the crash. We did see a lot of species come back actually unexpectedly. Not all of them, but a lot of them. And we thought, well, maybe this is a multi-generational effect. And so we repeated this experiment over the course of 4 generations. Erica Sonnenberg performed these experiments with Stephen Higginbottom in my lab. This is the high MAC diet control, microbiome data from the 200 most abundant species over 4 generations. So each row here is a different mouse at a different time point. Very stable species, more variable species, and some species down here actually do go extinct over the course of the 4 generations. But what happens on the low MAC diet? Huge decimation of the microbiota. So each generation we see this loss of diversity and a very interesting pattern. So we see actually at an early time point here microbes that we can't detect. We wean pups to the next generation, and then we put the parents back on a high fiber diet, and some of the microbes come back, but those are the ones that are not passed on to the next generation. And that happens at each generation. So you press microbes to low levels, they have a lower probability of being passed on to the next generation. So elimination of dietary fiber depletes the microbiome. I want to return to the Hadza data now. One of the interesting things that we saw was this seasonal variation. We saw the cyclic pattern of microbiome configuration in the Hadza. So each season, as foods would disappear, we see some microbes disappear. And then the following season, when the foods reappear, those microbes reappear. And so this is data from the 8 individuals that we surveyed over the 3 seasons. So this is dry, wet, dry season in the 2013 and 14. And what we see is Prevotella, spirochetes, Succinivibrio species disappear and then reappear the following season. That doesn't happen for these seasonally stable species. And what's notable to me is how similar this pattern is to the mouse experiment that we performed. What would happen if we were to lock the Hadza in the wet season diet for multiple generations? Would those species gradually disappear? And what's also notable is that it's the seasonally volatile species that are exactly the same ones that correspond to those that have been lost in all of us. So could this be a diet-driven effect? So this is our hypothesis that diet has depleted our microbiota. Is this meaningful for our biology? We don't know. Should we recover these species? Will they be helpful? Can we do this just with diet? Do we have to deliberately reintroduce them? These are all things that we want to figure out and study over the coming years. We do know that our microbial identity has changed. We also know that Western diseases are on the rise. Could these be linked? We know that the microbes in our gut control our immune system, and we know that most Western diseases are driven by chronic low levels of inflammation. Could it be that we're all sitting in this room with simmering inflammation that's driving us towards one of these diseases because we have the wrong gut microbes? That's the hypothesis. So we want to test this, and we've actually started the Center for Human Microbiome Studies at Stanford to start doing dietary interventions and starting to look at some of these questions. And so the idea is fairly simple. Enroll a cohort of people, perform an intervention, and then look at who responds to whatever you're looking at. Perform omics data for the immune system, for the microbiome over the course of the experiment, and then identify the predictive elements that tell you which individuals at baseline are likely to respond. Use machine learning to identify the predictive elements. You do this again and again. And you end up not only with precision interventions for these individuals to deal with whatever they're dealing with, chronic inflammation, obesity, autoimmune diseases, but what you also get is a roadmap for what mechanisms we should be studying. And so we have traditionally thought about mechanism to animal models to humans as a translational path. I think we have to start thinking about humans as the first step just because of the sheer complexity of the microbiome. Why not let studies in humans guide the priority list for what mechanisms out of this infinite list that we should be studying? This is an approach that definitely works. There are great published studies. We've seen more studies presented here where it's clear longitudinal studies in humans, if we're allowed to collect a lot of omics data, are a really effective way to hone in on important biology in the gut microbiome. And so we're starting to do this. We ran a pilot intervention over the past year. This is a high-fiber, high-fermented foods, 2 arms. We enrolled somewhere in the order of between 40 and 80 people. High fiber, we got some people up between 60 and 80 grams of fiber from plant-based foods over the course of this 10-week intervention. People eating 6 to 8 helpings of fermented foods, trying to load them up with foodborne microbes, probiotic-like microbes. And over the course of this, we're performing microbiota profiling at the time points. We're performing immune profiling using things like CyTOF, 370 different immune parameters to see if we can move the inflammatory status of the immune system. And then also performing clinical, behavioral, and cognitive tests along the way. And this is a collaboration with Christopher Gardner, whose group performs the dietary interventions, and we're collaborating also with Mike Snyder, Mark Davis, Kari Nadeau, and several others at Stanford to look at different cohorts and use different technologies to monitor these cohorts longitudinally. And so I just wanted to finish with a slide something that I think we should all think about. I think many of us in the biomedical endeavor think about solving mechanisms, trying to create drugs to treat diseases that exist. And I think something that we should spend more time on is thinking how to get people to eat differently to prevent diseases that are completely preventable. And I think this is doable. I think it's going to be difficult. This is shown by Hadza Food Preferences. The Hadza eat a huge amount of fiber, but it's not because they want to. It's because the plants are abundant in what they can get every day. If you ask them what they prefer eating, it's meat and honey. Those are the 2 foods that they would eat all the time if they could. If you put them in a restaurant or a convenience store, they make the same bad decisions that we make. And so how do you get, and this is a beautiful picture here that shows a Hadza man holding a piece of honeycomb. He, had to get stung, if you look closely here, to get that honeycomb. So there was a cost to getting this sweet substance. They can't get meat every day. So it's a very different ecosystem that they live in. And the question arises, how do we get people to eat healthy in a free and affluent society? Policy, education, food technology, addressing the Farm Bill, things like this. I think these are things that our data should feed into and should be cross-fertilized by NIH. And finally, I just want to acknowledge the people that did the work. Sam Smits and Erica Sonnenberg did the ancestral microbiome work. Liz Stanley and Wes Whitaker did the synthetic biology and glowing bacteria. Who else here? Kristen Earle did the imaging, and Stephen Higginbottom does a fantastic job of running our mouse facility. We have wonderful collaborators that helped with the, it really drove the Hadza project. Of course, thanks funding NIH, Burroughs Wellcome, and Stanford. And then just to mention, my wife and I wrote a book about the microbiome. If you or friends or relatives are interested in learning more, thanks for your attention.
Our next speaker will be Andrew Patterson, who will talk about intestinal epithelial cell receptors as modulators of host microbiota communication.
Thank you. Okay. So yeah, I'd also like to thank Sydney for organizing, and also to the organizers of the general meeting. I've really had a great time and got to meet some interesting people and get to see some very interesting science. So today I want to talk about a little bit slightly different topic, a little bit more mechanistic understanding of how the microbes perhaps interact with the host. In this case, by understanding the small molecule exchange that occurs in the various systems. So before we try to start thinking of the future, I think it's always good to step back and think a little bit about the past. And this is a quote taken from a pharmacologist from about 50 years ago, where they began to appreciate the understanding, or had a good understanding, that the microbes could themselves contribute to potency or efficacy of drugs. But at the same time, they did not have a good understanding or a way to measure or quantify these microbes and what they were doing to manipulate these effects. So going forward, I think we have developed a lot of really great tools that have given us this opportunity to do this in a much more mechanistic scale. So we've seen lots of excellent examples of sequencing-based approaches, excellent model systems, but I think one thing I really want to emphasize, and I think a place where metabolomics is really going to have a strong foothold, is here in the microbiome community, and that is trying to understand the products or the metabolism of these microbes and put this into more of a broader view of how this contributes to health and disease or some other metabolic phenotype. And of course, this is not made possible without really significant bioinformatics capabilities. And it's a really impressive display I've seen over the last few days about how important this is in driving this field. And I think it's even more important to convince people to move away from TAGC and start focusing on carbon, hydrogen, nitrogen, oxygen, and phosphorus. So anybody out there willing to do that, I think it's a ripe opportunity. But with all this information, it's really giving us a good opportunity to start to understand this chemical dialogue that exists between the host and the microbiota. So this is a signaling mechanism that allows the host to transmit signals to the microbes and vice versa. And I'll give you a few examples of this today. So when I started thinking about how to design this talk or what I would talk about, it's something that I think is really close to my own postdoctoral training, and that is really understanding this signaling pathway that occurs between the liver and the intestine. And so we can think about it in terms of the exchange of nutrients, drugs, environmental chemicals, of course. But importantly, we also see a lot of examples where bacterial metabolites are going to signal back and forth within the portal circulation. And within these organ systems, we have a network of sensors or receptors that are there to sense that gut environment and signal these changes to the liver. So I broadly classified them as metabolic sensors. You might recognize the top ones as being nuclear receptors. But broadly speaking, we see them being enriched in the liver and then in the GI, and they're very important there for mediating these signals back and forth between these organs. And interestingly, over the past several years, we began to appreciate that a lot of the ligands that are generated of microbial origin, so they could be generated specifically by microbes or themselves could be metabolized by microbes, can really modify the activity of these receptors. And it's becoming more apparent that these are really clearly there to sense and signal these metabolic changes from changes in the microbes. So I just want to focus on two. There's not a whole lot of time to, but I'll talk about the farnesoid X receptor and then end with a little story about the aryl hydrocarbon receptor. So with FXR, it's really a great example of the impact of the microbes with enterohepatic circulation. So this is a process of the exchange of material between the liver and the gut and back and forth. When we think about bile acid metabolism, bile acids are produced in the liver from the oxidation of cholesterol, secreted after a meal to help solubilize or mobilize dietary compounds. But I think we're moving beyond this appreciation that these are just soap or these are detergents, but in fact, these bile acids perhaps are serving more as important signaling molecules that allow the host and the microbes to interact or to signal back and forth to each other. A lot of data, from several decades ago really understood that this process was dramatically influenced by gut microbial processes, and it's really started to become more apparent once we've had the development of mouse models that have allowed us to investigate this a little bit more carefully. And so with FXR, we've been able to appreciate this change that occurs in bile acid metabolism using a variety of mouse models that we can manipulate FXR as well as manipulate the microbiome itself. And so I just show one example here of how bile acids themselves are transformed by bacterial processes. In this case, I just show an example of bile salt hydrolase activity of lactobacillus. This could be the same for Clostridio, bifidobacteria, a variety of other species as well. This is a very common process. It occurs on taurine in rodent models as well as in glycine, so more common to humans, bile acids. And remarkably, despite all we know about bile salt hydrolase, we don't really know necessarily what it does or what benefit it provides to the microbes themselves. It could perhaps provide tolerance to bile acid pools that are secreted. It could help the microbes scavenge nutrients, in the case of, say, taurine as a sulfur source. Or furthermore, it probably more than likely helps them maintain some GI persistence. Those that don't have significant bile salt hydrolase activity don't really have very good GI persistence. And so when we began to look at this a little more carefully, we did this study with either antibiotics. In this case, I show an example of a free radical scavenger, Tempol. And what we found is that there was a dramatic change in bile acid pools with these compounds that we treated with. And we saw a major shift from unconjugated bile acids to those that are more conjugated. And this began to give us this idea that perhaps a lot of these drugs that we were thinking as relatively innocuous were in fact manipulating the microbiome, as in the case of what we found with Tempol. And so what we saw is that, of course, as the BSH activity dropped, we saw an increase in T-beta MCA levels or other taurine-conjugated bile acids. And what this really had a tremendous impact on was FXR activity. And so this was demonstrated by our lab along with Frank Gonzalez, and as well as really elegantly demonstrated by Friedrich Backhead's group as well. And that these compounds, these bile acids, typically can serve as agonists of the receptor. So shown on the left, I just show a reporter assay showing increased FXR activity with the bile acid TCA. But you note that when we treat with T-beta MCA, the FXR activity goes down, suggesting that indeed this is probably some way manipulating FXR activity. And if we look at FXR target gene activation in the intestine, we see that indeed FXR activity is reduced. It's not dramatically knocked down to zero, but we do see about a 50% reduction in activity. And so we've gone on to demonstrate that T-beta MCA, as well as a variety of other bile acids themselves, serve as really potent FXR antagonists. And so this began to help us appreciate this really dramatic effect that's occurring with this massive pool of bile acids that are mixing around in our guts. And if there are ways of manipulating this bile acid pool one way or another, that perhaps we could use this as a tool for, or a means to modulate FXR activity in vivo. And so this just summarizes a lot of work into one simple flow diagram. But we found that with bile acid metabolites, they dramatically influence intestinal FXR signaling. And remarkably, what this showed to us was that the effect on the intestine was very profoundly observed in the liver as well. And so we found this was really beneficial for modulating fatty liver disease, so NAFLD as well as NASH. We found observations related to insulin resistance and obesity. And remarkably, a lot of this appeared to be driven through ceramide signaling, which I won't have time to go over today. So I want to switch over in the last 5 minutes or so to a different receptor. So this is not a typical nuclear receptor. In this case, this is a postomine protein, the aryl hydrocarbon receptor, typically or a long time identified as being the dioxin receptor. You probably have heard of it, to bind TCDD, as well as a variety of other environmental compounds, dibenzofurans, benzoate pyrene, and others. But over the last decade or so, we began to appreciate a bigger role for AH receptor, and that it serves as a really great dietary sensor. So it binds to flavonoids or indoles. It also more recently has been shown to be a very important gut microbial sensor. So it binds to products of bacteria themselves, in this case phenazines. It binds to endogenous metabolites that are manipulated by the host or by the bacteria, in this case tryptophan metabolites, as well as other cholesterol metabolites. And again, we started to think that this balance of AH receptor activity could be modulated by, in fact, by things that we consume in our diet, as well as things that we may be exposed to in terms of environmental contaminants. And so I just want to run through one very brief summary of how tryptophan gets catabolized and the importance of this for AH receptor activity. If we start with tryptophan here, we see that a variety of bacterial species are able to import through a variety of permeases. Tryptophan gets catabolized to a variety of things, including indole-3 acetic acid. And then a variety of decarboxylases can also produce things like skatol and tryptamine. And it's important to note that this is not entirely always mediated by bacteria. So in the case of indole, we see a lot of this being produced by bacterial properties. But these indoles can also be further metabolized by the host, in this case by Sip2E1 or sulfur transferases to produce things like indoxyl sulfate. So you may know these as typical kidney toxins. But the interesting thing about this is if we actually go and then investigate their ability to activate or to bind to the AH receptor, we can see that they really have profound and important properties for doing so. So in this case, I'm just showing a very simple reporter assay where we're looking at the ability of compounds to activate the receptor and thereby activate this reporter system. So using the prototypical ligand TCDD or dioxin, we see a rather dramatic increase in AH receptor activity, okay, shown in the green bar. And then we can see that as we try a variety of different tryptophan catabolites, we see that in some cases they can bind or they can activate the receptor just as well as TCDD, often thought to be one of the most potent ligands for AH receptor. So this goes to show, or goes to emphasize, the important properties of the microbiota in metabolizing these compounds to activate the AH receptor. And so one thing is actually quite interesting when we start to look at AH receptor activity in terms of its ability to be modulated not necessarily by dietary factors, but also by environmental compounds. This is a really strong contribution by the gut microbiota to modulate its basal activity. And so what I'm showing here is just a readout of AH receptor activity looking at its target gene expression, in this case in the ileum, of mice treated with an environmental contaminant. So CN stands for conventional mice, or we have germ-free mice, as well as AH receptor knockout mice, which we expect not to respond. And so what we can see with treatment of TCDD, either in the blue or the red bars, we see an increase in AH receptor activity. But what I want to point out, or I hope that I can emphasize to you, is that the basal activity of AH receptor is dramatically lowered in germ-free mice, again supporting this notion that the microbes themselves are able to produce important AH receptor ligands. And so we can actually even see this as a readout in the metabolic response of various organ systems. So here I'm just showing the change in the metabolome. In this case, this is just a pseudo-NMR spectra where metabolites that are increased are pointing towards the treatment group, or those that are decreased point away. So in the case of TCDF-treated mice, we see increase in lipid accumulation in the liver, very typical of what you see with AH receptor or high AH receptor activity. But what I want to point out to you is that when we compare these changes between conventional and germ-free mice, we see a dramatically different response. Again, supporting this idea that the basal AH receptor activity is really important for, or is importantly modulated by gut microbial processes. And so in preparing this talk, I really tried to think hard about what were some of the challenges or gaps that are actually facing us in moving this type of work forward. And that is really truly trying to understand what are these metabolic products, or what are these metabolites of the microbiome itself that are actually serving as signaling intermediates between the host and the microbe. And so there was a lot of really excellent talks yesterday where metabolomics has been helpful in trying to really capture or catalog the true the full repertoire of metabolites that are produced or modified by the microbes. And I think just to add one important addition to that, and that is to really try to understand these in a functional context. And by that I mean, can we actually show that these indeed are signaling through or binding to these receptors, and in fact they're actually providing a means for these signals to be transmitted to the host. And so in closing, if we just take a look at what a typical view of the gut microbiome, in this case it's just a human stool sample that we've profiled by mass spectrometry, and then using molecular networking through GMPS library searching. We saw really great examples of that yesterday by Peter Dorstein. We can see that there's a tremendous array of features that we're able to identify, and we can do pretty good at trying to figure out what they are. We have a general idea of the bile acids, lipids, aromatic compounds, as well as some drugs and environmental chemicals that these individuals perhaps were on. But you'll note that this is not fully annotated, and there's a variety of compounds that we're yet to identify. And in fact, this is a great example of where the field of metabolomics is today, and that is trying to further the development or the identification of these metabolites, such that we then can do functional studies on them. And so the development of a lot of these databases are helping to get us to this point. GMPS, for example, but MassBank, HMDB, PubChem, and Metlin are all really excellent resources that are helping to get us towards this elucidation or the illumination of the chemical dark matter. So with that, I'd just like to thank collaborators both at my university as well as the National Cancer Institute. So Li-Min Zhang, a really talented postdoc, as well as my longtime collaborator, Gary Perdue, and also a really excellent collaboration with the Gonzalez group here at NCI as well. Thank you very much.
So the next speaker will be Gary Wu, who will talk about diet, the gut microbiome, and its metabolome as a therapeutic probe in IBD.
Great, thank you so much. Can you hear me now? There we go. All right. Thank you so much. First, I want to thank Cindy and Lita and the rest of the organizing committee for inviting me to give a presentation here. This has really been a fabulous meeting. So I'm going to start off with my challenges and gap slide here. So there are challenges in characterizing the effects of diet on the human gut microbiome. Humans are poorly adherent to dietary regimens. Current tools to characterize dietary composition and intake are imperfect. The reciprocal nature of dietary composition to maintain isocaloric consumption make it difficult to determine factor responsible for observed outcome. Diet can have a profound impacts on the host biology that are independent of the gut microbiota. And both intensive controlled feeding experiments and large outpatient cohort studies are expensive and challenging to complete. So it's for that reason there's enormous utility to study animal models when thinking about the interaction between the diet and the gut microbiome for the following reasons. You can tightly control defined diets over long periods of time. You can do multiple biological replicates. Germ-free animals can be used to examine the effect of diet independent of the gut microbiome. Defined microbial consortia or complete gut microbiota studies can be performed in notobotic mice. And most importantly, you can determine cause and effect relationships involving diet and the gut microbiota. However, there is a challenge because mice and other model organisms are not totally equivalent with humans. So I show you on this slide the very first study that actually Rick Bushman and I did many, many years ago, where we were studying the impact of diet and genotype on the gut microbiome. And so we had a wild type and I had a knockout mice. And what you can actually see here is on a high-fat diet, there was this massive consistent effect of diet on the composition of the gut microbiota with a bloom of firmicutes and proteobacteria. So very, very significant effect. We then published a paper a couple of years later in a controlled feeding experiments on healthy human volunteers where we randomized them to either high-fat or low-fat diet. This was a 10-day inpatient stay with same meals each day, and we collected stool samples every day. So each individual is a different color. Each ball is a different day of the study, and for each color, there's always one ball that's different from all the other balls, and that ball that's different from all the other balls of the same color is day one of the study. So what that means is that within 24 hours, the human gut microbiota does change in composition in response to diet, with a couple of other really important points on this slide. First of all, you can see how all the colors are very different from each other. This is this intersubject variability, which is the largest source of variance in most human studies, and that's why humans are exceptionally noisy when thinking about the human gut microbiota. So the effect within 24 hours is highly statistically significant, but it's very, very modest relative to intersubject variability. The other point is that even though these individuals were on the same diet, half on one diet, half on another diet, they didn't converge over a period of 10 days. And then the movement from day one to all the other days is seemingly stochastic. It wasn't consistent in the way that we actually observed in mice. Now, maybe in a question and answer period, there actually is biological relevance and information in what seems to be noise here, but we can discuss that later on in a question and answer period. So the bottom line is it appears that the human gut microbiota, in terms of composition, may be much more resilient to the environmental stress of a change in diet than is a mouse. And we actually also observed this in another study that we had published on looking at the composition of the gut microbiota metabolome in vegans and omnivores. So you can imagine a vegan and an omnivore have very, very different diets, and the plasma metabolome is very, very different. Yet the composition of the gut microbiota, either by the weighted analysis or an unweighted analysis, is very, very similar between the two. Again, not necessarily replicating what we actually see in mice. So we're in a stage, I personally believe, where we have a lot of wonderful animal model data showing functional effects of the gut microbiome. And we have a lot of human association studies. And one of our goals is to get down here, think about novel therapeutics and diagnostics. And what we're using right now is simply stool for the treatment of Clostridial difficile infection. So this really is sort of the evolution of the microbiome field to get from here down to here. And we feel, just as Justin pointed out in his talk, that actually studying humans is actually really, really important in trying to get down this axis. To understand the biology of humans and the impact of various types of interventions on the gut microbiota in humans, and also to determine if what we actually see and observe in animal models is actually relevant in human biology. So we began as a group at Penn in the microbiome field studying inflammatory bowel disease. And the current treatments for inflammatory bowel disease largely focus on deep immunosuppression of the host. So if you ask the question, how much of inflammatory bowel disease is really due to the host, in other words, what's the genetic contribution to development of inflammatory bowel disease, is actually relatively modest. So a lot of inflammatory bowel disease in terms of the risk is actually inferred by the environment. And just one example of this is the increasing incidence of inflammatory bowel disease worldwide associated with industrialization. We also see this with many other types of inflammatory mediated disease processes. This began in North America and Europe and in Israel, and then about a decade after that we began to see a rise in Japan, and now we're beginning to see a very rapid rise associated again with industrialization of the incidence of inflammatory bowel disease in China and in India. So we think that that's an enormous opportunity. Think about how you might be able to engineer the environment of the gut, maybe through diet or change of the microbiota, that might actually make these types of interventions work better, or they could be a standalone modality for treatment. So if we are to think about using diet, antibiotics, microbial inoculation to engineer the environment of the gut to treat inflammatory bowel disease, there are a lot of really important questions that we need to address. What's the optimal method of administration? Can these modalities combine with immunosuppression to optimize therapy? What are the optimal clinical settings in which these interventions can be used? And if you do a human subject study, you should be collecting a lot of biospecimens to actually see how the human host actually responds to your microbial or dietary intervention. So there is actually a dietary intervention that is well documented to be effective in the treatment of Crohn's disease, and this is a defined formula diet. In its most simple terms, it's actually an elemental diet: amino acids, triglycerides, and simple carbohydrates. And for that reason, many of these are not palatable, so they have to be delivered by a nasogastric tube. Despite their high level of efficacy, we really don't understand how these diets, that particular diet actually works. So our paradigm is here. We know that diets are epidemiologically associated with inflammatory bowel disease, and we know that gut microbiota plays a fundamental role in pathogenesis of IBD. And we also know, and we've heard a lot about the impact of diet on the composition of the microbiota and as well as its small molecules that it makes. So our strategy was to use a diet that we already know works in inflammatory bowel disease as a therapeutic probe to understand the mechanisms behind this. Because if we could understand the proof of concept mechanisms, we could define better diets for patients with inflammatory bowel disease. So I'm a physician scientist, so I'm a big believer in astute observations in the clinical domain. It's important to note that fine formula diets for the treatment of Crohn's disease are the most effective when they're consumed completely in place of a normal diet. That's why we call it EEN, or exclusive enteral nutrition. Just to let you know how little we actually understand of how these diets work, there are actually two fundamental questions that are implicit in this clinical observation. Does EEN provide something good for patients with IBD that's not abundant in a regular diet, or does the consumption of EEN exclude something that is bad for patients with IBD in the regular diet? Well, we're beginning to see a body of literature arise that suggests that things that have been added to our food supply over the several decades, past decades, can change the composition of the microbiome in mice and lead to an inflammatory response. Artificial sweeteners, dietary emulsifiers. Now, I'm not saying that artificial sweeteners and dietary emulsifiers cause inflammatory bowel disease. What I am saying is that our diets have actually changed over the past several decades, and it may be one of those many environmental factors that may be playing a role in increasing incidence of various types of immune-mediated diseases like inflammatory bowel disease. Keep in mind, however, these are mouse studies. So we don't really know whether or not these observations are relevant in human biology. So we're working together, Jim Lewis and myself at Penn are working together with Andrew Gewirtz, and he talked about this dietary emulsifier work, in a controlled feeding experiment in humans to really determine whether any of the phenotypes that Andrew observes in mice and or other types of biomarkers can be reproduced in human biology, which again I think is fundamentally important in thinking about the relevance of these different types of observations. So in thinking about the impact of elemental diets on the composition of the gut microbiota, our first step was this study, and we called it PLEASE, Pediatric Longitudinal Study of Elemental Diet and Stool Microbiome Composition. These were 90 pediatric Crohn's disease patients that were either treated with elemental diet or anti-TNF therapy. This is a longitudinal prospective cohort study. We're a big fan of doing these prospective longitudinal studies to try to reduce and identify signal amongst a very noisy background of intersubject variability. This is previously published information, so I'm not going to really show any of the data. All I'm going to say is that we actually learned a lot from doing this study. It turns out that dysbiosis is not simply one thing. It's a compilation of many things. It's dysbiosis induced by inflammation, use of antibiotics, alterations by diet, and whether or not you just simply have a diagnosis of IBD or not. In compilation, that gives you that signature of dysbiosis. And we do know that elemental diets did have a particular microbiome signature, and one of the things we actually observed was that there was a decrease in fungus. What I do want to show you is some unpublished data integrating taxonomy, the structure of the microbiota, that we determined by shotgun metagenomic sequencing and metabolite production. So we think that the intersection, looking at not only the taxonomy but the small molecules that they make, will tell us a little bit more about function. And we think that diet is a really good example of this because diet, as I mentioned, alters the composition of the microbiota, but it also serves as a substrate for the microbiome to produce a lot of small molecules that we actually end up absorbing. So the study that I'll tell you about is called Food and Resulting Microbial Metabolites, or FARM. The objective of this was to determine the relation between dietary composition, gut microbiome composition, and the metabolic products that are ultimately present in the gut lumen and the plasma of humans. So this again was a 2-week, very intensive inpatient controlled feeding experiment in vegans, omnivores randomized to consume exclusive enteral nutrition. The product that we used for this was Modulin. So these are healthy human subjects. We looked at the impact of these different diets on the composition of the microbiome. And then we did a very intensive intervention to try to map the metabolites that are in plasma and the feces coming through the microbiome. We've done something similar in Crohn's disease in our PLEASE study, and we think that the intersection between these very two large data sets will give us some indication by which the diet alters the composition of the gut microbiome, its metabolome and the host metabolome, that reduces disease in patients with inflammatory bowel disease. So this study is part of the Crohn's and Colitis Foundation's Microbiome Initiative. That initiative was actually designed to identify new therapeutic targets. Our contribution to this consortium was actually to use diet as that therapeutic probe to find new therapeutic targets. So let me show you about 2 slides about what this study was all about. So there were 3 phases in this study. The first phase was actually looking at the impact of diet. And then halfway through, we did a very deep intervention to reduce microbial load using antibiotics and a gut purge called polyethylene glycol. And then we followed the reconstitution of the microbiome at the end here. Our rationale was that looking at from this time point to this time point would give us some idea of diet-induced changes in metabolites in the microbiome. If we were to compare this time point to this time point, the differences would identify metabolites produced or consumed by microbes, and as the microbiota actually reconstitutes, we can correlate increase or decrease in metabolites with the recurrence of microbes and diet to identify key drivers. So just to show you that the intervention was actually very, very robust, I showed you here the shotgun metagenomic reads of each day of all the individuals in this study. And all the green are microbial reads, and all the blue are human reads. And you can see you start out, and almost all the reads in stool are microbial. But with the intervention, we largely extinguished most of the microbial reads, and they're largely human reads. But then the human reads disappear, and you begin to see microbial reads as the microbiota reconstitutes itself. We did CFU counting and 16S copy number PCR, and so we estimate that we reduced bacterial load in humans by about 5 logs, about 100,000-fold. So this really was a very meaningful intervention. So what do we see in terms of Shannon diversity? Well, on a Western diet, you can see Shannon diversity is disrupted and decreases with the intervention, and then it recovers over the next couple of days. On a vegan diet, you also see that decrease in Shannon diversity, but it doesn't decrease to the same degree and actually recovers to a higher level. So that really means that the vegan's microbiota is actually more resilient to this very intensive intervention. And then on a modulant diet, you can see the significant decrease in Shannon diversity, but it actually does not recover. So what actually happened to the composition of the microbiome? Well, each diet is a different symbol. Each day of the study is actually color-coded over here. And let me just walk you through this diagram here. So pre-intervention, before we use the antibiotics and PEG, everybody is over here. Now, all the vegans and omnivores are clustered together here. So this basically substantiates what we had previously seen by 16S sequencing, that the vegan and omnivore microbiome are actually very similar to each other. But the modulant microbiome actually switched and changed after 5 days from up here down to here. So modulant has an effect on the composition of the human microbiome. And then upon reconstitution, the vegans and omnivores are clustered again together, and they're switching over here. But now modulant, that microbiome is very distinctive and now completely separate. So we have now engineered the microbiota into a different configuration. Now, along with the shotgun metagenomics, we've done a lot of metabolomics in plasma and feces, and this was done by Clary Klisch and Ramnik Xavier, and John Braun helped support this. I don't have time to really show you any of that data, but let me just show you this final slide here about the concept of engineering the intestinal environment for treatment of inflammatory bowel disease. Just like any other chronic disease, we believe that there probably will need to be an induction therapy and a maintenance therapy phase. So what we're doing is taking our most refractory patients with inflammatory bowel disease that do not respond to any types of pharmacologic immunosuppressive agents. And we have a study that we're recruiting for right now called Holiday. We're using antimicrobials in a different fashion to really try to get these individuals into remission. And we're cautiously optimistic that we're going to be seeing some pretty interesting things. Now, if we see a subset of individuals that actually significantly respond to this intervention, there are two possible outcomes. One is that they're going to be better for a long period of time because we somehow reset something. The other possibility is that the disease comes back when you stop the antimicrobials. And that's a notion that you're going to have something to maintain remission. So maybe you could engineer the microbiota in some different configuration to maintain remission. And Josini in our laboratory has a paper thinking about bacterial nitrogen flux as one way of doing this. I just showed you on the previous slide diet being able to do this. The other notion might be diet as a maintenance therapy. Maybe it's a combination of a defined formula diet with safe whole foods to eat. But the problem is we don't know what those safe whole foods are. So we're taking a very systematic approach. I talked about the farm study. We have two other large human subject intervention studies ongoing to actually define what those safe whole foods might be. So I just want to acknowledge I represent a very large team. We began with Rick Bushman, Jim Lewis, and myself, but with support from the HMP, actually our group has actually grown to very large size, and I want to acknowledge our really tremendous collaborators in many different types of projects. Thank you very much.
Our next speaker will be Nicole Kuropatkin, who will talk about how gut bacteria eat your veggies, molecular details of glycan scavenging at the cell surface.
And just as soon as I get this up, we'll begin. So first, I just want to say thank you so much for the invitation to speak here today. Let me explain myself first. I'm a structural biologist, and so I really think about the details of how proteins and carbohydrates interact. And I'm interested in using that information to understand how the gut microbiota specifically responds to diet. And so the way I've structured my talk actually is to spend less time telling you about the specific details of my work, but hopefully trying to impress upon you why we need to return a little bit more to understanding some of the basic biochemistry of these organisms that live within us, and how this information, by taking a detailed structural and biochemical approach and understanding how gut bacteria perform certain metabolic processes. I'm going to be telling you about carbohydrate degradation, but you can kind of take some of the examples I'll give you and extrapolate that to almost any metabolic process. Why it's important that we understand these details and how these can help better inform our various data omics sets and maybe allow us to predict the way gut microbiomes are going to respond to different types of environmental perturbations. So of course, as Justin and others have mentioned, the gut microbiota responds to diet in very different ways. And these organisms are extremely prolific in their capacity to degrade different types of carbohydrate nutrition. And they have many different categories of carbohydrates to choose from. Some can degrade different dietary and plant cell wall polysaccharides, other types of carbohydrates that we would find in our diet. And then some can turn and degrade host mucosal glycans, some of that protective lining of the intestinal tract that keeps us healthy. So a lot of different carbohydrates to choose from. And this means that bacteria actually have quite a difficult task at hand when it comes to recognizing these discrete glycan structures and deciding which ones they're going to eat. So how are they able to select and scavenge and process these different carbohydrates? And the reason they can is because their genomes are full of carbohydrate active enzymes, which I'll refer to as K-zymes. Now, as humans, we can't digest very many different carbohydrates. We can't just digest most of the stuff up here. We can really only digest 3 carbohydrates: sucrose, lactose, and starch. But these guys can really degrade just about everything else. A single gut bacterium can have hundreds of enzymes for the degradation of carbohydrates in its environment. So it's been used as a proxy to look at basically the genomic K-zyme content of a bacterium or a metagenomic data set in order to try to understand, well, what kind of carbohydrates is this community capable of utilizing? But there are a lot of caveats in relying strictly on some of these annotations, even very well-established databases like the Carbohydrate Active Enzymes database, which is very well curated. For example, and I'm going to take you through a list of different examples as to why we need to delve deeper and beyond annotations in order to understand function. So for example, in the glycoside hydrolase family 5 group of enzymes, this was typically thought of a group of enzymes that can only degrade cellulose. But as you can see from the list up here, enzymes within this family can degrade a wide variety of different carbohydrates. So you have protein sequences and protein structures that are all going to look very alike. Some of them can degrade cellulose, some of them are endoxyloglucanases, some of them can degrade mannan. How do you know which ones can perform which activity? Okay, so you could say, well, maybe you can break them up into subfamilies, but that really requires a detailed understanding of what specific amino acids and what specific protein structures lend this sort of functionality. And so another example, really beautiful example that I want to give to you is from Matt Redenbaugh's lab. So they were studying a group of enzymes in the human gut, gut bacterial enzymes that are able to deglucuronidate different xenobiotics. In this case, it's a chemotherapeutic that can get deglucuronidated and reactivated in the colon and cause diarrhea. So individuals that were taking this drug had to come off of it if it was being reactivated in the colon because of the side effects. The thing is that almost all gut bacteria have these types of enzymes. They all have glycoside hydrolase family 2 enzymes, and a lot of them look like this. So what his group did was bin these into very distinct structural classes, and they found that really subtle changes in the active site, it really all comes down to these different loops surrounding a completely conserved active site, were what dictated whether or not certain bacteria, certain bacterial enzymes were deglucuronidating these drugs. And we can never get at this information if we just look at sequences and we just look at annotations alone. It really takes drilling down to the individual features of protein structures. Another problem with annotations is that some enzymes, some protein sequence that we label as enzymes, actually lack activity. For example, within the Bacteroides ovatus genome, there's a discrete genetic locus for the breakdown of the plant cell wall polysaccharide xylan. And there's a protein within this genetic locus that looks just like a xylanase, however, it's missing the catalytic base. It's not an enzyme, one single amino acid change. There's another example from the xyloglucan utilization locus. This is another plant cell wall hemicellulose. There's a surface glycoside hydrolase within this locus that looks like it should be really a robust degrader of cellulose, and yet it has almost no enzymatic activity. It degrades xyloglucan just a little bit. And yet it seems to actually be important for xyloglucan utilization. It seems to be acting more like a binding protein. But my point is that there are these protein sequences that, unless we drill down into the details, we don't really know exactly what they're doing. And again, of course, the other problem, and as many individuals have pointed out, I think Catherine Pollard mentioned yesterday, that something like 53% of genetic information really can't be assigned functionality. And certainly when it comes to looking at whether or not a bacterium or a community can degrade carbohydrates, we have the same problem with annotations. Nature always finds a way to surprise us. And so these were some surprises that came out of a study where this group in Harry Gilbert's lab was looking at how rhamnogalacturon 2, RG2, considered the most complex polysaccharide on Earth, it can be broken down by a single gut bacterium. And as they studied the sequential breakdown of this molecule, they uncovered 6 new enzyme families within this genome. And they discovered 2 existing enzyme families that have separate catalytic activity. And again, we don't have this information, we don't know these things until we take a step back and do some of those hard biochemical studies. So my group studies the molecular details of glycan utilization. We're really interested in how proteins recognize carbohydrates in the gut, how they process them and import them. And the reason we think it's important to drill down to these biochemical details is so we can understand why diet changes the community the way it does. We see in a lot of dietary studies that some people respond to diet in some way and some people respond to diet in another way. And of course we can say, well, you know, their microbiotas are different. Of course their microbiotas are different. But if we drill down and looked at the proteins that are present in the communities, can we, by using biochemistry, find specific molecular signatures within protein families that lets us know whether or not the capacity is there to recognize or degrade a particular carbohydrate. So moving really beyond the annotations. Another reason why it's important to understand the biochemistry of how carbohydrates are being broken down in the intestinal tract is because we can stop some deleterious activities from occurring. We can stop bacteria from binge eating the things we don't want them eating. Bacteria always have a choice of the different carbohydrates that they eat. And this was one study that came out of Eric Martin's lab, where they had mice on either a fiber-rich diet or a fiber-free diet. And you can see when the gut bacteria didn't have any complex polysaccharides to eat from the diet, they began to degrade the mucosal lining there. Well, which bacteria are performing this task? Which specific enzymes are they using? Rather than using an antibiotic to get rid of the quote-unquote bad bugs that are performing this activity, perhaps we can find targets to prevent this specific activity from happening, even in the absence of polysaccharides or in certain disease backgrounds. And so I think understanding which enzymes are participating in certain carbohydrate degradation activities can really provide us a window into changing the metabolism of the gut microbiota. And so I just want to address that. I hope that I've kind of demonstrated a little bit for you through these examples a critical knowledge gap, and that I think that we need to identify individual species, regulatory pathways, and proteins to help explain, predict, and develop personalized ways to shape the gut microbiome with diet. So how do we fill this gap? In the words of Bernie Onrezant, who was the curator of the Carbohydrate Active Enzymes database, we need in a lot of ways, those of us who do this work, to get back to the bench and to, as Kurt Huttenhauer said, do that hard biochemistry work in order to assign functionality to proteins and enzymes that we don't fully understand, to find those molecular signatures. We need to bridge collaborations between microbial ecologists who have large omics data sets, who have the data that shows how microbiomes respond to diet, and pull more biochemists and structural biologists into this so that we can help bridge this gap and figure out why some of these changes are occurring, why some people respond to diet and why some people don't. So like I said, we need to do the protein characterization. And particularly in the field of studying carbohydrates, we do need better tools for studying carbohydrates, for identifying them, for quantifying them, because this type of glycomics technology is severely lacking behind the technologies that we have for assessing bacterial community structure and even for identifying other metabolites present in the gut microbial community. So in the last little bit of time that I have, I'll just tell you a little bit about some of the work that I'm doing in my lab. Like I said, we're interested in the nitty-gritty details of how bacteria recognize and process different carbohydrates in the gut. And we look at a number of different organisms. The particular fiber that we've looked at the most is starch. So starch is a carbohydrate that humans can digest themselves, but there's a portion of it termed resistant starch that we can't process, and only a subset of gut bacteria can process resistant starch. The thing is that if you were to look through the genomes of every bacterial species, whether they can degrade resistant starch or not, they all have the right enzymes for degrading starch. So which ones can degrade resistant starch and which ones can't degrade resistant starch is one question that we're trying to answer. But the thing is, all of these bacteria not only have different enzymes and different enzyme specificities, but they also have organized their binding proteins for carbohydrates along with their enzymes in different ways on the surface of the cell that changes the way they see carbohydrates in their environment. So the Bacteroidetes kind of split carbohydrate binding and break down into different polypeptides on the surface of the cell that all work together. In the Firmicutes and other gram-positive organisms, there might be one large catalytic protein that has multiple carbohydrate binding modules that holds onto a fiber, breaks it down into pieces, and then a separate transporter that imports it. And then we're finding these really unique systems in some Ruminococcus, which is almost like a molecular Lego system, where there's a scaffolding protein and all these different enzymes can dock onto it so that you have this massive amount of hydrolytic activity for the breakdown of difficult to degrade fibers, including resistant starch. And so I'll just tell you very briefly about some of the stories in these different systems that we've worked on. We've been particularly interested in how these systems within the Bacteroides Bacteroides are organized. So this is a system that's involved in soluble starch degradation in Bacteroides theta iota omicron. And we had structurally characterized all the proteins that are on the surface of the bacterial cell. There's this one protein, SUS-D, that works with a transporter, seems to be incredibly important for getting pieces of starch into the cell. Even though it turns out this is a starch-binding protein, it doesn't need to bind starch to get things into the cell. And then there are all these additional starch-binding proteins on the surface of the cell, including an amylase, and somehow these proteins all work together. And we wanted to understand how these proteins work together to process and import carbohydrates. And so we decided to take a single molecule approach with Julie Bettine and her postdoc, Krish Krunitz-Laka. And what we did actually is we substituted this carbohydrate binding module that we see in SUS-G, this alpha amylase, with a halotag protein, which we can give a fluorescent ligand to. And now these are live anaerobic bacteria, and what you're seeing moving around the surface of the bacterial cell is an individual protein molecule of SUS-G. And when we have the cells fixed, of course, these proteins don't move around at all. And so we did this a bit in an iterative fashion, where we looked at the movement of our labeled SUS-G in the presence or absence of starch. And so here you can see that when starch is present, so up here it's not moving at all, you can see that SUS-G is adhering to the starch particle and then coming off. So it's really this dynamic assembly and movement of this enzyme on the cell surface. So we were interested in how the mobility of the protein was affected when other members of the SUS system were present. And so we could look at the diffusion coefficient of SUS-G in the absence of starch, and then see how the diffusion coefficient changed when we added starch, and then when we took away other components of the system. We can see that SUS-G slows down in the presence of starch. It speeds up again when we move other protein players within this system. And so we think this argues that there's this these dynamic interactions, this dynamic assembly going on while this system is degrading and catabolizing starch, helping Bacteroides to use starch. So not just the specific enzymes it has, but how these are organized with their binding proteins, how these are organized on the cell surface can make a difference in the way an organism is able to utilize a particular carbohydrate. We've looked at the Firmicutes. I'm not going to enter my iCloud password right now. I think I'll skip that. We've looked at the Firmicutes a bit just to see how some of their systems are different. And again, like I mentioned, what they do instead is they have one or a few different enzymes on the cell surface that have not only a catalytic domain for breaking down a fiber, but also several carbohydrate binding modules along with it. And the interesting thing about this system is that all of these CBMs that are associated with this are specific for cornstarch, but they don't recognize granular potato starch. So starch is not starch, and it comes down on a molecular level to how a protein actually recognizes specific motifs on a fiber. And finally, we've been looking into these Ruminococcus amylosome type systems, this molecular Lego type system of all of these different proteins that can assemble within a scaffolding. And we've been interested in how some of these enzymes can break down alpha-1,6 linkage, these branch points that we find in starch. And here we've crystallized one of these proteins with two different oligosaccharides positioned just as if this branch point in starch had been broken. And we think that these types of enzymes may actually represent a weak point where certain gut bacteria can begin to break down resistant starch, because this is a linkage that human enzymes simply can't break down. So with that, I hope I have in some way impressed upon you that there is room for biochemistry in the microbiome, and that maybe taking a step back and doing some biochemical experiments and looking for molecular signatures within omics data set can really improve our predictive capacity to understand how microbiomes work. So I have a small group of really wonderful people, and I'll just point out that all the work that I do with Bacteroides is in collaboration with Eric Martens. I mentioned Julie Bettine with the single molecule imaging, Daryl Coburn, who is now a professor at Penn State University, did a lot of work with E. rectale as well as Ruminococcus bromii. Ryan Kibler, Rebecca Pollitt, and Matthew Foley are also members of my group that have contributed to these projects. Thank you.
Our next speaker in the session will be Johanna Lampe, who will talk about targeting diet-gut microbiome interactions, the use of controlled feeding studies in humans.
Great. Well, thank you very much to Cindy and Lita and the organizing committee for an opportunity to participate in what's really been an exciting day and a half, and I expect this fun will continue as we move forward. I'll wrap up this session talking a little bit about some of the issues involved in using controlled feeding studies and testing diet microbiome interactions. And we've heard through the course of the last day a number of very elegant examples of some of these approaches. So I'll sort of fill in some of the gaps with regards to the hows and whys, some of the challenges, and where we go from here. We use the word diet very loosely, and I think this is one of the things that sometimes catches us unawares or can trip us up, in that we really are talking about an extremely complex exposure of thousands of compounds, particularly the human diet. For those of you who work in Drosophila or in some of the more simple models, you can really simplify the diet extensively. We've yet to find study participants who will put up with long-term feeding on the same food every day. So we really are talking about a complex group of mixtures, which we can characterize on many levels. We can talk about dietary patterns, we can talk about specific foods or classes of foods that are certain sources of compounds, or we can focus on individual compounds, whether they be nutrients that we've identified as essential for life or that we've identified as contributing to improved health or increase in disease risk. And particularly in Western countries, we also have a whole slew of components of our diets that are actually supplements, either as food or as specific compounds. So when we talk about controlled feeding studies, we're really speaking about a situation in which we provide the food to the study participants. So these are diets that are designed, prepared to specifications related to the hypothesis that you're interested in testing. And as in the case of Gary Wu's example, you may conduct these studies in the context of a metabolic ward where people are sequestered in particular space for a certain length of time. Those are extremely elegant but complex studies to conduct because they're expensive. You've got to find study participants who are willing to be locked up. Often it's graduate students writing their thesis in the case of healthy individuals, which it does sort of limit the generalizability of your activities. I think one of the things to keep in mind is that these studies are really designed to measure biologic, physiologic effects of dietary manipulation. They're really not designed to look at the application of a dietary intervention in real-world conditions. So we're really, as some of my colleagues would say, you're doing rat studies in humans. And if that's the way you want to think about it, sure, but it really does sort of speak to the idea of really trying to get at the biology, trying to address specific hypotheses as far as mechanisms to the extent that we can in intact humans. So as you might imagine, there's a number of useful components of controlled feeding studies, but also limitations that just come with both the approaches as well as working in humans. The nice thing about this is from the standpoint of thinking of what you can control, you are not only controlling the intervention, i.e. what is the specific food, components of food, et cetera, that you're wanting to test, but you can also do this on the context of a controlled background diet. Putting everybody on either a high-fat diet or in the context of a particular designation, relation, combination, carbohydrate, protein, fat. You can set these up to test dose response, where you're either providing specific doses across a group of individuals, or you're actually running this on a continuum of dosing on the basis, for example, of body weight or other factors that allow you to examine this. Really, as I said, with an emphasis on biologic and physiologic properties, you're really focusing on biomarkers of effect. And we can also use these types of studies to develop biomarkers of dietary exposure and in many cases also, as particularly in relation to this conference, really thinking about biomarkers of bacterial metabolism. Now, of course, there are always some limitations. These are typically short-term interventions. Nobody likes to be provided all their food for weeks to months on end. I will say there may be a few who are willing to do this, but for the most case, we typically start to see mutiny after 6 to 8 weeks. So really, this is something that we rely on short-term effects. We also have to rely on intermediate biomarkers. We can't carry this out to the point of development of disease as the outcome. So in that context, we were slightly hamstrung. So by way of example, I'll show a couple of studies that have really used controlled feeding studies to test dietary pattern microbiome interactions. And these are probably the most complex of the approaches compared to something where you're looking at the addition, for example, of a specific supplement or particular food. And we really are looking at several questions. What is the impact of a dietary pattern on gut microbiome? And how does the diet pattern microbiome interaction affect disease risk biomarkers in the host? So why bother with dietary patterns? Well, at this point, most humans still eat foods and a variety of different foods. And we really have come to recognize from the standpoint of making dietary recommendations and public health recommendations for long-term behavior change or for long-term viability of approaches that we really need to focus on proposing food choices over the broad continuum of types of foods that are available to us. From a research standpoint, it really helps to offset the limitations of a single nutrient approach, where we know from the standpoint of epidemiologic studies, or even in the context of foods in general, that nutrients are often intercorrelated. Types of foods that are consumed together are often correlated. This also helps to preserve the multidimensional aspects of foods as opposed to feeding specific nutrients. We can include all components of the diet, both nutrients and non-nutrients. And we can take different approaches to studying dietary patterns. You may have run across some studies where there are comparisons between traditional dietary patterns compared to Western diets, some mechanism-based indices where we know that particular diet patterns are associated with increased systemic inflammation, so characterizing a less inflammatory diet versus more inflammatory diet, for example, and then also comparing typical diets against an established recommended dietary pattern. If we're going to go out and tell people that you should make these dietary choices, what is the impact of that diet pattern in contrast to what people are usually doing? So by way of an example, there have been a number of extremely elegant short-term feeding studies looking at rather extreme differences between diets, and this is work out of Peter Turnbaugh's lab from a number of years ago. A crossover design of 5 days of either a very heavy animal food-based diet compared to a diet that's devoid of animal foods and is predominantly plant food, compared to individuals' baseline traditional habitual diets. And as you can see, the percentage of energy from fat varies across these, with animal diet being very high in energy from fat, high in protein, and in contrast, less percent energy from fat, lower protein intakes, as well as a substantial intake in fiber, both compared to the animal as well as to the individual's habitual diets. And as they showed, that when you look at the effects of these diets on the gut microbiome, particularly in the context of looking at metatranscriptomics, you can see substantial differences when individuals are on the plant-based diets versus the animal diet. In contrast, and we've seen this over the course of several presentations, is that we often don't see the same effect from the standpoint of changes in the relative abundance of particular taxa. And here the black dots indicate that really you see no separation of the same individual dependent on whether or not they are on the animal or plant diet. But as indicated, we see differences in microbes involved in bile tolerance, changes in microbes that are metabolizing plant polysaccharides, as you might expect given the fiber content. But really the idea that in the context of humans, in contrast to many of the rodent work, much of the rodent work, is we often don't see those same beautiful separation on two different diets. Stephen O'Keefe's work also looking again at substantially different diets, feeding African Americans a native South African diet, i.e. going from a diet for the African Americans that was at 14 grams of fiber per day up to 55 grams of fiber, and a substantial reduction in dietary fat. In contrast, taking the native South Africans, reducing them from their usual 66 grams of dietary fiber down to around 12 and upping the fat. In the context of this, you see substantial changes in microbiome activity, again from the standpoint of the fiber fermentation story, as well as conversion to secondary bile acids. I think the additional component of this is they took colon biopsies at the end of the intervention period. This was, again, short-term intervention, but saw substantial changes in colorectal cancer-relevant components in the colonic epithelium, seeing decreases in proliferation in the African Americans when they're put on the South African diet, and in contrast in the Africans on the African American diet, substantial increases in proliferation. Similarly, marked changes in inflammatory markers in relation to changes in the diet, suggesting that both the combination of the effect on the microbiome as well as on the host may be contributing to some of the risks that we see an increase in colorectal cancer risk in the U.S. African Americans. So wrap up here with a couple of examples from some of our work using controlled feeding studies that were controlled feeding study that was designed to look at low and high glycemic load diet patterns in healthy individuals. So these were individuals who were not insulin resistant, had normal fasting blood glucoses, and looking to choose food sources that either had low glycemic indices or high glycemic indices, fed at eucaloric diet levels to maintain weight over the span of 4 weeks. And really the low glycemic load diet ended up to be substantially high in fiber, at about 55 grams per day, high glycemic load, although it was about half of that. It's still a relatively high fiber intake for our typical US population. As part of the initial study, we were interested in a host of circulating metabolic and inflammation biomarkers. This was part of the TREC study, or Transdisciplinary Research in Energetics and Cancer. And this is also by way of encouraging those of you who don't necessarily do feeding studies and don't see yourselves doing feeding studies to engage colleagues who do, or even if you read a study and you think, oh, cool, I'd like to ask this question in the context of this type of intervention, to contact those individuals, because we typically squirrel away lots of samples in order to be able to continue to work in these areas, because these are expensive studies, and so this provides us with good opportunities. So just by way of the effects of these diets as far as glucose excursion, you see substantially lower area under the curve for glucose as well as insulin, as you would expect in this type of study. So we're now using this as in a multi-omic approach to look at the effects of these two diets, the impact of the gut microbiome in relation to the plasma metabolome, and ultimately biologic pathways relevant to cancer risk. From the standpoint, again, a controlled feeding study, this was the crossover design. So we've got stool samples that are being interrogated for 16S, metagenomics, metatranscriptomics at the end of each intervention period, plasma metabolome, a couple of different platforms, and the plasma proteome using an antibody array approach for analysis. So just by way of example, when we look at 16S, we really don't see any difference between the effects of the low or high GL diet or comparison to the baseline. These black dots here are all our QC control, which are clustering together in one spot. Looking at the cladogram, we do see differences in certain taxa with regards to the low and high GL diet, changes in relation low GL diet with Parabacteroides, and then in the high GL diet, a couple of members within the Clostridia group. As we can move into the metatranscriptomics and the metaproteomics, we'll have another opportunity to look at some of these additional questions. From the standpoint of the metabolomics, we are able to separate slightly better the low GL diet with the triangles as opposed to the high GL diet. And there are a number of targeted compounds that are different. Some are higher in relation to the low GL diet, and some are lower, i.e. they're higher on the high GL diet. So integrating these two pieces will give us more information in this area. By way of thinking about, we've talked about phenotypes with regards to microbial metabolism, and I think particularly in the area of metabolism of plant compounds, there is a lot of heterogeneity in how individuals' microbial communities handle these. This is by way of example, the lignans, which are compounds, diphenolic compounds found in plant foods, typically whole grains, fruits and veg, and we actually which undergo metabolism by a consortia of bacteria to produce enterolactone, a bioactive that's been shown to be associated with lower risk of several cancers. Now, if you give everybody particular doses of lignans, you don't necessarily see a nice dose response as far as enterolactone excretion. This is not unusual, but it again speaks to the heterogeneity in response. We do find that in context of this feeding study that actually there are several of the secondary bile acids that typically are positively associated with enterolactone excretion. These are plasma bile acids, not luminal bile acids, suggesting some differential response on the basis of capacity to produce enterolactone. Some of you may have had a chance to see a poster yesterday of Rob Chapkin's, again, considering the issues of effects of the gut microbiome on production of enterolactone and the variation in the capacity to produce this. This was a study actually looking at the effects of lignin supplement on gene expression in colonic epithelium, in which we took sigmoid colon biopsies at the end of each intervention period, and we were looking at response to the intervention. What we found was that although there was a modest response to the intervention, it really varied depending on whether or not the individuals were low or high producers. So more to follow on that. Knowledge gaps, needs, and challenges. I think this recapitulates what's been said several times, is our strong need for well-annotated metabolomics platforms that really capture the range of endogenous as well as exogenous compounds and their metabolites. And also, as we get to the point of establishing all the genomes on the various types of bacteria, to actually be able to improve our approaches to characterize the microbial metabolism that involves consortia of bacteria. We often focus on specific microorganisms, but to find ways to then integrate this in order to get through consortial-based metabolism. And I'll skip the summary since I'm over time. Just very much want to thank Meredith Huller, who's a microbial ecologist who's been working with our group for the last decade or so, as well as collaborators at Texas A&M, Rob Chapkin, and my collaborators at the Hutch. Thank you.
The session's now open for questions.
Hello, Brandon Eicher. Excellent discussions. I don't know if anybody really knows there's a raging debate of all things dietary fiber going on in the FDA right now. I think that there's a lot of challenge around trying to define what dietary fibers are. And I think I would like to pose to the panel and the group of you, that we need to mind the fiber gap. And I think our colleagues at the FDA need a lot of assistance from the scientific community to help understand more about what that does structure function for the host. And let me cite an example. So things like resistant starch are not currently being considered a dietary fiber on the list of approved fibers. So I think there's probably several people in this room that would disagree with that. And I was wondering, I kind of want to take it to a further step and really try to find a way to define microbiota accessible carbohydrates as an essential nutrient. And there's some challenges around essential nutrient because it's something that we must have for life. And I think there's a bit of an argument against that in the FDA currently. So I'd just like to hear your opinions on defining dietary fiber as an essential nutrient and how we might be able to do that in the future. Thank you.
Yeah, great question. I'll be curious to see how the debate plays out right now. I think there's a lot of questions. I think we saw with Andrew's talk this morning. I think there's, context determines a lot. What microbes you have in your gut, what other diet you may be eating, how rapidly things are fermented, where they're fermented in the colon. So there's still a lot that we have to learn. I think it really validates the approach that I and others have pitched here, which is to study some of these in a controlled setting where we can assess what they're doing in humans. I think we don't really have a good grasp of how complex this is, and I don't think there's going to be a simple answer to that question. It's a little bit of a bailout answer, but I'd be curious to hear what other people have to say.
I think that's a really good point, and I think one of the challenges also relates to the fact that in human studies, we often don't take into consideration the structure of the food from the standpoint of Justin's approach of looking at what's microbial accessible carbohydrate. It really depends on whether or not it's covered in a bran layer or it's chopped up in little pieces. And so from the standpoint of thinking about fiber, you could argue that anything from the standpoint of a carbohydrate that gets to the lower gut is really going to act as a fiber, and it really does muddy the waters when we try to come up with a particular definition of specific compounds, et cetera.
I would just add that, the gut microbiota, as we heard, is deeply fermentative and has a lot of enzymatic activities, as Nicole had pointed out. And so we focus on fiber, keeping in mind that the human diet is to a certain degree, somewhat reciprocal. In other words, high plant, high fiber diets tend to be lower in fats and meat, and vice versa. And meat and fat, we do know, has effects direct on a host, maybe independent of the gut microbiota. So I just want to make the point that it's not just one thing that changes when you change a diet. It's probably other things that change in a reciprocal fashion that may also have an effect on health.
Hi, Amir Zarampar, UCSD. So I have two questions. One is, 4 different labs have shown that the gut microbiome is highly cyclical and has circadian changes and in fact controls host circadian rhythms. For example, we know bile acids are circadian in terms of which ones exist in the lumen. We know that all the FXR downstream transcripts are circadian. How can we, and this is true for practically anything that the gut is absorbing, so how can we really understand this system when we're not looking at it as a dynamic system? That's number one. And number two, most of this digestion, 95% of the bile acids are absorbed in the terminal ileum. So wouldn't the intestinal microbiome play a far more important role in digesting fiber, digesting fibers, unless it gets down to short chain fatty acids, or digesting proteins, or digesting fats, and all these things, more so than anything that we're measuring in the stool?
Well, I'll give a shot at one or two of those things. So the I think what you're saying in terms of bile acids, I'm not sure. I mean, I think of bile acids as being important emulsification and the absorption of fat-soluble vitamins and lipids and things like that. I don't know. I don't really have a good knowledge of how that would deeply affect fiber digestion by the microbiota. Having said that, as Jeff mentioned this morning, I think an unknown domain for us largely is the small intestinal microbiota. And I've made the point that I think when I think about nutrient intake and the response of the microbiota to diet, I think, and although not a lot of evidence because we haven't really explored it, that the small bowel microbiota actually may be much more dynamic. And when you think about all the receptors in the small intestines, all the enteric hormones that are made in the small intestine, FXR, nuclear hormone receptors, and bile acids, that type of biology is largely unknown. And I would say that that's another area, a significant gap that Jeff pointed out that I actually do agree with. The circadian rhythm thing, I think that it's a fascinating area. I think some of that is sort of dependent on when you eat and perhaps what you eat. I think it's a challenge, and I've actually had people come to talk to me about studying circadian rhythms in a microbiome in humans. And it is actually quite a challenge, right? Because mice you can take out of a cage and you can put in a box and it'll poop all the time. But humans don't do that in a way that you can actually study circadian rhythm. So there are inherent challenges. I would be interested in knowing other people have figured out a way to go about doing that. I'm not smart enough to think about a solution to that problem.
Yeah, just one quick add-on to that. I think the concept of sensors, not just flowing through the digestive tract, but perhaps being tethered to sites where you can measure what's happening at a given site over a long period of time could be really valuable.
I'm not here to talk about sensors. I guess when I listen to your talks, and I think they're really, really impressive, but since this is a panel discussion, I feel like every time I hear these, we're seeing one part of the elephant. Somebody after fiber, somebody after this, somebody after that. And I do feel like we need a Foodome project that's no less powerful, impressive than the Genome Project was, where we really lay out a map and systematically go after all these different variables. And I hadn't thought about circadian, although I do every time I eat breakfast. Wonder whether the effect would be different at lunch. But that point aside, I do feel like we need the systematic method, if you will, or approach to really go after this in a comprehensive fashion where we could really tear this apart. What are your thoughts there? It's a wide open.
I think it sounds lovely. I think trying to get it funded would be extremely difficult. When you think about trying to integrate all the possible combinations, that if you're thinking about this as a totality of diet, to do each one separately is going to give you one little snapshot, but it's not going to cover the totality of diet, which is what people are eating.
Yeah, I mean, you'd probably have to do it in combinatorics in some fashion and then tear it apart with some sort of multivariate analysis. I recognize some of the issues associated with that. But nonetheless, I would argue that if we go the way we're going, it's going to take several decades to sort this out. And I think that's too long. If you look at the epidemic that exists, we're just not approaching this right. Again, given the magnitude of the problem, this problem is no less than cancer in terms of diabetes and obesity and things. Yeah, again, we're going at it in a very trivial fashion, if you ask me, one R01 at a time, and that's just not going to solve the problem, I think.
Yeah, I mean, I think the reason I presented the work on dietary patterns is that really is a direction that the nutritional field is also taking from the standpoint of being able to make recommendations. We know that looking at large-scale epidemiologic studies of thousands of people, that individuals who follow a diet pattern that, as recommended, higher in fruits and vegetables, low in red meat, et cetera, have lower risk of cardiovascular disease, have lower risk of several different types of cancer in prospective studies. So starting at that point where you say, okay, what does that diet look like? What are then some of the biologic and physiologic as well as microbiome pieces would be one way.
I actually agree with Joanna on this. We're not starting from zero. So I use inflammatory bowel disease as another example, all right? So we have diets that work in inflammatory bowel disease. Antibiotics, metronidazole and ciprofloxacin, have been used for a long time for the treatment of Crohn's disease. And even now we're seeing that fecal transplantation with numerous inoculations can have a modest effect. So these tell us that these things are actually effective in human treatment, but they just don't work as robustly and as significantly as consistently as we would like them to work. And so that's adding our newest technologies to try to come up with better ways of actually doing what we actually sort of see to a certain degree. So I don't think we're starting from zero or trying to improve something that we've already seen, at least in some diseases. I have to say that in terms of nutritional biology, as a lot of you know, there is a trans NIH initiative now thinking about the next decade of research on nutritional biology. I don't know if the program officers want to mention this, but that the same issue sort of applies there. Like nutritional biology is exceedingly complex. How do you actually study it? We have now wonderful technologies. Maybe we can actually better define biomarkers for what actually people eat, because just measuring using dietary questionnaires is the best that we have right now, but it's a very poor surrogate in terms of reliably quantifying what people actually eat.
I might just add one comment. You know, I don't think the problem is any less complex than when people thought about the Genome Project back in the early 80s. It sounded impossible. It sounded too daunting. But I think because it was put out there as a goal, people then came up with ways to go at it, and new technologies came along the way. And I think, to me, that's how I would try and solve a complex problem. Sorry, Justin, didn't mean to cut you off.
Yeah, I mean, I think that one issue is thinking about treatments for specific diseases, but I think the other way to think about the diet microbiome axis is as this incredible lever that we have on human biology to toggle so many aspects of metabolism, immune function, our central nervous system. But we know very little about that wiring. And I think what you're getting at, Mike, is a systematic effort to try to figure out that wiring so that if somebody needs immunotherapy, we know what to feed them to get their microbiome to move in the right way, to move their immune system. Alternatively, if that same person were fighting an autoimmune disease, it would be a different treatment to move their immune system in a different direction. And I think, thinking along those lines and figuring out how to do that systematically would be a wonderful thing to do. I was at a diet microbiome workshop earlier at the summer at the NIH, and there's really no consensus in many different methods for how to even measure what people are eating. And just to be able to come together and find what best practices are for that that's a major hurdle in this field, because that's what everything is based on, that your downstream analysis is relying on. So I think that there really could be huge improvements really rapidly, and then a really nice big effort would be great.
So we're going to have to wrap things up, but just in response to what Gary was talking about, one of the things that NIH is working on now is a 10-year strategic plan in regards to nutrition. And if anyone has thoughts or suggestions, please feel free to contact Chris Lynch. If anyone needs his contact information, we can get it to you. But please join me in showing our appreciation for all the speakers in this session. And in order to get back on schedule, the break will be 20 minutes, and we'll be starting again at 3:50. Thank you.
Okay, for the interest of time, I'm going to start if everybody sits. Pretty much there. So welcome to session number 8, the role of the microbiome in disease initiation and exacerbation, moving beyond association. My name is Liz Kaller. I'm a program director at the Heart, Lung, and Blood Institute in the Division of Lung Diseases. And the purpose of this session was to highlight concrete cause and effect mechanistic examples of the microbiome role in disease initiation or exacerbation, what is not yet understood about the microbiome, how it initiates or contributes to disease. And hopefully we can get enlightening clues from our wonderful speakers. We have 4 speakers. Andrew, you cannot hear it now? Here now? Yeah, the echo is hot. Can you hear me now? No? Okay. Should I start over? Okay, now it's better, I think. Can you, is it? Yeah, okay. Sorry about that. So I was saying we are in session 8, the role of the microbiome in disease initiation or exacerbation, moving beyond associations. And my name is Liz Kaller, Program Director at NHLBI, Division of Lung Diseases. And the purpose of this session is to highlight concrete cause and effect mechanistic examples of the microbiome role in disease initiation or exacerbation, what is not yet understood about how the microbiome initiates or exacerbates disease, and what is needed to make progress in this area. We have 4 wonderful speakers: Andrew Goodman from Yale University, Wei Jia from the University of Hawaii, Cindy Sears from Johns Hopkins University, and Gary Hefnagel from the University of Michigan. So we're going to start with our first speaker, Andrew Goodman, and the title of the talk is Efforts to Understand Causation in Microbiota-Host Interactions, 2 examples.
It's actually just going to be one example due to uncertainties of our budget climate or just due to lack of time. So I'm going to just jump right in to some of the approaches that we've taken to get at this question of causation in host microbiome interaction. And I'll start with this. So maybe some of you have seen this before. So this is a correlation. You can see there's 2 types of data here that are very well correlated with each other, 91% correlation according to data from the USDA and the American Bar Association. So what you can see that these two measurements are the amount of sour cream that's consumed in the US per capita and the number of lawyers in New York. So what you can see is that although there's this very nice correlation between these 2 increases, I hope you can realize that if you find yourself eating sour cream, it's not because there's more lawyers in New York. But in the context of the microbiome field, this can actually be quite a challenge because we can see, again, these very nice trends, and then I think part of our challenge is to think of different ways to disentangle and differentiate this kind of example from the types that we're actually interested in. So one way to do that is if we had the ability to manipulate the variables that we think are related to each other. So if we can change one of the 2 variables and ask whether the other one changes in response, then we can start to get a sense of whether there's a causative element one way or the other. And that's what I'm going to talk about today, is some of our efforts to manipulate elements of the microbiome, in this case specific genes and enzymes within gut bacteria, in order to identify causation. And the example I'm going to talk about, I'm going to give you an example of how we applied this approach to understand this observation that there are many pathogens that seem to be associated with antibiotic depletion of the microbiota. So this is a pattern that's seen in many examples, but the underlying mechanisms for how antibiotics change the microbiota to facilitate pathogen invasion are still being discovered. So before I get to this example, I want to tell you about this challenge that we have, and I'm going to bring this up right at the beginning. So I'm going to tell you about some approaches for looking at the Bacteroides, which is a member of the Bacteroidetes here. By some estimates, the most prominent genus in the gut, but certainly not the only one, and in certain individuals, not even the most dominant one. So right now I'm going to say that I'm going to propose that if I convince you of any utility of the approaches we're taking, none of them work in the firmicutes. And I would say that one of our challenges is to spend the time to develop some of these approaches for this very important majority or major phyla in the gut. Okay, so this wish list that we had for working with members of the microbiome in general was really pretty simple. We had for many years wished that we had an ability to turn genes off and on in the microbiome. And further, we wanted to do that using some kind of signal or inducer that wasn't already in a mouse or already in the diet of a mouse, so that we could really control what's going on in a gut commensal while it's inside the gut of a live wake animal. And I also don't need to tell you that although these tools may seem modest, especially if you work on E. coli or other proteobacteria, E. coli is actually quite a rare member of the microbiome and phylogenetically very distinct. And so it's been a general pattern that tools developed for E. coli don't work in members of, for example, the Bacteroidetes, which are a different phylum entirely. So the approach that we set out to take was to use some tools that had been developed in E. coli and then completely rebuild them from scratch in order to use them in members of the Bacteroidetes. And so the approach we used was this TET-R-based gene regulation system. This basically has three parts. The first is an TET-O operator sequence here. So this is a DNA element that is you can put into the promoter of a gene of interest. This operator sequence is bound by the TET-R repressor, this protein here, and when the TET-R repressor binds to TET-O, it blocks gene expression. And then the third part of the system is a tetracycline or a tetracycline analog, such as anhydrotetracycline, which is a synthetic molecule that binds to TET-R and basically sequesters it away from the TET-O operator. So in this way, this double negative means that ATC is an inducer of gene expression. So again, these systems had been developed in E. coli, but none of those promoters work in Bacteroides. So we started from scratch. I should say that the way we measure this is by putting some sort of reporter gene downstream of the engineered promoter. And so in many cases, I'm going to show you data using the nanoluc luciferase, where we can measure luminescence that's produced in direct correlation to how much of this protein is made over many orders of magnitude. So we started with a genome approach where we took highly expressed promoters, 180 highly expressed promoters from across the phylum Bacteroidetes, and made a consensus sequence here. And what this showed us is that there are regions of high conservation, but then also quite variable regions that potentially we could insert these TET-O operator sequences in. And so with a lot of trial and error, we did that. We found two locations where is either an insertion or a partial substitution in a specific orientation, we were able to get these TET-O operators into a representative promoter without killing the activity of the promoter entirely. And we did this quite a bit to study this in vitro. I'm not going to show you any of the in vitro data. I'm just going to go right to the in vivo data. So we took first gnotobiotic mice that had been colonized with 14 different species of human gut microbes, including an engineered version of Bacteroides theta iota omicron that has this nanoluc reporter. And so what you can see is when we put ATC in the drinking water of the mice on day 4, that we get this increase in luminescence that increases by orders of magnitude when we add ATC. When we remove ATC from the water, then the bacteria inside the gut turn off this reporter, and then we can see luminescence go down. And so what this tells us is that there's nothing in the mouse or the mouse diet or these other species that's interfering with this inducible system. And that's because this inducer is synthetic. It's not present in the gut unless we add it. We can also do this in SPF animals that have a complete microbiota. I should say here that when we do quantitative PCR analysis of the rest of the community, we see that although there's some drift over time, there's no specific ATC-dependent differences in the microbiome, suggesting that this inducer is not impacting other microbes in this defined community. We can do the same thing in conventional mice that have a complete microbiota of their own. So in this case, the engineered B theta strain makes up about 0.1 to 0.5% of the entire microbiota. Yet when we add ATC to the drinking water, we can turn on this one gene inside the genome of this one bacteria that's 0.5% of the entire microbiota. And again, what you can see is that in the absence of inducer, we don't get any expression, telling us that there's nothing in the complete microbiota either that is either turning on gene expression when we don't want it to, or preventing the ability of this inducer to activate gene expression at a specific time. We used high-throughput culturing to capture Bacteroidetes from unrelated healthy donors. We inserted this genetic system into those wild isolates and found in every case the system works the same as it does with our type strains. So now I'm going to come back to this question about trying to understand some of the mechanisms of how antibiotics and microbiome and pathogen infection may interact using this system. And I'm going to start with an example that was very nicely worked on by Justin Sonnenberg in, I think, 2013, looking at one of these mucus glycoproteins that have sialic acid as a terminal residue. And so what's clear is that there are many commensals and also pathogens that can consume sialic acid once it's liberated from these glycoproteins on the gut epithelium. There are certain commensals that have sialidase enzymes that have this sialic acid liberation capacity, but even though these enzymes are ubiquitous in the microbiome, the levels of sialic acid are actually fairly low in the lumen. And that's because there's also other commensal microbes that can consume the sialic acid. However, if this balance of things is disrupted, then the sialic acid can serve as a nutrient for pathogens, including Clostridium difficile and Salmonella typhimurium. And so, what Justin's group showed in 2013 is that they could see C. difficile colonization levels dependent on the ability, at least in notobiotic mice, of B theta to encode this sialidase. So this could be complemented either by restoring the sialidase or by providing sialic acid directly. And what they also observed was that if they gave antibiotics to disrupt the microbiota, this somehow changed things, and the sialic acid levels in the lumen of the gut went up significantly. But it wasn't clear how this was occurring. And you could imagine a lot of different explanations. It could be that there's a certain microbe in the gut that responds to antibiotics. It could be that there's a specific antibiotic that's necessary to cause this effect. Or it could be that this is something general that we'd expect would translate to a different mouse colony, or maybe even to other organisms, including humans. So I think that this ability to control gene expression can help us address these kinds of questions experimentally. So what we did is we took this engineered system, and instead of coupling it to a reporter, like a luciferase reporter, we coupled it to this sialidase that's responsible for releasing sialic acid into the lumen by B theta. We call this strain BTRS for regulated sialidase. So then we colonized germ-free mice with this engineered strain. We gave them a drop of ATC in their drinking water, and then we followed a few different measurements over time. So the first thing we measured was the inducer itself, the ATC, and you can see that after we add it to the water, we can see it in fecal samples collected from these animals, and then it disappears within about 2 days. The next thing we wanted to measure was actually how much sialidase protein, an active protein, was being produced as we turn on and turn off gene expression using the engineered system. And so to do this, we developed an ex vivo assay where we took fecal samples from these mice and then incubated them with an artificial substrate that included sialic acid. So this is this p-nitrophenol sialic acid here. Sialidase cleaves right here, releasing this nitrophenol group, which we can then measure. And so we developed an enzyme assay on fecal samples where we could convince ourselves that we provide the substrate in excess, so we're measuring the initial velocity of this reaction. And you can see that as we dilute the fecal samples, we get different reaction rates, as you'd expect for a typical biochemical enzyme assay, except this is being done on feces. So then we could confirm that if we take fecal samples from mice colonized with wild-type B theta, the ex vivo sialidase activity is consistent and high. And if we take mice colonized with a mutant that doesn't have the sialidase, then the ex vivo sialidase activity is low, as we'd expect. Then we can do the same thing on the regulated strain where we're tuning gene expression. And so what you can see is that after we add the inducer, sialidase activity, as measured by this ex vivo assay, reaches the levels of a wild-type organism and then goes back down to 0. And then finally, we can measure sialic acid itself that's being released from the epithelium into the lumen, and then we can measure that in fecal samples, and that's shown here. So these measurements taught us two things. The first thing is that when we look at when the protein is present and when the product of this reaction is present, you can see that they're not exactly concordant, and that there's this window of time here where we have the product, the free sialic acid, is present in the gut, but the bacteria no longer have the sialidase activity. We know that because we turned it off, and we measured that it was turned off. So for us at least, this is the first time where we've started to think about what the dimension of time means when we look at microbiome phenotypes. So when you see that there's a certain metabolite that's higher or lower in a certain sample, is it necessarily an activity that the microbiome is doing at that time, or is it a legacy effect of what the community was doing earlier? And at least in this case, this is several days where the product of the reaction stays around even when the activity isn't there. The second thing that we learned is looking at the dose-response relationship. So here, this is the same data just plotted against each other. Each point is a mouse, and we have the activity, the sialidase activity on the x-axis, and the amount of product on the y-axis. What you can see here from this relationship is that there's a part of the curve where the amount of sialidase activity you have tells you how much sialic acid you're going to produce. And that kind of makes sense. But there's this other part of the curve where the release of sialic acid is not determined by how much enzyme activity you have. In other words, if you have these high levels of sialidase activity or these relatively low levels of sialidase activity, the amount of product is the same. And what that tells us is in this part of the dose-response curve that the release is not enzyme limited. And this is, again, something that we, at least in my lab, haven't really thought about in the context of the microbiome. We see a gene upregulated. Does that mean that the actual consequence of that enzyme will be more? And I think that may well not be the case. And in conventional mice, we can see that the sialidase activity is very high, well into this range where changes in the amount of sialidase activity, if we deplete how much sialidase-producing microbes are there, is going to have no effect on how much sialic acid is actually released. And so we can think about this in the context of antibiotics in terms of supply and demand. So what we know is that free sialic acid is not determined by sialidase levels. We know that because we can tune it and see the response. But we know that the demand is determined by how many microbes are there because those conventional mice don't deplete sialic acid again to zero. So in other words, when we think about what antibiotics will do, they'll decrease the demand on sialic acid, but they won't affect the supply. And that explains in a very general way why sialic acid levels go up with antibiotics. We don't need any specific microbes or specific antibiotics to explain what happened. So to summarize, and we can model this using parameters from the experimental data showing how much of an antibiotic effect would be expected to increase sialic acid. So just to summarize, the first thing I hope to leave you with is, again, New York lawyers do not cause more sour cream consumption, and you should think about that in terms of microbiome studies also. I hope I've convinced you that it's becoming possible to tune microbiome gene expression, and then that may help us connect genes to functions. And I think for many of the things we're thinking about with the microbiome, whether or not we're considering dose-response curves and timing is something that should be considered. And this was recently published, and I'll just stop by thanking the group, especially Bentley Lim, a postdoc who did the project. Thank you.
Wonderful. Our second speaker is Dr. Wei Jia from University of Hawaii, and the title of his talk is Bile Acid Microbiota Crosstalk and its Effects on Liver Cancer.
Thank you, Liz. Good afternoon. I'm very glad to have the opportunity to talk about our research in the area of bile acid and microbiome crosstalk and their effects on the liver cancer. And once again, my name is Wei Jia. I'm the professor at University of Hawaii Cancer Center. The UH Cancer Center is a National Cancer Institute designated cancer center. So the cancer research programs are supported by several well-established, cutting-edge core facilities at the Cancer Center, one of which is Metabolomics Core Facility. I am the faculty director of the Metabolomics Core. One of the highlights of our lab is that recently we have developed a microbial metabolomics protocol that quantitatively profiles somewhere between 200 and 250 small molecule metabolites that are derived from host microbe co-metabolism. We believe this is a very important tool that provides unprecedented opportunities for functional characterization of gut microbiome, in addition to the metagenome sequencing. In a review paper that we published along with Dr. Jeremy Nicholson and a few other collaborators, we summarized a number of important metabolic pathways that are involved in the gut-liver and gut-liver-brain metabolic interactions, and one of which is bile acid nuclear receptor axes that we are going to focus on today. Personally, I really like to study bile acid metabolism because I believe that is the best model, or that is the best pathway to study the interactions between the gut microbiome and liver, because the bile acids are produced in liver, in hepatocytes, from cholesterol to what we call primary bile acid, CA and CDCA, and then will be stored in bile after they are conjugated with amino acids, glycine or taurine. And then they will be released into the small intestine, where they are extensively metabolized by intestinal bacteria through deconjugation, through 7-alpha dehydroxylation, all the way to what we call secondary bile acid, DCA, deoxycholic acid, and lethocholic acid. And these secondary bile acids are, comparatively speaking, much more cytotoxic than the primary bile acids. And these bile acids will be transported back to the liver through a number of intestinal transporters like ASBT, OST alpha, OST beta, MRP3, through the portal vein, and then to the hepatic NTCP transporter to complete a enterohepatic bile acid circulation. So we know that altered bile acid synthesis and metabolism has been considered a common etiological factor for almost all human liver diseases, including alcoholic fatty liver disease, NAFLD, and hepatic cellular carcinoma. And we also know that the intracellular accumulation of bile acids, especially the microbial metabolized secondary bile acids, they are more cytotoxic, and it has been proposed as a mechanism of cholestatic liver injury. And we suspect that increased intrahepatic bile acids may be a mechanism of liver carcinogenesis. So we recently conducted a targeted bile acid profiling study of liver fibrosis patients, where we recruited 1,006 participants, including 504 patients with biopsy-confirmed chronic Hep B or chronic Hep B associated with fibrosis of various stages, as well as 502 age and gender matched healthy subjects. So the bile acid profiles pretty much tells us that they can clearly differentiate the different liver disease phenotypes. We have healthy control, black points here. We have chronic Hep B. We have chronic Hep B plus fibrosis. We have cirrhosis. And these results were verified in an independent cohort of 339 patients with chronic Hep B with liver fibrosis, cirrhosis, and HCC. And one of the striking findings is that the blood bile acid levels are substantially elevated in patients. Some of the bile acids, especially when they are conjugated with taurine or glycine, are increased by a factor of 100 or 175, in this case 156, compared to the healthy controls, which suggests that these high levels of blood and hepatic bile acids may not only be a result of altered gut microbiome and liver function, they may also contribute to the pathological development of liver diseases, given the cytotoxicity of these bile acids in nature. So we conducted an animal study to find out if there is such a mechanism The animal model that we developed is STEM mice model, where we inject a very low dose streptozotocin into the baby mice shortly after their birth. And then after 4 weeks, we feed them with a high-fat diet, and at 5 weeks, immediately, 1 week after, the fatty liver phenotype shows up, and at 7 weeks, the NASH becomes evident, and 9 weeks, fibrosis phenotype becomes evident, and 12 weeks, nodule evident, and 16 weeks, the HCC has been developed. So by the time we sacrifice all the animals at 20 weeks, all the male mice developed HCC. So this is a very nice animal model in that it rapidly produces the liver cancer phenotype, and also it gives a very nice window of different stages of liver diseases to allow us to take samples at different time points to find out what changes are involved, including the gut microbial alterations. So compared with the controls, the STZ, the high-fat diet group, exhibited significantly lower diversity based on the number of observed species. And we also observed the extensive inter-species interactions and strong co-exclusion and co-occurrence relationship between the phylotypes. And the correlation study suggests that altered microbiota at different stages of liver disease is significantly correlated to hepatic and fecal levels of bile acids. So we also see the significant increases in the abundance of gram-negative bacteria, such as Bacteroides spp, Bacteroides vulgatus, and Desulfovibrio spp, leading to the increased levels of LPS in plasma, liver, and stools. So in the liver of the model mice, we found that key bile acid transporters and a number of metabolic enzymes were significantly inhibited. One of the major transporter called BSEP, which is responsible for transporting the hepatic bile acid into the bile, into the gallbladder, is significantly inhibited. And when you can imagine that when this major bile acid pump is shut down, we are going to see increased intrahepatic bile acid concentrations, which in turn would potentially promote the progression of liver diseases. And one critical step towards the development of liver fibrosis is actually the activation of hepatic stellate cells. We observed that bile acids, particularly those taurine or glycine conjugated secondary bile acid, can readily activate the hepatic stellate cells. And these microbial metabolized bile acids, when conjugated with glycine or taurine, have the most significant effect of activating liver fibrosis and promoting HSC proliferation. So another interesting finding is while all the male mice successfully developed the liver cancer phenotype, almost none of the female mice developed liver cancer. When I say almost, I think 1 out of the 10 female mice developed a tiny liver tumor here. So it is significantly decreased in terms of the formation of the liver cancer. So what is the reason behind this? So we found that there were significant differences in gut microbiota, which are correlated to bile acid metabolism, BSH activity, between normal male and female mice. And such differences were amplified when mice of both sexes were exposed to STZ and high-fat diet. And as a result, the hepatic bile acids are significantly increased in male mice, as we can see here, compared to the female mice upon the STZ and high-fat diet intervention. So we pretty much concluded that it was the increased levels of secondary conjugated bile acids accumulated in liver that caused the problem. So we decided to try to remove these or reduce the production of intestinal bile acid by using an ion exchange resin called cholestyramine. And to our surprise, the therapeutic effect of this treatment is very good, that none of the treatment group has developed liver cancer. And of course, the increased levels of bile acid in liver were attenuated after the resin treatment, and relative abundances of significantly altered bacterial genera, such as Clostridium and Desulfovibrio, etc., were normalized after the treatment. So the clear, we are getting a kind of a clear picture now. So the high glucose and high fat diet would immediately hit the gut microbiome, causing the microbial compositional change, leading to the increased production of secondary bile acids, DCA and LCA, which in turn to circulate back to liver and conjugated with glycine or taurine to induce the hepatic stellate cells, and then to develop, to promote the development of liver fibrosis, which then leads to further progression of liver disease such as cirrhosis and liver cancer. So we thought this is a novel strategy to study the disease mechanism involving organ-organ interactions, but I realized that this is also a challenge in such an omics-driven mechanistic research that often involves a large number of factors. We have microbes here, we have a large panel of metabolites to identify. We also have multiple protein targets involved in a mechanistic pathway that are all co-located in multiple organs and tissues. So that is technologically challenging for a research group of average size to completely figure out what the picture is. So with that, I will stop here by acknowledging my group members and collaborators, and more importantly, the NIH grant support. Thank you.
Thank you. And our next speaker is Dr. Cindy Sears from Johns Hopkins University, and she's going to talk about biofilms as a risk factor for human colon cancer.
I too want to thank Liz and Lita and the organizing committee for the chance to speak at this very thought-provoking meeting. I think you're going to find what I say builds on concepts introduced by Harris Wang yesterday, and then Andrew Gurwitz and Justin Sonnenberg today. And I too want to thank the NIH. I have a career because of support from the NIH, and most of what I'm going to present has been supported by the NIH. So what I'm going to do is tell you why study colon cancer in one slide, and then I'm going to talk to you about biofilms as we've observed in sporadic colon cancer, tell you a little bit about biofilms in hereditary colon cancer, sum up, and then tell you what gap I would suggest be worked on. So the hypothesis and goal of this work is the thought that by defining the microbial drivers of colon carcinogenesis, we can design new approaches to the prevention of human colon cancer. And we would argue that that's important, and there are several domestic health reasons why we think that's important. So when diagnosed early, colorectal cancer, which is a leading cause of cancer death in men and women, is a completely preventable disease. Yet there will be about 135,000 new cases in the US this year, and in a rather alarming report published by the NCI in February, it appears that there are now rising rates in both men and women under the age of 50 years of age of colorectal cancer. If we wish to work on healthcare disparities, and indeed we do, the highest incidence and death rates for colon cancer in blacks in the United States. We would also argue that this is important from a global health perspective. in that colon cancer rates are rising globally with a projected 80% increase by 2035, when it's anticipated that we may see 2.4 million cases of colon cancer around the globe. And if we think about global economies, not everybody on the globe is going to get a screening colonoscopy at the age of 50 or younger. And so we need new approaches to try to identify those who are at risk. So the microbial colon cancer field tends to toggle between two extremes. One, that single species may play a role in the initiation and progression of colon cancer. There are several organisms that have been targeted. I show pictures from the mouse colon of mice colonized with enterotoxigenic Bacteroides fragilis, an organism we worked on, as an example where you can show that you add a single organism to that complex microbiota, and indeed in a susceptible mouse model you get a dramatic change in the biology. But there are similar, although less robust data for the PKS-producing E. coli or the PKS island-containing E. coli, Fusobacterium nucleatum, among other Streptococcus galolyticus and Enterococcus fecalis. On the other end of the spectrum is the idea that there's something about the complex microbiota that is able to be procarcinogenic in the colon. And it's this end of the spectrum that I'm only going to talk about today, so focusing on the power of community to be oncogenic in the colon. So you understand the pictures that I'm going to show you. I want you to understand how we've gotten the samples that we've worked on. So it's taken a large group of people in coordination of a large group, but what we do is we go to the pathology suite and with the permission of the pathologist, take some of the surface of the tumors, and then we take some of the normal colon mucosa as far away from the tumor as we can get in that particular resection. So those are the cancer-related specimens, and they're studied as a pair. In the colonoscopy suite, individuals undergoing screening colonoscopy, we've gotten random biopsies from either the right colon or the left colon of normal mucosa. So this is sort of the bottom line of what we saw in work initiated by Christine De Gea and carried on by Julia Drews. This is the sum of samples from both Johns Hopkins and a second cohort that was studied in exactly the same way in Kuala Lumpur, Malaysia, with Dr. Jamuna Valdivelu at the University of Malaya. And all the red dots are biofilm-covered tumors and flanking normals, and the blue dots don't have a biofilm. And what we initially learned at Hopkins, much to our surprise, was that essentially every tumor proximal to the hepatic flexure had these complex communities. What we learned in Malaysia was that the one difference between the Malaysian population and the US population was that there was an increase in left-sided tumors with biofilms in Malaysia, such that we recognize in the sigmoid area this appears to occur. So when biofilms are present, both the cancer and the normal tissues taken as pairs are nearly always concordant for biofilms. And doing this requires that in the pathology suite, we're putting the tissues into a Carnoy's fixative, which fixes the spatial organization of the mucus and the microbiota relevant to the mucosa. So what do I mean by a biofilm? This is a cartoon from Gunnar Hansen, and here is the dense inner mucus layer that coats the entire length of the colon and is largely impenetrable bacteria. A few get across, but for the most part, it excludes bacteria. And above that is a looser mucus layer that has lots of bacteria in it, right adjacent to this layer. And then, of course, there's the lumen. So when I say a biofilm, what it means is that we detected bacteria in the mucus layer at a very high density and going at least 200 microns along the length of the colon. And in addition, in that mucus layer, they're of course swathed in both their polysaccharide capsules as well as glycans within the mucus. So when a fluorescent bacterial, all bacterial probe is used, this is what a biofilm positive tumor looks like. So DAPI is staining the nucleus of the cells, and there you can see this dense layer of bacteria is right adjacent to the epithelial cells. If you go up to high power, you can see the individual bacteria. And here's this tumor is flanking normal, where you see exactly the same relationship. And you don't see that in these tumors from the left colon, where you can visualize the empty mucus layer here. Now, this is confirmed by scanning electron microscopy, shown here in biofilm positives, but not seen in a sample that was thought to be biofilm negative by the microscopy approach. We went on to use about a dozen different bacterial probes, different groups, and even down to the species level, and determined that in sporadic colon cancer, these were all polymicrobial. And a feature that we picked up in more detail on was that a portion of this community invades every tumor that is biofilm positive. Now, if we turn to those individuals undergoing screening colonoscopy, do exactly the same types of analyses, what we learned was that about 15% of individuals undergoing screening colonoscopy will have similar polymicrobial biofilms right on top of their epithelial cells, but they don't tend to be as thick, really, as those in the cancer situation. Oh, and the other thing I wanted to say was, in this case, the biofilms are random throughout the colon. So this geography on the right side is seen in the cancer patient, but in the healthy patient, they're actually randomly distributed, and they're equivalently present in the right and left colon at that point in time. So as Justin Sonnenberg said, we're looking at the spatial organization of the microbiota in this setting, or the biogeography would be another word. Now, they come in 3 microbial flavors, if you will. The most common is those that are dominated by Bacteroides and Lactobacillus, as shown here. There's about 30% that are also have Bacteroides lactobacillus, and then embedded in there are blooms of Fusobacterium. And the third type, which is rare,
4 hours is
that are dominated by Proteobacteria, particularly Enterobacteriaceae. And I can't tell you much more about that at this point, but it's certainly a focus going forward, trying to unravel what that means. Now, I don't have time to show you the primary data, but this is a summary of how we put the data together as we pursued the hypothesis that biofilms in healthy individuals would increase the risk of developing adenomas or colon cancer. So the data suggested that biofilm formation changed barrier function as we saw changes or loss in E. cadherin. There was immune activation within the mucosa as detected by the pro-oncogenic cytokine IL-6, a well-known activator of a transcriptional factor that is highlighted in cancer research, STAT3. And in addition, working with Gary Zuzdak and Carolyn Johnson at the Scripps Institute, we identified a rare polyamine metabolite as increased in the setting of biofilms, an N1-N12 diacetyl spermine molecule. And the data suggested, this question came up yesterday, the data suggested that this was being made both by the bacterial biofilm and by the epithelial cells. These factors were associated with clonic epithelial cell proliferation, and this is taken from a left colonoscopy control biopsy. Just to show you an example, this is Ki-67 staining as a measure of proliferation, and you can see that there was increased proliferation in a biofilm positive compared to a biofilm negative sample, which was supported by the aggregate data. And it's to remind me to say that any biology we've seen on a right sample has been the same on the left-sided sample. So now, to try to extend this work to understand the epidemiology and significance of biofilms in healthy individuals, with the support of the NCI, We're in the midst of a 2,000-person longitudinal colonoscopy study. We've managed to enroll about 600 patients in the first year, and as consistent with some of the discussions about longitudinal studies yesterday, we estimate we'll end up with about 50,000 samples in the freezer to do additional work to try to understand the relationships between biofilms and human biology. Now, a question, of course, is, are colon biofilms carcinogenic? And to try to answer that question, we're collaborating with Christian Jobin at the University of Florida in their germ-free facility. And what we did was we created an inoculum of biofilm-positive tissues, either tumor, flanking normal, or the colonoscopy biopsies. And then as a negative control, or a negative sample, the biofilm negative colonoscopy biopsies. And these were inoculated into mice that are APC heterozygous and are therefore prone to get colon tumors. And we use MIN, or called multiple intestinal neoplasia mice, or MIN IL-10 knockout mice. And what we saw, the aggregate data are here, the biofilm positive samples, but not the biofilm negative samples, induced tumors in the colons of these mice. The type of mouse didn't matter to the tumor result. And the surprise was that each of the biofilm positive samples were approximately as oncogenic, which was a surprise in that these thinner biofilms in the healthy host were as carcinogenic as those tumors in the cancer host. So we view this as a great opportunity to be microbial sleuths, to try to understand what within those communities actually is contributing to oncogenesis in the colon. This is just one slide, a snapshot of our data from hereditary colon cancer. This is data from the syndrome known as familial adenomatous polyposis, which are APC heterozygous individuals. These individuals will get polyps in their colon and they will get colon cancer. And so they eventually all lose their colons, which are removed to prevent colon cancer. Over about 6 years, we had the opportunity to collect samples from 4 colectomies, as shown here. The red dots, again, are biofilm positive, the blue are not. And you can see that throughout the axis of the colon, in each patient, there were biofilms detected. In addition, I'm just showing you the species-specific probes here because much to our surprise, the biofilms differed from what we saw in sporadic colon cancer, in that you see there's a lot of red. There was E. coli, and then there was Bacteroides fragilis, with E. coli being dominant in each setting. Similar to sporadic colon cancer, we saw invasion of the bacteria into the tissue. And when these biofilms were cut out and PCR done, we were able to detect the colipactin gene, that is the virulence factor of the E. coli, that are associated with carcinogenesis, and the BFT gene, which is a toxin gene associated with the tumors that I showed you with ETBF. And lastly, we were able to go to Bert Vogelstein's bank of samples, which is 25 years in the making, and identify another 25 patients and then controls. And using now microbiology, we're able to recover PKS E. coli and ETBF with about half the samples having both organisms, and overall more common in the tumor patients than in the controls. So how do we put this together? Well, we know actually that ETBF and the PKS E. coli commonly colonize children before the age of 5. We know much less of what happens in the epidemiology of those organisms subsequently, although a small amount of data supports that at least some teenagers and young adults remain colonized. As has been discussed extensively here, there are many things that modify our microbiome over time. And so we might suggest that there may be host bug collaborations occurring, and we have mouse data that suggests that these two organisms are synergistic in their capacity to form colon tumors. We would further suggest that biofilms may develop over time, such that in late sporadic colon cancer, we see biofilms in about half of the tumors. So my gap to be addressed is not novel. It has been already discussed here, and that is defining causality or not of the microbiome in human disease. And specifically in the setting of colon tumors, does biofilm formation or the presence of any specific microbe or community consistently precede development of colon polyposis in humans? This is, of course, a challenge because we know that the time from the clonal expansion of that cancer cell in the colon to the visualization of a polyp is at least 10 years and maybe up to 40 years. So these chronic, slow diseases, many of which are being discussed here, pose a particular challenge to trying to link the microbiome to disease. So thank you very much. And oh, I terribly forgot to, I showed you pictures of who did the work over time, but I really do want to thank the entire lab and all of our collaborators, as well as, again, the NIH, which has provided most of the support for this. So thank you very much.
Okay, our last speaker is Dr. Gary Hufnagel from the University of Michigan, and he's going to talk about the role of the lung microbiome in respiratory health and disease.
Okay, cool. You can hear. So first, I'd like to thank Lise and Lita for the invitation to come speak today, and so also to represent the unrepresented mucosal site in the microbiome, which is the lung. Okay. So this statement in this book and many similar to this has not only kept me up many nights. Seen my grants sometimes go down in flames, and driven my research for the past 7 years, which, the question is that a cause and effect relationship? That's a different story. But the other hat that I wear at the University of Michigan, besides being a researcher, is I now teach introductory microbiology to the undergraduate campus. So I have 200 students in a Roman Colosseum style thing. And if there's one message that I want them to take away from that course after an entire semester, it's this, which is that there's virtually no environmental niche on the earth that's so extreme that microbial communities cannot be found. So it really does beg the question for that statement, why did we ever think that a warm, moist environment that is just a few centimeters away, over which air continuously flows, would actually be completely free of microbes, would be sterile? So that would be a whole nother long discussion, and I'll show you the data and things like that. But if you're going to believe me at that stage, then let me take it one step further. While the lungs are not sterile, they are not a great environment when you're healthy for microbes to grow, and especially for colonization by mucosal anaerobes, gram-positive bacteria, and others. And why? Well, first off, if you're an anaerobe in the mouth and you get whipped out and thrown into the lungs, it's an aerobic environment. So you're not a happy camper that way. But there's also cilia that is constantly pushing the upper airway mucus back up, so you have to fight that. We don't think about it, but we think about the body temperature being 37 degrees. Turns out that the upper part of the lower airways is actually not 37 degrees. It's actually lower temperature. So it takes a number of bifurcations before finally the wall hits 37 degrees. So you've got then lower temperatures up in your trachea and the first few bronchi. And we can't dismiss surfactant. So surfactant is a lipid, is a detergent. So you have microbes coming from a mucus-rich environment being thrown into a lipid-rich environment, and especially if you're a gram-positive organism, it's just going to blow a hole in you. And finally, there's very little mucin There's food, very little food down the alveoli. And we also don't think about the fact that the lungs are the only place in the body in which there is always a leukocyte that's outside the external surfaces. It is a patroller. It is the alveolar macrophage. It constantly patrols that that external surface where things are coming down. In other words, it's a predator. So if we use ecological models, it's like having a protozoa. So you have bacterial-protozoa interactions, essentially. So not a great place when you're healthy for organisms that are being seeded from the upper part of the airways to come down. So thus, the lung is a low microbial biomass site when you're healthy. Okay, so I want to point out now, I think you would typically learn this in Chemistry 101, the idea between accuracy and precision. Okay, and so remember that precision is you can make the same measurement again and again and again. Accuracy is, are you actually correct? Okay, and so when we talk about things being below signal in an assay for a microbe, that is not the same thing as being sterile. Okay, just because you can measure it many times and say, I don't see it, doesn't mean that it's not necessarily there. And why would that be? We've heard a number of presentations at this meeting about technical limitations of our assays, reagent issues. Again, I could go on for a long period of time and bring Rick and Ron up here to talk about reagent and kit noise, especially when you're dealing with low biomass samples. Are you using the correct assay, et cetera, et cetera? So I bring this up because there's a paper that just came out 2 months ago or a month ago that actually claimed that mice, their lung microbiome, that a portion of them have a sterile lung microbiome. So I just want to point that out, that it's not sterile, it's below the limit of detection of the assays. And by the way, yes, we see the same thing in mice also. And I will get to that at the end of the talk. Mice actually have an even lower biomass when healthy in their lungs than humans do. So a major challenge for this field is the study of the lung microbiome has been and always will be the fact that the lower airway sampling is highly invasive and not amenable to serial sampling over time. And so this whole idea of, the whole elephant cartoon we've seen come up many times is really, really, really relevant to the study of the lung microbiome. Because there are many ways that we can look at the lung microbiome between bronchoalveolar lavage, bronchoscopic brushes, biopsy, sputum, and postmortem tissue. All of them give us information. Not a single one of them gives us everything. Okay? And so it's the collection of putting them together that then give you the big picture of what's going on. So let me give you an example of one of those sampling techniques. So this is bronchoalveolar lavage. And so this is a study that we had that was supported by the NHLBI. It's 28 subjects, and we took samples from the nose, the mouth, a gastric aspirate, and also a bronchoalveolar lavage, and analyzed the microbiome. And so this is an ordination. If we plot all the samples on here, and the very first thing you can see, which is exactly what was described in the HMP, is that the nose is indeed distinct from the mouth. The gastric aspirate is very similar to the mouth, which makes perfect sense, saliva flow, okay, from the way that we were sampling this. If you impose on top of that then our bronchoalveolar lavage samples, what you'll see is that they are indeed distinct. Statistically, we can show that these samples are distinct from the oral samples, the gastric samples, and the nasal samples, but you can see that it's not actually separating until you get to this second component here. So it fits the model of the lung microbiome being a target community whose source is coming from the upper part of the airways. But let's dig a little deeper into this data, actually, and it tells you something also a little bit more about human variability in the lung microbiome, which is if we actually look at similarities and dissimilarities between the oral communities of an individual compared to what we get from the BAL. So each individual, we can use them as their own control. What we'll see here is that there are some individuals in which the BAL community is much more similar, in fact, look at this one down here, to what you get out of their mouth, and others in which it's very distinct. If you go over to the nose as potentially a source community as air flows through, you'll see most individuals, there's no similarity, but indeed out of our 28, we've got, 8 there? That you can see some similarity to it. So there is this variability between what we're actually looking at in the lungs. So this is the type of charts that we've seen in all the microbiome reviews, and so I just throw this up here because now we can actually put a little pie chart up there for the lung. So we've known for a long time that the microbial communities, let me put it this way, when you have respiratory disease, you quite often can isolate bacteria from it. What we've learned over the past, 5 to 10 years is that the microbiome that we can measure by culture-independent analyses differs during disease states compared to what we see in healthies. But the note that I put up here is that the magnitude of these changes that we're seeing here has not historically been captured by standard culture techniques. And that's been kind of the shock. It's like, wow, like there is more difference during disease than what we would have expected based upon what we can culture. And I want to give you one example of this that we've been following, which is that if we look by culture-independent analysis at the pseudomonads, and so these are operational taxonomic units here, so we've got 6 different ones. All of these can be taxonomically identified as pseudomonads. We actually get a diversity of pseudomonads that we see in the human lung. Not a whole lot when you're healthy, but definitely during disease states. So these are lung transplant recipients. They're both symptomatic and asymptomatic for subsequent disease. These are bronchoalveolar lavages from interstitial pulmonary fibrosis patients. These are brushes from chronic obstructive pulmonary disease. And you see a number of different types of pseudomonads. One of these is aeruginosa. The other 2 are not. So you can throw out there, well, it might be kit contamination or some environmental sort of thing, and that was definitely something we dealt with. I don't have time to go into it here, but I can tell you that these indeed are real. It's just that we're not finding the right way to culture them, and we recently have identified a way to actually culture these out. And it's actually, again, telling us more about what that environment looks like that these organisms are flying into. And so what has surprised me is that unlike studies of the GI tract microbiome, culture-dependent analyses of the airways have not identified significant numbers of unculturable bacteria. And what I mean by that is I expected to find all these new bugs and things like that. It's still the same ones we've known for years. However, we're not growing them out all the time. And so, and if you actually go back to the literature, you see this all through discussion of respiratory diseases, of chronic colonization by, for example, Pseudomonas aeruginosa, and then it disappears. But we can find it by PCR. Oh, then it reappears. So what's going on? And so what we think it means is that there's a change in the in vitro conditions required to culture these, and so it reflects a change in the metabolic activity of the bacteria, a change in the nutritional environment in the lungs, and a subversion of host defenses. So our context with which we're looking at all this is the following model. That we believe that the constitution of the human lung microbiome is determined by 3 factors. The first of which is immigration, so organisms coming in. And we think the main thing when you're healthy is microaspiration. But you could clearly have inhalation of bacteria, you could have sort of just direct mucosal spread, and obviously during disease, you can also have blood and lymphatic immigration. That is balanced by microbial elimination, so cough, mucociliary clearance, innate and adaptive host responses. And so I want to point out for that last one, for innate host defense, I've just completed a study. We looked at 40 inbred mice, different cages, different vendors, a lot of information about that. But the one thing I want to point out to you is this following graph, which is that these are completely healthy mice. And if we actually look at the diversity of the microbiome in their lungs and we compare it to the concentrations of IL-1 alpha in their bronchoalveolar lavage fluid, we get a wonderful correlation. And it's actually really stands out if I put quadrants on here. And if you look down here, this is basically saying that when the community becomes less diverse, we no longer have low levels of IL-1 alpha. In fact, the only thing we ever find are higher levels of IL-1 alpha. IL-1 alpha is of interest because it's a preformed cytokine. It is a very fast-released cytokine. And so our interpretation of this is this fits a model that when something actually tries to colonize, it is pushed out quickly by innate immunity. And in fact, this diversity of probably what's coming down from above is actually the normal state. That was in mice. This fits perfectly with what the NYU group led by Leo Segal had found, that with the human lung microbiome, that when you start to get the domination by organisms, you also then get these low-level inflammatory cytokines being made in healthy individuals. So the other part then of the model is that all this, once you have this balance between immigration and elimination, once a microbe gets down there, the regional growth conditions determine what you're actually going to see down there. And so in relevance to this session, what are the mechanisms we need to be looking at? Well, one of the things that we've been looking at, and others have also been looking at, but borrowing from the gut research, especially a number of things that are pointed out here, is inflammation in and of itself changes this environment. And so you can see the emergence of pathogenic strains from otherwise non-pathogenic bacterial epithelial biofilms, or potentially the outgrowth of otherwise transient microbes. You have host-derived factors and metabolites that we think are being generated by the inflammatory response. They can include catecholamines, reactive oxygen species, potentially tryptophan metabolites. And so this results ultimately in shifts in microbial physiology, and I will argue can also result in changes in culturability. Why is culturability of interest? Only because that's what we've been used to try to detect these organisms. So in the end, when we're healthy, it's mostly about immigration and elimination, because once you get down in the lungs, bacteria can't grow. But once you start to have lung disease, then the growth conditions in the lungs themselves change. So my final point that I want to make here, which involves some really high-tech animation, is to ask the question, when we talk about animal models and studying the human microbiome, does this lung ecology model then apply to other species? So if you think about humans and airflow coming in and then going down, well, what happens if we take our subject creature here, turn the airway sideways, change their breathing patterns? So instead of it mostly going through the mouth, they're obligate nasal breather, which means that the vast majority of the airflow goes across through the nose, which is an even lower microbial biomass site than the mouth. And then you add in an extended complex nasal terminate. What are you going to end up with? Lower immigration rates into the lungs. In other words, if you're going to use mice to study the lung microbiome, don't be surprised if you're not going to, quote unquote, find any bacteria in the lungs of healthy mice, because it's there, and we've got that data. It's just really low, because same thing. Same when we talk about pigs, because there's been studies out there about pigs in the airway microbiome. The model does fit. So, in summary then, the lungs during health are not free from bacteria. Rather, they harbor a low level of bacteria that are derived from the oral cavity and the nasal cavity. There's individual variation in the degree of similarity between the lung microbiome and the oral and nasal microbiome in the same individual. This lung microbiome during health, it's dynamic, represents a balance between influx, elimination, and growth. But we know that changes in the lung microbiome associate with respiratory disease. So the final point is then, what's the gap? And it's the gap we all are trying to solve here, but it's really acute in the study of the lung microbiome, which is, are changes in the lung microbiome the cause of airway inflammation, or the result of airway inflammation, or both? And so our goal as a research community is to move from these associative studies to mechanistic studies in human respiratory disease. And so with that, then, I'd like to thank my laboratory at the University of Michigan, plus a number of wonderful collaborators that have made all this work possible, and then, of course, my funding source, the NHLBI. So thank you very much.
Okay, thank you. And we have about 10 minutes for questions.
Hi, I have a question for Dr. Zia. So, in your model, you have been suggesting the hydrophobic secondary bile acids are responsible for NASH and the development of hepatic carcinoma in this mouse model. The question I have, the concern I have with this interpretation is that it's based on cholestyramine, which is very not specific. It binds to many hydrophobic molecules in the gut, in addition to secondary bile acids. In addition, cholestyramine treatment of mice changed the microbiota in a very major way. So the many molecules are expected to be changed in cholestyramine-treated mice. So you have any of the data that supports the model that secondary bile acids are responsible for NASH and hepatic cancer in this model?
This is a great question. I don't believe the hydrophobic secondary bile acids are causing the fatty liver and NASH. What I was saying is that these hydrophobic bile acids, when circulated back to liver, after they conjugated with glycine or taurine, they are more. They can activate the hepatic stellate cells, which induces the liver fibrosis. And that's probably a major mechanism for the bile acid-induced liver disease. So the cholestyramine treatment, removing or reducing the intestinal levels of secondary bile acids, would really help the elimination of the hepatic bile acids, especially those secondary bile acids. So that pretty much stopped the liver fibrosis process, I believe. It's not stopping the NASH and fatty liver because we feed the mice with the high-fat diet. So it wouldn't be surprising that the liver will become, steatosis.
But the secondary bile acids, the enzymes are enzymes to remove through PXR and other receptors use enzymes in the liver, in the hepatocytes, to remove this toxicity from secondary bile acids after they are conjugated. So I think this is more complicated.
That's another way to do it, by changing the activity of some of the transporters. You accelerate the bile acid circulation, and you can also reduce the intrahepatic levels of bile acids that way. Yeah.
Thank you.
My question is for Dr. Sears, Yiping Han from Columbia University. Nice talk, Cindy. My question is, in your germ-free mice study, when you inoculate a human biopsy, have you looked what kind of bacteria do you recover from those mouse tumors?
We're just starting that work, so I don't really have any comments. Christian did sequence them and found similarities across the samples that induced tumorigenesis, but the correlation work and culture work hasn't been done as yet.
And also, do you see a difference in the potency of the biopsy to generate tumor, whether it's what kind of a biofilm from the host?
So the inoculum was actually a composite of 5 patients, and we put some patients with each type of biofilm in the tumor inoculum. Now, in the healthy patients, we only see one phenotype, and that is the polymicrobial. We don't detect any fusobacterium in those, and there's no proteobacteria dominance that we've seen to date. So they're predominantly in the healthy host Bacteroides and Lachnospiraceae, we think. But again, we're just developing a strategy as part of our prospective colonoscopy study to try to get at this in a little more detail.
Thank you.
Sure.
Hi, Phil Dashner, NCI. Quick question also for Cindy. Given what we heard this morning from Dr. Ghanem on the fungi in biofilms, have you had have you looked for any fungi in the biofilms that are tumor associated?
No, we have not specifically used probes that would pick that up, but it's a good suggestion.
So it's only 16S that you've used?
Exactly. We've only used 16S to date. Now we're trying to design a study to do in parallel 16S metagenomics, RNA-seq, to try to look across the spectrum. And in that setting, we should pick it up, I believe, if it's there as well.
Okay, if no more questions, then thank you very much.
Okay, so I'm going to just kick off this roundtable discussion with just an introduction to who I am. I'm a program officer in the Division of Microbiology and Infectious Diseases in the National Institutes of Allergy and Infectious Diseases. My name is Ryan Ronaldo, and I'll be guiding you through this next 30 minutes. Hopefully we'll have both a series of conversations and as well as discussion. So one of the things that I definitely wanted to do is thank everybody, certainly the speakers. I know I see many of your faces, and I've seen you here for the numerous workshops that we've had over the years, and I feel like some of you guys need an office here. You're here so often, but nonetheless, so when Lita, a year ago, whenever she started planning this meeting, one of the things I asked her, I said, what's the purpose? And that's really outlined, and the speakers have done a great job of identifying gaps, technical hurdles, and new opportunities that potentially would lead to future interventions, and honestly, new approaches. So those 4 things are what I was focusing on for this. So I'm happy to have a Q&A and have an extended Q&A, but I'm working for Lita today and now, and again, so that's my goal, is to focus on those 4 things. And as I said, the speakers have done a great job. So one of the things that I thought would be an interesting idea, rather than perhaps the speakers focus on their own challenges or their own things that they brought during their presentation, perhaps to think about what other speakers have highlighted and to maybe echo those comments. I think that might be an effective way to both get questions and to maybe help us, certainly us as program officers and administrators, thinking towards the future as HMP winds down and as the institutes contemplate their activities in this area. So I tried to generate just a brief list, just to jog everybody's memory about maybe some gaps. And so some of the gaps that I've heard perhaps repeated is really, we had a significant focus on mechanism and then determination of causality. Some gaps in terms of annotation of metabolites and the need to go back and look at those annotations. I think we had 2 wonderful talks on the inclusion of the other aspects of the microbiota other than bacteria, so phage, viruses, and fungi as a gap. And I would challenge everybody at least to say, why are you still ignoring them? It's just the question is why is that? The other thing that I heard in terms of a gap is the generalizability. of findings and model systems. So some technical hurdles I wrote down. I thought maybe that it was interesting to look at validating results in a different lab. The comment of novelty over rigor, is that a cost issue? This is a big technical hurdle. What's in food? I heard that a lot this afternoon, and you know, is that a key challenge? And then some new approaches: spatial biogeography, looking at biofilms, new model systems like fecal bioreactors or engineered tissues such as enteroids and organoids. You know, sampling the entire gut. Culturing the unculturable. So that's what I had listed. As I said, I'd like to hear the speakers talk to each other, perhaps, initially. If that works, we'll go with that. Otherwise, we're happy to take questions and answers. So I'll look at the speakers and see if they have any thoughts on what I had just said, and perhaps maybe echoing that.
I guess I could say, this is Andy Goodman, I could start. So, to be controversial, I'm not sure that we all need to look at everything. And I think this theme of that viruses and fungi and eukaryote, other eukaryotes are in the microbiome is certainly a fact. But I don't know necessarily that the most effective way to move forward is to have everybody look at everything all the time. And another model would be, yes, we all agree that these things are all there, but that certain labs would focus their expertise in certain areas. And so I don't know if people have other comments about that, about whether we should all be looking at all of these things, or if another acceptable way forward is to not do that.
I just have a comment about that, just based on the studies that we've done in the mice. I mean, we had this crazy phenotype in mice, and we could have done, a microbiota analysis and shown that there were certain bacteria that were up or down in this model. But what we found, it was a parasite that was inducing that effect. And I think if we would have, if you ignore that and focused on the bacterial side of things, we would have made different conclusions. So I think I agree with you. You can't be the expert in everything, but I think you also cannot ignore the contributions of other kingdoms to the effect that you're looking at. Again, that's probably most model specific, but.
Andy, I really like that question because it really speaks to this issue of whether there's groupthink, whether there's some sort of prescribed orthodoxy or religion around an analysis. The analysis should be informed by the biological question, the biological system. And if there's novelty in analysis, it should be driven by the questions. I hope that the next phase of microbiome research is directed at 1,000 garages rather than a few tall towers of thinking. That was one of the challenges and problems with the first HMP project, limited exploration of diverse populations and systems. It wasn't that it was an error in thinking, it was just the way it began. But I do worry about some sort of prescription in the way people do science. But I also think that subsumed under your question is, what needs to be co-evolved in the field, and what are the conditions for innovation? And I think the conditions for innovation in an interdisciplinary field like the microbiome, because it really does bring together, we heard Nicole's talk, we heard your talk, you combine structural biology and genetics in very elegant ways, and other people's talk, is what kind of learning environments do we have to create for students in this area, where you can encourage people to come together within academic units where there's enough support, kindness, and generosity so people aren't afraid to say, I don't understand, and you can teach one another. But also where you bring together the capacity to apply really rigorous statistical and computational tools together with whatever other biochemical and metabolomic, et cetera. So I really worry about the education side and how we're missing an opportunity to to ensure our future in that sense. And the last part of at least my response to my gut response, pun intended, to your point is that there was a lot of appropriate plaintive and not so plaintive pleas for better understanding of gene function. That comes back to something that you not complained about in the Portnoying sense, but rather we don't have genetic tools to deal with a lot of these organisms. And that's a shame. It's a shame because it's hard. It's a shame because there's a lot of innovation in terms of gene manipulations with CRISPR-Cas, et cetera. And if we reserve that investment now for the future, I think we're really going to be handicapped. So know your targets, look at mechanism, but be able to manipulate the system in whatever ways you think are wise.
I also wanted to speak to what you said. I think that we do better to actually sort of combine what you both said, which is we do better to develop serious expertise in our area and then bring that expertise to collaboration rather than all of us trying to do everything in our system.
And so I would add one other thing to that, which is then also the need to continue to develop an expertise or support individuals who are indeed the jack of all trades. Because if everyone has their areas of expertise, which we absolutely need, especially in an interdisciplinary field with that, then it's also really helpful to have people whose research is just constantly moving about back and forth across that to stimulate ideas and to basically help sew all the parts together.
You know, thinking about sort of a broad landscape versus being somewhat more focused, you know, I think there is sort of a divide between the basic science world, where we have wonderful technologies, really understanding mechanisms and clinical relevance of disease pathogenesis. And I think that actually Justin and I were at a meeting recently at NIH, and we had a conversation with one of the program directors, and the conversation was sort of oriented towards, you know, having a clinician that really understands what's really relevant in disease. What are the biggest gaps and what are the biggest struggles that physicians and surgeons have in the treatment of their patients? And then having a conversation with basic scientists who have the technologies and the physiologic and mechanistic understanding, sort of bridging that gap. I think having that real deep conversation might help the scientific field focus on things that maybe are a little bit more low-hanging fruit, or the things that are the most the highest priorities that the scientific field can use their best technologies to try to address.
Bill Joss, Scripps. We've heard a couple people talk about reproducibility, and in the metabolism, this is more for mouse research, in the metabolism and aging field, there's been a lot of debate about housing and diet. For example, temperature, whether animals should be housed at thermoneutral versus the more common room temperature, which is cold for the mice. I was wondering if this debate has moved on to the microbiome field and whether temperature has been studied in the context of microbiome research. The other question, or the other parameter, is diet. We've heard a lot about standard chow, and we saw the picture of the beautifully dissected gut and what the healthy gut looks like. My colleagues, though, seem to always complain about standard chow in mouse studies. And so I'm just wondering, how do we know, or what evidence do we have that standard chow is actually an optimal or great diet? And the only specific question I have in that context is, what does the gut look like in a wild-caught mouse, And when you take that wild-caught mouse and put it on standard chow for some amount of time or multiple generations, does that gut still look good or different or not?
I guess I'll just make a comment because there's this silence here. So I had a conversation with somebody out in the audience during one of the breaks, and this gets to the question of whether or not you use a defined diet that's controlled, whether or not you're going to study wild-type mice, like mice in the field, right? Because they're going to be more physiologic than the inbred mice that we use in the laboratory. And my comment was, I'm not sure that that's the right way to go because, as we sort of discussed, humans are exceptionally noisy. There's a lot of intersubject variability. And there's a reason why we study mice and we study notobotic mice, and I study mice, inbred mice, and that is because we can lower the noise level so we can actually see a signal. So if you wanted to do mice out in the field and undefined diets, you're almost defeating the purpose of actually using these model systems because the noise will be so great that it'll be very difficult or very expensive or take a very long time to see some type of signal. I think the reason we use these defined model systems is actually see that signal, and then we can extrapolate and figure out, is that signal large enough, or is there some way that we could actually study this in human biology? But if you can't actually see the signal, then you actually can't take the first step from a mechanistic standpoint.
I think to follow up on that, though, at the same time, finding what that standard diet is on which you're going to add something and look at the effect is, you have to keep in mind that if you start with, say, a high-fat diet, your chance of maybe impacting that with a preventive component may be higher than if you start in what might be a more normal diet. And in that context, unless you go back and do those additional studies, you're under the impression that your dietary supplement or your phytochemical of interest is going to be a great magic bullet, but in reality, under normal circumstances, it may have no effect at all. So I think fine to standardize, but to really understand what is your standard diet doing.
Just a quick comment about the standard diet. This was a significant topic of conversation at this earlier workshop this summer related to the microbiome and diet, and it was it's a complex subject. I think one of the things that we grapple with in the field is trying to maintain a community of microbes that's diverse and representative of a community that we might see in humans, and to do this with a defined diet that's very to do this in with a elemental diet or a diet that's constructed from the ground up is very difficult. I think Eric Martens has tried this with diverse fibers that he's put together and hasn't been able to reconstitute something that mimics the complexity found in like that list of ingredients that you saw today for a chow diet. So it's not as reproducible. There are a lot of ill-defined things in there, but I think it serves a purpose for a particularly large number of experiments.
I think it was pointed out today in the slide that the components of a mouse chow diet are ill-defined, and the sourcing changes over time from a vendor. So the lack of definition applies to the vendor as well as the recipient. I don't know that we'd ever be able to constrain sourcing from the vendor. I think that would be difficult. I do think it's helpful, however, to use germ-free animals fed a diet that you are applying to other colonized animals to see what the bioavailable contents are of that diet in the absence of microbes. So targeted mass spec for amino acids, certain micronutrients. The thing that is a huge gap that people were alluding to was the ability to define the molecular composition of food. And I do think there should be a campaign, and I know there has been individuals who have raised their hands and are starting to do this, not just in words but in deeds, to look at the macromolecular content of the major food staples in the world. 1,000 staples, different cultivars of a given staple. That'll start out primarily, I think, with attempts to look at glycans, but just even protein content and micronutrients. But what I was getting at is that it's unbelievable to me that the advances in nano-LC mass spec that were applied to breast milk haven't been advanced largely because glycobiologists are endangered species to plant-based glycans and even host-based glycans. And it's not just the monosaccharide and disaccharide content, it's the linkage analysis. And there are tools that are available now to start doing that work. And the NIH, with enthusiasm, with focus, and with nutrients, i.e. cash, should, I think, sponsor that campaign to be able to quantify the monosaccharide and linkage characteristics of major sources of glycans. So if we restrict the diet to one major standard chow, I think you're going to lose a lot of biology. But you have to know what you put in as well as what comes out. May the force be with us.
I just wanted to speak to the issue of temperature. I don't know if anybody's studied this or published this, but anecdotally, temperature is a very good point. Temperature is a serious issue, and temperature shifts cause profound shifts in the microbiome. So that probably should be addressed in a more systematic way. Or noting what the temperature is that you maintain your mice.
So you're talking about mouse chow and how, even though it's called standard mouse chow, at least there is some, I would argue, uniformity. And then you're talking about human studies where you do interventions with high fat, low fat, whatever. I would argue that there has to be some standardization there too in what you consider, and as Dr. Gordon said just now, that if the bioavailability of different grains is different, a high, even though you can say calorically it's high fat, it seems to me a bit of apples and oranges. And the difficulty for me as an editor is, if I get 2 papers, yes, we're mandating reporting of everything these days, but when science is a process of building on existing science, you've got to have a way to say something is common, some comparison, particularly in human studies. I think this came up, and I think Mike was trying to make a comment about this, at the diet session. So I would argue, even in human studies for diet interventions, some standardization. It doesn't have to be what's given to IBD patients or ulcerative colitis patients. Clearly, that's not palatable. But it'll be great if the community came together and said, if we say high-fat interventions for humans or high-fiber interventions, this is what we mean, and define it more precisely.
The fact that the microbiota transforms ingredients in foods forces us to have a higher resolution view of what the ingredients of foods are. The point I was trying to make is that I think it's important to be able to characterize the diets you put in. I only worry that there's just one diet used by everybody for all their experiments, because although that's a form of standardization, I'm not sure that the long-term consequences of that may be helpful or deleterious. But I do think it's absolutely critical to be able to define the ingredient. The autoclaving of chow for gnotobiotic experiments, and everybody knows how valuable your autoclave is, but it varies over time, and whatever settings there are. So, how is food transformed by processing? Either processing to create the raw chow and the processing that occurs when we irradiate it or autoclave it. So again, I would go back to the issue, at least in a nosebiotic setting, of trying to characterize what nutrients are available in a few regions of the gut in the absence of microbes as a way of determining the bioavailability to microbes in that mouse genetic setting in your vivarium at whatever temperature and at whatever time of year, and whoever's president will be.
So Vicki Sutherland, NTP. I was wondering, along with the diet discussion, if there's an idea of the best methodologies or procedures we would use to determine functional change. And what I mean by that is, so a standardization for diet, and what about where along the GI tract we should best measure to look for changes, as well as do we do fecal matter versus ileal scrapings, things along those do we think we're at the point where we can identify what are the best things to do to see whether or not there will be a functional change in the microbiome or in the animal?
I'd argue no. So I can tell you that as an immunologist, one of the areas that the entire microbiome field has largely ignored is actually the biology of the small intestine in terms of its microbiology. When you, even as you read reviews, it's like the GI tracts are, the intestine is just one long tube, but it's not. It has not only major featural differences, it even has small differences, and yet we haven't gotten to the point of actually placing microbial community structure, microbial metabolites on top of these specialized cells and structures at different points in the intestine that actually inform the rest of the immune system how to act. So I think a lot of what's the way things are driven is largely by cost effectiveness and reproducibility. But I think there's still a gap in terms of there's no one special site or special recommendation of how to do it. I think it's back to what Jeff had mentioned. I think it's your question, your biology has to drive how you do your sampling.
And I would say you have to understand your particular organism because they will each vary in how they affect various portions of the GI tract. And I think you should presume that until you prove otherwise. And I would also say I don't think the small intestine of the mouse is the place to study biology, except perhaps as a totally model system because mice are coprophagic. So if you're really interested in the biology of the small intestine, you are wedded to very difficult human studies.
There's a lot of enthusiasm and potentially hype around the impact of microbial communities on brain biology. And I think this is a very profound area of science that we should approach with sobriety. So the issue of regions of the brain that are analyzed and careful description of where you do the dissection is important. It's also important, and I use this as an excuse to bring up that topic, to invest money to determine how we can apply, at least to nosebiotic systems, standard procedures for looking at learning and other facets of behavior. How much can you import into a nosebiotic setting? What are the improvements that we can generate in being able to test physiological responses within a nosebiotic setting? One is learning. How can we monitor physiology over time with implanted sensors and transmitters to be able to capture data like, even things like state of oxygenation, activity of the animal, all sorts of things that we can think about. And I think that's an investment that could bring together bioengineers and this field in ways that would be very productive. A related and final comment I'll make, had to do with something that was brought up during the course of the day, which is what are people's definition of biogeography, and at what scale is the relationship between microbes and members of microbial consortia being quantified, and what kind of imaging procedures can we apply to this problem. And it's really daunting, but really important. I mentioned in my talk, Gary Borsi has spent a lot of time on that. Justin has made investments. It's really time for imagination and creativity. But if we don't determine, beyond the computational approach of co-occurrence, where you're taking a fecal sample and asking which organisms travel together, to understand the nature of the journey at a micron scale and to determine the degree of admixture is really going to, I think, be foundational knowledge to determine how microbes compete, collude, and collaborate with one another, whether those microbes are bacteria, whether those microbes are across different domains of life. So I would make a plea to the NIH at this moment in time to think about how you would sponsor initiatives, but sponsor it in a way that would evoke creativity, and again, sort of this idea of how much funding should be for garage-like behavior as opposed to tower behavior. Tower behavior is just a huge consortia trying to get together around the problem, which sometimes has very valuable outcomes, but also you want to have a degree of versatility and nimbleness and bring people together around these compelling problems.
Hi, my name is Abby Glasscock, and I'm from Virginia Commonwealth University. I'm just representing the Twitterverse today. So I have a question specifically for Dr. Gordon and Dr. Wu. Our Twitter person asks, have you considered antimicrobial residues in food, such as meat and milk products, and their impact on the diversity of the microbiome? In the US, levels of antimicrobial residues are legal in food animal tissues, and how do you think this impacts studies that are performed here in the United States?
I think there's an advantage to try to formulate human diets that are representative of those consumed by the target population that you're studying and apply them to some of the preclinical models. I think your question is a very good one, and are different sources of food ingredients, and is the food science that is applied to formulate those ingredients into diets affecting components of the microbiota, not just structurally but functionally. So I think it's possible to directly address that important question, and there are already the means and path forwards for doing so. Gary, what do you think?
Yeah, so we formally haven't studied it, but my notion is that the levels of antibiotics that are used in food may have a long-term effect. Short-term effects may be relatively modest to the degree that it may be difficult to actually visualize in some way. Although I do worry about the antibiotic resistance because that's sort of a long-term effect. We do, and it also really varies by the type of antibiotic. We found in both mouse studies, but also in human studies, that there's certain types of antibiotics we use even at pharmacologic levels that have a very minimal effect on composition. I don't know anything about the function, but the composition of the human microbiota. So again, it's the dose, the duration, and the type of antibiotic can lead to very specific effects. And so I do worry more about the antibiotic resistance and the long-term effects than just short-term effects.
As a decommissioned gastroenterologist, I'm at risk for logorrhea, so I'm going to try to avoid it. But I interpreted the question of the Twitter user as more general, which is to apply analytic techniques to food ingredients or diets, including mass spectrometry, prior to its administration to humans. And with that knowledge, can you do correlation analysis to see whether the responses of the community are related at all to some of the ingredients in the foods? And to do so in an unbiased way, as much as possible to create testable hypotheses that could then be carried forward to a notobotic animal. The whole issue is, what are the bioactive ingredients in foods? The raws, the food formulations after applying certain processing, and how does that relate to the bioactivity of that food?
So I think then you, on top of that, have to layer the fact that antibiotics are one of the most commonly used drugs in the community, as well as proton pump inhibitors, all of which have been shown to affect the microbiome. And depending on your antibiotic, you need to keep in mind that individuals, I think an interesting example is when very large studies were done with IV vancomycin in individuals with deep-seated serious Staph aureus infections. The IV vancomycin over time begins to leach into the gut enough to precipitate Clostridium difficile. So it's not an effect seen with short-term antibiotic administration, but that becomes an effect with long-term antibiotic administration. So dose, duration, frequency, and these drugs are very common. So it's very hard to think to it. I think it's a great question, but it becomes a very hard problem to study because of the complexity of care, just simple health care in the United States.
I want to just add one thing about especially the thing that Jeff said about bioactivity, and that is, it's absolutely true that we think of antibiotics at their therapeutic concentrations causing enrichment of resistance, but antimicrobials are a carbon source, right? They're a food source on their own. They have various biodegradation pathways, and there's a lot of rich literature in model systems, at least, to show that the breakdown products of antibiotics may not have antimicrobial activities, but they have sort of microbial perturbation type activities. And so that's another concern, which I think, again, the Twitter user could have been alluding to, that the way our diagnostics currently exist, and for instance, antibiotic use in dairy farming, there are very, very specific analytes that are analyzed based on what we think their bioactivity is. And so I think there is a huge amount of work to be done, antibiotics being only one of those chemicals, as to what happens to these along the pathways of biodegradation, sometimes microbially mediated. What do those breakdown products do to microbial structure? In fact, if you take the example of tetracycline degradation products not only can perturb microbial communities, but they can, in some cases, increase the frequency of horizontal gene transfer of tetracycline resistance genes, right? So it can get much, much more complicated than that. So.
I'm Cliff McDonald from the CDC, and I wanted to just bring up a question, and maybe we'll think about it tomorrow too, because this is kind of a big can of worms, and it's about translating to the clinic and translating to public health. I heard one question today, which was about something, I think it was in one of the food sessions, around, if we keep going this way, it'll be 30 years before we get translated. And I think that we had to keep in perspective here, I'm not talking about the IP issues around product development, drug development, or the regulatory hurdles either. Let's leave those. That's not the focus here. But just around these questions, and I'm a little bit worried, and it's a little philosophical, is because, back in the sequencing of the human genome, after that, there was a long time of development of getting anything translated. Now, part of that was the technologies, which were just catching up. But now we're on this steep trajectory of technologies, and you just see all this enthusiasm that we know we could answer and find functional answers to things. And they're always just a couple more years. And the question becomes for the research community, because remember, you speak to the industry in a way collectively, and that is to try to figure out where enough understanding is adequate to go ahead and do something. I mean, we've seen associations, we've seen some outcomes. I guess as long as something's repeatable over and over again and you have the desired outcome, do we have to understand it? I think there's something to just think about in how we start asking. I think the focus here was to define gaps. So I agree with that. We keep defining gaps, but maybe one of our gaps is to understand when something could go to translation. And where is that break point, so to speak? So maybe it's something we keep thinking about.
So this is Jen Fetways from VCU. There's a gap that was called out through email today, and this is something I'm going to read that came through from Thomas Schneider, who's a senior investigator at NIH. And this came mic ready, so I'm just going to go ahead and read. I've been watching the excellent human microbiome emerging themes at the horizon of the 21st century talks today. I've been analyzing a lot of microbiome data, but in many, if not most cases, the OTU counts are unavailable, hard to find, or are only partially published. Incomplete data, for example, top 20 species, is useless for careful data analysis. Availability of sequence data is nice, but analyzing it would add another layer of heavy computation. So a critical need is to have full microbiome OTU counts of raw data processed by the scientists. Simple CSV files, not proprietary binaries, should be provided so that all data are accessible and permanently available. And then there was a question that came in from Anna Edlin that was also related to data sets, and she's at JCVI. And here she said, when working with assembly of sequencing libraries from HMP, one realizes quickly that there are shallow libraries, sometimes only 3 million read depth. And she called out a need to sequence more in depth to understand the human microbiota and asked, can we set a common standard for sequence depth of community shotgun libraries representing the human microbiome?
I'll at least try to answer that last question. I think a lot of us have been saying the same thing. I think you need to define your resolution, you need to define your experiment based on the biology that you want to answer. And I would strongly resist any kind of community-enforced standard of sequencing depth, right? If I'm sequencing only the first few days of a premature infant when there are 6 species in there, I shouldn't be held to the same standard as someone who happens to have a remote hunter-gatherer microbiota with 10 times the diversity. And so I think what's much more important is for us to define upfront why we chose what we did, make the data available, I completely agree, so that other people can analyze it as they need to, and to do that at the raw data level rather than the process data level. And then, people can reanalyze your data, but I think this idea of predefining sequencing depth is dangerous.
And just on the simple point of making OTUs, there's several different ways to do it that give really quite different answers, and then you can pick different cut values for piling up your OTUs, and so there'd be a danger of, if you were pooling from several different studies, that they were made in quite different ways. So I'm afraid the annoying business of going back to the sequence data and making OTUs de novo is probably the best solution.
But I would just like to add, it would be good if journals would reinforce their typical policies of depositing and making sure the data does get deposited with associated metadata that's required to do the analyses. That is generally highly inconsistent in work that we're trying to do at present, and requires then months of effort to go to the authors and request the data needed to analyze.
Thank you. Brandon Eicher from Nutrilite. In my own personal research, I've been kind of embracing the idea that there is variability in the microbiome, and I think that's actually the cool thing about the microbiome. It explains a lot of the reasons why our experiments and our clinicals don't work sometimes. And in fact, I was in a conference for the ag microbiome a few months ago, and one of the things that they were doing was to get to the field faster. The companies that were going to real life situations, and in the situation would be humans in our case, we're actually getting to learn and understand and capture that variability and make products and get to market and actually solve problems by going down that path. So I kind of took that with excitement, and when we start to do clinicals for nutritional studies, it's right into humans, and the cool thing here is it's food, so we're not feeding things, dangerous things to people, and start to realize that we are all different. We all have different microbiomes. So, I can give you, everybody in this room, a single polyphenol or phytonutrient, and 30% of you will have a response and 60% of you won't. And that explanation comes from the microbiome. And I think the future of this space and the ability to personalize nutrition is to grab that by the horns and start to understand why it is that I'm different than someone else, and then start to formulate things or drive towards understandings in medicine that can start to tease apart those differences so that when we offer solutions to a population, it's not the follies that we have with drugs. Drugs typically have 3 cohorts. There's drugs that work for 30% of the population, drugs that sort of do nothing, and then drugs that actually hurt people in a negative fashion. So that was my general comment. I just think that instead of trying to wipe out the variation and normalize everything down, I think the cool thing here is to try to understand it at great depth, get to humans. I like Justin's commentary of like, let's start to figure out how these foods work in populations, and we can maybe solve some of these nutritional issues, both from the Western side of the thing where we overeat and consume lots of not food foods, to all the nutritional gaps that we see in other parts of the world. Thank you.
Okay. Yeah, with that, I think, comment? Sorry, Gary, go ahead.
Sorry, I just wanted to agree with that comment. Intersubject variability can often be viewed as a negative thing, as noise, but actually it actually can be very informative. So I showed you that principal cohort analysis of how people's microbiota changes in response to diet within 24 hours. It seems to be stochastic, but in fact that's actually quite informative. And an example of that would be Aaron Segal's work from the Weizmann Institute in making predictions about postprandial glycemic response. And the reason they picked that up was that there was a high level of intersubject variability in postprandial glycemic responses. And I talked to him about what we actually observed many years before about the intersubject variability in response to diet. And I think that was one of the features that was very meaningful in coming up with that postprandial glycemic response indicator, was exactly as you say, that intersubject variability.
Even though you might think NIH has plenty of money, we had a very limited budget for this workshop, and we become pumpkins at 6 o'clock. Otherwise, we get charged more for this room. Thank you, everybody.
Thank you, Lita.